I'd like to all the participants to please mute your.
Maybe it is my browser. Let me reconnect through Google Chrome. Maybe that will help with Google Meet.
But you are hearing absolutely fine.
Okay. Because I think my presentation will also be easier on Google Chrome. Since this is Google Meet.
Okay.
I'll join back in just a second.
Okay. মোনালিসা,
হ্যাঁ বল।
বলছি শান্তন জয়েন করে গেলে আমরা তাহলে শুরু করি স্ট্রিমিংটা।
হ্যাঁ উনি জয়েন করেছেন। Can you hear us, Dr. Bose? We can't hear you. I think you are muted.
Okay, can you hear me now?
Yes, we can hear you.
Yeah, I think it was a problem with the browser. There's also, I can hear everybody now. Nice.
আমরা কি শুরু করব?
হ্যাঁ, শুরু করো।
Good evening, everyone. I, on behalf
বাকি ডিভাইসগুলোকে মিউট করতে বল। বাকি সবকটা ডিভাইসকে মিউট করতে বল না।
all the participants, we request all the participants to please mute their devices. All the participants, please, can you please mute your devices? Otherwise, it will be very difficult for us. Thank you. Good evening, everyone. I, on behalf of the Physiology Department, Vidyasagar College, would like to welcome you all to this one-day international webinar on contemporary innovation and challenges in modern biology research. Our respected principal, Sir Dr. Gautam Kundu, will be unable to attend this webinar as he is preoccupied with some government meeting in Higher Education Department. However, he has conveyed his greetings and best wishes to all the participants of this webinar. So I would like to request our respected head of the department, Dr. Shubhadeep Ganguly, to inaugurate the event and to deliver the welcome address.
On behalf of the Department of Physiology, Vidyasagar College, it is my pleasure to welcome you all today for our one-day webinar entitled Contemporary Innovation and Challenges in Modern Biological Research, jointly organized by the department and IQAC. I welcome Dr. Shantan Bose, an eminent virologist of Autonomous Therapeutics and Maryland, USA, postdoc fellow from Harvard Medical School, USA. Dr. Bose is known to me personally for more than 2 decades. It is not only an usual webinar, but a nostalgic moment for me also. This is the second international webinar of Vidyasagar College conducted on a digital platform in line with the social distancing norms due to COVID-19 pandemic. It is a great opportunity for me to welcome our chairperson for scientific sessions, participants, delegates, and students. Thank you all.
Thank you. To begin with our first technical session, now I would like to request our respected former IQAC coordinator and senior most faculty member of our department, Dr. Alok Ghosh Chaudhary sir, to introduce our eminent speaker for the evening. Over to you, sir.
Thank you, Mona Lisa, and good evening, everybody. Welcome to the first international webinar in the sense organized by the Department of Physiology, Vidyasagar College. We are fortunate enough to have Dr. Sayantan Bose as today's speaker. Dr. Bose is now one of the leading virologists in USA, and he has published a considerable number of papers in Journal of Virology, Proceedings of National Academy of Science, USA, and other journals of very high repute. Thank you, Dr. Bose, for your hitherto achievement and best wishes for your all future endeavors. I strongly believe that your today's lecture will enrich us immensely. Lastly, thanks are also due to Dr. Gautam Kundu, our principal sir, for giving this necessary permission, and Dr. Subodip Ganguly, head of the Department of Physiology, and other teachers of the department for necessary arrangement. And with these few words, now I hand over the microphone to Dr. Mona Lisa Dey to continue the program. Well, thank you all.
Thank you, sir.
Welcome.
Now I would like to welcome and request our distinguished speaker, Dr. Sayantan Bose, to enrich us with his scientific lecture. Over to you, Dr. Bose.
Thank you very much for this lovely introduction. Thank you, Dr. Alok Ghosh Chaudhary. Thank you, Dr. Ganguly, for this great opportunity for me to present here. This is actually kind of a homecoming for me because my entire scientific journey started from and then I went forward. I was a student at the Presidency College and Department of Physiology. So physiology has been my background throughout. And I know that some of my teachers are also here. So with due respect, thank you everybody for making me what I am today. And especially for physiology students, I would say studying in physiology also gives you a very unique perspective to biology. And when you go into further in the future, that also gives you a very unique perspective where you can see the holistic view of the human body or the overall outside the molecular biology aspects. And then you can understand things at a much higher level. So with that, we're going to talk about a virus which is currently, let me see, I start sharing this. Okay, now I got it. Share. So are you able to see my screen with presentation?
Yes.
Okay. So this one now at the question how I do here. Start the presentation. So can you see the presentation on full screen?
Yes, it is full screen.
Okay, so I'm going to talk about this SARS Coronavirus 2, which is basically the virus that has been taking over the world today. And the 30 kilobases of RNA that I have in my title is actually the genome size of the virus. So to understand the virus, let's go to first understand what other viruses very similar to it behave like. And for that, we need to go to its family, which is the family coronaviruses. Usually, viruses are grouped into families which have very similar characteristics in terms of their replication, how they enter cells, how they infect cells, cause disease. So we can learn a lot when we study the family of these viruses. So coronaviruses were known to us from 1930s, and they cause minor diseases in humans and animals. Some of the first coronaviruses isolated during that time from the 30s and the 40s were the infectious bronchitis disease virus, mouse hepatitis virus, and transmissible gastroenteritis virus. So you can see just by the names themselves, these viruses were quite insignificant. And in the 1930s, there was the flu pandemic going on, the Spanish flu, which was 1918, that had killed over 1 million people. And so in terms of the importance of viruses, coronaviruses never really featured during those decades. But later on, as time went on, few more human coronaviruses that are even that still exist to this day, like NL63, OC43, and 229E, these names that you see here, have been infecting us for hundreds of years. And they also cause minor gastric and respiratory distress. And these viruses started to be identified. And so we'll come more and we'll keep on talking about these other coronaviruses over the presentation. But these viruses have infected pretty much 99% of the human population. So at some point or the other, you would have gotten one of these viruses as a small common cold or slight stomach upset or something like that. But only in 2003, the first SARS outbreak happened. And after that, coronaviruses first started to come into news. And then again in 2012, the MERS outbreak happened. And so you can see on the right here, when you group these viruses, all of the viruses from the coronavirus family, based on their sequences, on a sequence tree, on a family tree for the viruses, you can see that the SARS coronavirus, the original SARS coronavirus is here, very closely related to bat coronaviruses. The MERS coronavirus is here, very closely related to camel coronaviruses. And these are also, the MERS was in fact known to be transmitted from a camel. And these are all the human viruses, slightly separated from these. So then what happened that in spring of 2020, which we all know now, and the reason why I'm giving this webinar here, is that a new coronavirus basically brings the world to a halt. And once we zoom into that picture, which we saw earlier, we see again that these coronaviruses, these strains of the SARS-2 coronavirus, highlighted here in red from the original name, they are most closely related to bat coronaviruses, as you can see here. So these human coronaviruses, and they're these animal coronaviruses. And that brings us to a very interesting point and a very interesting feature of some viruses, which are called zoonotic viruses. Zoonotic viruses are those that jump from one species to another, and most common ones are ones that jump from other animals to humans. Or in fact, we are most interested in those ones which jump from one animal to us. So zoonotic viruses are not new. They have been infecting us forever. And whenever there is human and animal conflict, they have caused some of the worst viral epidemics and pandemics in history. So as you can see on this graphic here, viruses like Marburg, Ebola, Nipah, the first SARS, MERS, all of these different flus, bird flu, swine flu, they have all come from animals, and they are all zoonotic viruses. They started out their life as some animal virus, and then due to mutations, they gradually moved into humans. And one thing you'll notice in this graphic is that a lot of these animals that the viruses come from are bats. Now, bats have a very unique biology that prevent them from getting sick. Their body temperature is extremely high, and their metabolism is extremely fast. So as a result, the viruses cannot take over the bat cells that easily and kill them. And as a result, the bats can carry the viruses for a long time, but they do not get sick themselves, and they are that's why they are a transmission agent for these viruses. And SARS coronavirus probably had an intermediate animal host between bats and humans. So as virologists, we've often pondered like what would happen in a scenario like this? How would a virus actually achieve this pandemic potential, that something that we call a pandemic potential? And how did SARS coronavirus do that? So it is a respiratory virus, and the fastest propagating virus is usually through droplets and aerosols from respiratory, through sneezes and breathing. So it becomes a super fast spreader that way. It has to be an RNA virus, which is a reason because RNA mutates much quicker than DNA. And we'll go a little bit more into detail for that just when discussing the genome of the virus. And then this is an important and interesting point, actually, point 3, which says that the virus should not have a high mortality rate. This means this is interesting because if the virus has a higher mortality rate, then the host is gotten rid of really quickly. And as a result, the host does not have enough time to spread to the next person or the next animal. And so as a result, viruses like Ebola, which have very high mortality rate, have killed a lot of people and have caused epidemics, but only in localized places. It's because they kill their host really quickly. And then the 4th point is transmission without symptoms. So obviously that means that people do not know that they're infected, and so it's easy for them to infect others. And then the ability to jump between animal hosts. And we talked about how they are zoonotic and how they came from animals. But in fact, for this virus, cats and dogs and other animals have also been infected from humans. So it is constantly has this ability to jump species. And then besides these biological reasons, there's of course social reasons. That highly connected world in the 21st century, lots of flights, people traveling all over the world. There are megacities that are close living quarters for millions of people. And then unpreparedness of governments and inability to accept scientific facts is always a factor in these cases. So let's go more deeper into the biology of the virus and what contributed to it becoming the worldwide pandemic, some of the points that we just discussed. So here's a picture of a group of electron micrographs of MERS, SARS, the SARS-2 virus, L63, 229E, and murine hepatitis virus. As you can see right away, they all look kind of very similar to each other. They all have this appearance of these things that are sticking out from their sides from all around. And they give an appearance of a corona or a crown. That's how the name coronaviruses came about. They are encased by a lipid bilayer, which is thus the same as a plasma membrane, as you can see here in this image. And the main glycoprotein or spike, that is what you see it decorating the outer side of the outside of the virus. That is the spike glycoprotein, or S. S is the short term for spike glycoprotein. There are 2 other membrane proteins, E and M, which are also present on these viruses, but they're usually not visible in these electron micrographs. So when we look a little bit more detail into the spike glycoprotein, the main glycoprotein that is used by the virus to enter cells, it kind of, this is a cartoon of the virus. So here's the membrane, and you see the spike glycoprotein. Unusual for this group of viruses because all other viruses, which are kind of similar with membrane-bound viruses, they have glycoproteins on the surfaces as well. But these are really big. These proteins itself are really big. So the SARS glycoprotein binds to cellular membrane proteins on cells, and those proteins are known as receptors. And so the cell obviously does not make proteins that it will invite a virus in. So it does not express proteins on its surface waiting for the virus to come in. These are proteins that the cell uses normally for its own purposes. The virus has just adapted to use these to enter cells. They have been usurped by the viruses, basically. And so as a result, there are different coronavirus spikes that can bind different receptors. For instance, there's a list down here. The two SARS viruses, NL63, bind angiotensin-converting enzyme, ACE2, which you would have heard about. MERS binds DPP4, and OC43 binds sialic acid. Flu also binds sialic acid. Almost all strains of flu bind sialic acid as a receptor. So you can see that the different virus families can bind the same receptor, and different viruses in the same family can bind different receptors. So there's a lot of variety amongst these. And ACE2 is not the only receptor that is being used by SARS viruses, because NL63 also uses it. So then, now that we know a bit more about the spike glycoprotein, let's look into why it is most successful in infecting our respiratory passages and our lungs. So the spike glycoprotein actually requires a proteolytic cleavage in order to be activated. So a certain protein, if you have to cut a certain part of this protein by proteolytic cleavage, and then the protein refolds in order to cause fusion between the cellular membrane and the virus membrane. So that's why this cleavage is absolutely important. Without it, the protein is inactive. So the cleavage site of the SARS protein is such that it has evolved to be cleaved by a resident tissue protease called TMPRSS2. Now, this is a very important protease that is present in the lung, including a lot of other proteases in this family, number 2, number 3, number 4. But specifically, number 2 can cleave the SARS protein cleavage site. These are tissue proteases, so they are actually present outside the cell. So they are all extracellular proteases present inside the lung tissue, lung interstitial space. So what happens due to the presence of TMPRSS2? In cells that don't have TMPRSS2, which is shown on the left here, the virus has to bind its receptor as usual, going through an endocytic pathway. It goes into the endosome, then into the deeper late endosome, and then into endolysosome. So where it is actually converting the endosomes into lysosomes. Now, at this point, the pH of the endosome gradually drops, starting from the point that it enters. And in the lysosome, it's extremely low pH. And at these low pHs, there is another protease called cathepsin, which can only cleave at very low pHs. This cathepsin can also cleave SARS-2 spike. So so that's why the virus has to go all the way inside, very close to the lysosomes, in order to reach this protease to get cleaved and activated. But in cells which have TMPRSS2, they can be cleaved right outside the cells because it's an extracellular protease. And so the spike protein is active, it enters, and it can immediately release its genome from the early endosome itself. This gives a big advantage to the virus, because if the virus has to go all the way into the lysosome, then we know that there are a lot of damaging enzymes in the lysosome which are used to kind of destroy microbes and other other extracellular foreign material. So that's why the virus can absolutely avoid that happening if it gets TMPRSS2 at the surface. So the virus uses both of these pathways to enter cells. One other thing that one other thing about the protease cleavage site is that for SARS-2, this protease cleavage site is polybasic, which means it has a number of basic amino acids, as you can see here, a number of arginines, RRR. And these cleavage sites are present in a bunch of different viruses, not just coronaviruses. They are also present in highly pathogenic influenza viruses. And they are easy to cleave and activate. So they're much more easier to cleave by cellular proteases. So the one common thing between all viruses that have polybasic sites is that they are usually highly pathogenic. And that's usually a sign for a virus that it is pathogenic. It could be very easily activated. There's another thing that the ACE2 binding site. Now this goes a bit more into how the virus infects various different animals, because various different animals have ACE2, the same protein that we have, and the sequence of that protein is quite different. But this sequence alignment shows the binding of a binding site of ACE2. So this is spike 2 of SARS protein, and this is binding of and this is the region that binds ACE2. So you can see that is quite similar, and a very few evolutionary amino acid changes can change the binding specificity of spike to human ACE2 from bat or some other animal. And then again, it can change from human to some cats or dogs that have been infected, and that's most likely what happened in those cases. So that region is very similar. And the other thing is that on the left, if you see this diagram, this shows a list of different organs and the expression of TMPRSS2 protease in these tissues. And what I've highlighted here is lung. So lung is one of the tissues that expresses this protease at one of the highest levels. And on the right is the expression of ACE2 in different organs. And you can see here that lung also expresses ACE2 at good levels. So combining these two, the lung kind of provides the perfect environment for the virus. It has ACE2, its receptor, and it has TMPRSS2, which is the protease that needs to cleave the virus to activate it. So that's why the lung is a really good place for this virus to infect, and it happens to be a respiratory virus. So now that we know how the virus kind of enters the cell and starts off its infection, we're going to go a bit more in detail into the genome, the positive sense RNA genome, and figure out like what aspects of this genome contributed to the zoonotic jumps between species and generation of new variants. So if you look on the top part of this figure here, there are the entire viral genome is about 30 kb in size. It is the largest genome of any RNA virus, and it is single-stranded, and it is positive sense. So positive sense means it is the same sense as mRNA. Just like DNA has Watson-Crick strands, as we know, they have positive and negative strands. So RNA also is characterized the same way. So this is positive sense. And 2/3 of the genome on this side, highlighted in these blue bars, they are non-structural proteins. Non-structural proteins are those for virus which kind of enter cells, which get made in cells, and they help establish the infection. So they are replicates. They replicate the genome, they build structures that a virus requires to replicate, and they are usually not incorporated into the virus particle itself. On the other hand, structural proteins, which is the remaining 1/3 of the genome, are grouped together here. They form all the structures and all the different proteins that the virus needs to create its own, to create progeny and progeny particles. So, and then it's a single-stranded, as I mentioned. So how do these viruses mutate so rapidly, and how all these variants arise? So nature is constantly playing roulette with viruses, and RNA is a major determining factor for that, because RNA can be mutated much more rapidly than DNA because of its chemistry. And so a virus with an RNA genome like Sars Coronavirus 2 can mutate naturally at a rate of 10⁻⁶ or 10⁻⁴. What it means is that every time the virus replicates, there's a chance that there would be one mutation coming up in 10⁴ or 10⁶ nucleotides of the entire viral genome RNA per infected cell. So that means that a virus of 30 kilobases can have, every time it replicates, it can possibly have maybe 1 to less than 1 mutation for every replication cycle. And once a virus enters a host, it replicates many millions of times. And also the other factor is this is a single-stranded RNA genome, so it does not have a complementary strand that can be used to correct errors, which we know happens with DNA, and there's like the Watson strand and the Crick strand, and one strand, there are repair mechanisms that can correct errors during replication based on the other strand. So these mutations are spontaneously, and they're constantly happening in billions and billions of viruses and in millions of hosts. And so when the mutations are happening across the entire genome of the virus, they are also happening in the spike protein, the part of the genome that encodes the spike protein. And so it's happening in the same region which I just showed, which can bind receptor. So when that happens, it keeps introducing mutations. And so bat-like viruses for SARS-2 can change their specificity to human SARS viruses as well. But the good thing is that it does not mean this constant mutagenesis. It does not mean that the virus, all the mutations make the virus more pathogenic. The saving grace is vast majority of mutations are bad for the virus. Because, if you mutate a protein, and we constantly do make mutations in lab for various proteins, for cells and other viruses, etc. But like even single amino acid changes often just completely change the folding of the protein, and as a result, the protein is degraded. So the same thing happens with viruses, and vast majority of mutations are bad for the virus and makes it less effective. And this mutation, this evolution, we are actually seeing it happen in front of our eyes, because if we sampled virus samples from infected individuals from the early part of the pandemic to the late part of the pandemic now, we have seen certain virus species with a certain mutation take over. For instance, this is still like pretty old data now, but this paper analyzed the mutation D614G that came up in the spike protein, and this was found to have increased infectivity but not pathogenicity. And very soon, within February to March in 2020 last year, the entire population of viruses was taken over by this new strain or this new mutation. So the more the virus gets a chance to replicate, the more its RNA genome can pick up random errors, and more it can spread. And as a result of this, WHO has currently designated these variants of concern, which we have heard about quite a lot: alpha, beta, gamma, delta. And so all of these are more infectious, but so far they have not been found to be more pathogenic. So the thing is that sooner we can stop this transmission between individuals, sooner we can put a stop to this pandemic, lower the chance of getting new variants. Because if the virus gets to replicate more, it will produce new variants because of that chance of creating a chance of changing its RNA. So then a bit more detail into the coronavirus life cycle. How do coronaviruses establish infection in cells? So we've talked a bit about the entry of the viruses, the spike protein, how it uses the receptor to get into cells, how it's using TMPRSS2. All of that is here. And then we see the virus releasing its genome into the cytoplasm of cells. And what the genome does right away is it starts translating these, the first 2/3 of the genome, which is the part which is the non-structural protein section. And then once you, this is a busy slide, but I'll try to go step by step. So here is the entire genome. The virus enters, as you can see here from the corner, and immediately starts making these non-structural proteins by translation and by proteolytic cleavage of a large translated section. And then these proteins that are made, these non-structural proteins, start building this entire replicase complex, or something that we know call replication factories. And here's a picture of this, an electron micrograph of these real replication factories in cells. You can see how the virus has kind of built these membrane-bound components inside cells, and they are concentrating all their proteins in here to replicate. So once they build these, their genome is being replicated, and more viral proteins are being made. The structural proteins are made by a very unique way. So I mentioned that the genome is positive sense, which means it's the same as an mRNA. But the weird thing is, it only uses this part of the first 2/3 of the genome as an mRNA, but the remaining 1/3 of the genome, it does not use as an mRNA. It uses it makes the negative strand first, little bits of negative strand RNA, as you can see here. These are known as small genomic RNAs. And each of these small genomic RNAs now make an mRNA, which then makes the protein. We don't know why the virus does this, but this is how the structural proteins are made. Probably a way to distinguish between non-structural and structural proteins and to time it. So non-structural proteins have to be made first to create this complex, and then the structural proteins come in to make the virus. And once the structural proteins have made the virus assembles and it comes out and it takes along the cellular membrane from an internal compartment from the Golgi or ER. And that's how the virus gets its bilayer membrane, which surrounds the virus. So this takeover of the cellular machinery, establishment of replication factories, all of that severely hampers cellular level physiological functions. And now, so we'll go a bit more into the pathogenesis and more of the pathophysiology of SARS and see what happens during, like, what causes the disease. So we all know now that the disease caused by SARS-2 is coronavirus disease of COVID-19. And multiple organ systems have been shown to be affected by COVID-19, including the respiratory effects. It has cardiovascular effects, gastrointestinal effects, neural effects, and more increasing number of systems are being found where there are clinical manifestations. So it has a pretty wide systemic effect overall, the disease. We are going to look at some of these aspects and what is known so far about the physiology and biology of how this happens. So how does COVID-19 damage the lung? So we know that alveoli are structures in the lung that help exchange oxygen and carbon dioxide. And so these alveolar epithelial cells that line the edges of the alveoli, they express the ACE2 receptor, which binds SARS-2. So the virus can enter into cells, into the alveolar cells. They are kind of residing all along the alveoli here. And it starts replicating. And as a result of this replication, certain pro-inflammatory molecules are released from these cells, which then recruit macrophages and neutrophils, cells which are normally brought in when there's tissue damage. So when there's tissue damage, the cell macrophages come in and they chew up the damaged cells, the damaged debris, and so these cells are. It is kind of signaling that the tissue has been damaged in the lung. The virus somehow signals that, and we'll see a bit more into detail how it does that. And then as a result of all of these cells coming into these small interstitial spaces in between the alveoli where the real gas exchange is happening, they kind of cause blockage in this region. So gas exchange is physically blocked, and they also cause inflammation and by filling them with these cells. And the patient, as a result, can't exchange the oxygen and suffocates. So this is one of the major manifestations and one of the major clinical reasons why we have seen so many people going on ventilators and eventually going to their death in a lot of cases. So going a bit more into detail about how the virus mediates cytotoxic effects, well, we can see there's, we talked about the lung alveolar epithelial type 2 cells in particular in the alveoli, which can be infected directly by SARS-CoV-2. But there are also a bunch of different cells from other organ systems, a lot of them from the respiratory system, some from the GI system, there's some from the pancreatic system, renal system, in the kidney, and all of these cells, when they're directly infected by the virus, can cause significant cell damage, as I just described. We just have time to discuss the lung part, but we'll talk a bit more about the other systems in a bit. So here's another way that the virus can also affect another system in the body. So ACE2, so as I had initially mentioned at some point early in the presentation, that cells never make proteins to invite viruses in. Cells never make receptors for viruses. They are proteins that the cell normally uses for other purposes. So ACE2, the protein that the virus uses as a receptor, is actually a membrane-bound aminopeptidase. And it is a potent counter-regulator of the renin-angiotensin-aldosterone system that regulates the fluid and electrolytic balance of the body. It also has roles in blood pressure regulation, vascular permeability, and tissue growth. So you probably would have learned in your classes for the students that angiotensin converts to the angiotensin 2, and then there are other conversions that happen with angiotensin 1 to 9, angiotensin 1 to 7. The ACE2 is actually important for these conversions. And so it regulates the amount of angiotensin present in the blood. So when the virus co-opts these molecules, the ACE2 molecules, they actually downregulate these. And as a result of that, the system is disrupted, and these conversions cannot happen. And these have big implications for heart, liver, and kidney diseases. So what happens? Let's go step by step, try and understand what happens after virus infection. So first, virus infection occurs. We've talked about this part. And then there's this process called pyroptosis, where when the virus replicates inside cells, the cells release certain molecules, including ATP, nucleic acids, and ASC protein oligomers, which are very distinct indicators of inflammation in the body. So which is basically telling the cells that there has been, telling other cells that there's been inflammation happening over here. And when these small molecules are released, neighboring cells like epithelial cells, endothelial cells, and alveolar macrophages senses them using the receptors on the surface, and then they start releasing pro-inflammatory cytokines and chemokines. Now, these pro-inflammatory cytokines and chemokines, they further attract monocytes, macrophages, and T cells to the site of infection. These are migrating monocytes, macrophages, which can actually infiltrate tissues and go inside and then attack certain parts of the cells. So, and then on the other hand, these migrating cells are also releasing a lot of these cytokines as they go in, and they try to get rid of this foreign material. And this feedback loop leads to something that is known as a cytokine storm, which you would probably have heard about. So there's a huge amount of cytokines released. All of these proteins come in, they infiltrate whatever tissue is infected, be it lung, we talked a bit about the lung, be it kidney. This also happens in the GI tract as well, causing GI symptoms. And this may lead to pneumonia, edema, widespread inflammation, and multi-organ damage. So that's how this pathogenesis, the pathophysiology of SARS-2 happens. And it's still not clear like how this virus particularly triggers this system. And so what happens is the body's own immune system ends up damaging more than what the virus can do all by itself in damaging the few cells that it infects. So eventually, COVID would manifest in a wide variety of systemic pathologies, including neurological, cardiac, endocrine, renal, dermatological, GI, etc. So we obviously won't have time to go into all of these, and each of these symptoms are being found now, and they're constantly being added upon as newer, more and more data is being generated. So it is quite like a big systemic disease. So now that we've talked about the bad parts of the disease, so here are the good things that we of course have very good COVID-19 vaccines. And so let's see how our immune systems are working after vaccination. So this is something that is very basic, but just as an introduction, you would probably also already know this, that the antibodies are extremely specific. And so every antibody has a specific region of a pathogen or a specific antigen of a pathogen, which could be, usually it's just like a surface protein. And so there are unique antibodies that bind to specific measles surface proteins, unique ones that bind to flu surface proteins for chickenpox. But when a new pathogen like SARS-2 comes in, there's no antibody that is generated in the body. So at that point, the body has to generate new antibodies, and then that has to be expanded in order to protect the body against a future infection by that particular virus. So antibodies are generated by both virus infection or by vaccination. And certain cells in the blood, they sense the new antigen coming in, and these are usually naive B cells. So B cells which have not been exposed to any antigen before. So the first infection happens, the B cells are activated, which means that they have sensed an antigen. They have been presented with a small bit of the antigen, which could be a part of the spike protein or something else. And they start producing antibodies directed to that particular part of the spike protein. Each B cell will do that. So a wide variety of B cells will get small bits of the entire spike protein. So there's a wide variety of B cells that will produce increasing amount of antibodies, an increasing variety of antibodies against the spike protein. And then, so after some time, the expansion of B cells happens. More and more of these B cells start getting produced. And then they constantly start releasing these antibodies into the blood. There are different types of antibodies. IgM subtype of antibodies is the first one that's released. After that, IgG is released. So, but what happens is that after some time, the level of these antibodies go down as the patient is cured, as the virus is removed from the body, and after a few days after vaccination, this starts to happen. That the level of antibodies circulating in the blood goes down. But on the other hand, there are these long-lived plasma cells that are present in the bone marrow. They are known as memory cells, and they can live in our body for many, many years. So every infection that has ever happened in our body has been stored in some of these memory B cells, or the particular peptides that the infection produced has been remembered by these plasma cells in the bone marrow. So what happens when a repeat infection happens? A repeat infection or the second dose of a vaccine. These memory B cells start replicating and making more and more B cells, which can make that specific antibody for that specific antigen. And then the next peak of these plasma cells, the secondary antibody response, is much, much higher. And as a result, the overall amount of antibody production is way higher. And plus you have more memory B cells, more memory B cells as well overall, which will then stay in our body for years and years. So that's why the second dose of the vaccine is very important. So not only B cells, the body, there's another second regiment of the body, which is the defensive regiment of immune cells called T cells, which can target and kill or destroy specific infected cells, getting rid of the sources of virus. So they can do it by phagocytosis, they can do it by cell destruction, releasing certain cell-destructing molecules. So these are the T cells which we had talked about before, which actually damage our tissues. But when they are directed against a particular spike protein, they can specifically target cells that have been infected by SARS-2 and destroy them. COVID-19 vaccines have been shown to have a very good T cell response, which is very good and T cells also have memory T cells, which can last for a very long time. So we do have very good protection from these cells, and this should stay for years to come. However, occasionally we might need boosters, so not everybody might be generating these at the same levels. There'll be exceptions for immunocompromised individuals. So there would be booster doses that will probably be needed. But otherwise, the response of antibodies to these vaccines is pretty good. So the question arises, so if vaccines and infection by the virus both produce antibodies, why do we need the vaccine? So the first reason, obviously, is you do not encounter potentially fatal or sickening disease, but you still get the antibodies. You do not have to face the bad part, but you get the good part. And then the booster or the second dose of the vaccine, as we just saw, can make an optimal boost to give sufficient amount of memory cells and keep us protected in the future. So you are not probably going to get infected again, unless you're unlucky. But if that happens, then you probably get a suboptimal level of dose. For the vaccine, you know that will give us a very strong dose of antigen that will make more and more antibody for the second dose as well. And also the other issue is that suboptimal immune response can often trigger a severe condition known as ADE, or antibody-dependent enhancement. So in that case, oftentimes the next infection is actually even more serious. And so, but the vaccine produces a much better immune response, which is much stronger with a higher antigen dose, which minimizes this risk. So then we ask the question, so we've talked about the virus quite a lot. So what do we need to achieve to end the pandemic? And how will we deal with new variants of SARS-2 in the future? So let's see how vaccination protects a population. So when no one gets vaccinated, the disease can spread easily. But when some people get vaccinated, there's kind of a ring around an unvaccinated individual, then the spread is slower. The virus finds it less easier to move. But when most people get vaccinated, the disease cannot spread. So that's the basic principle of vaccination. But we've heard a lot about herd immunity. So what is it? So I found this really good mathematical model that actually shows how different populations, depending on how much vaccination has happened in that population, can get infected by a virus. So we have 6 populations here in this figure: 0, 25%, 50%, 75%, 90%, and 90%, and 95% vaccinated. So the unvaccinated individuals are blue, and the vaccinated individuals are yellow. And the infected individuals, let's say the infection starts here, is red. So in the next slide, we are going to see in each of these individual populations how the virus can spread and what happens when one of the individuals are infected. So this is how the 6 populations look like. So when there's nobody who's vaccinated, the virus can spread extremely fast because it can reach every neighboring individual. But as the percentage of vaccination increases, it finds it even more and more difficult to reach individuals that are not vaccinated. So as a result, you can see 90% of vaccination, like literally there's no transmission. So to get to this stage, we at least need to get about 75% of vaccination benchmark in order to be safe and stop the spread of COVID-19. So what does the future hold for us, and how can we better detect pandemic potential viruses? So this is something that is really important, and virologists have been studying these for many, many years, but this has never gained that much importance, which it should have had, that virus surveillance, this field, it involves sampling of zoonotic hosts like bats, mice, and others. They obtain the droppings from these animals, then sequence them, get the sequences of various different viruses that could be there in these animals, and they look for novel viruses from all different families. And I particularly start off with showing this report that was published. It was publicly published in 2012 about a study where they studied bats in the Hubei province, in China, where the first infections of SARS-2 happened, and they could find a wide variety of bat coronaviruses. That's the biggest population lurking in the bats in that region. So, I mean, this is a very clear indicator that we would have, we should have understood and kept an eye upon, like what kind of coronaviruses could have come out from there. And in fact, it happened that the SARS-2 came out from a bat coronavirus, or evolved from a bat coronavirus. So all of these studies are, they have previously been ignored, but now they need to be really understood, and then these places need to be very carefully monitored. And it's not only in China. These studies have been happening all over the world. One of my favorite ones was published in 2011, and this encompassed all the continents, samples from all continents, and they found literally every family of viruses from every family in bats. So again, like as I mentioned in the beginning, bats have very unique biology, which allows them to harbor a lot of viruses. So then there are other studies from Kazakhstan, Vietnam, US and Canada and North America. So all of them have detected novel paramyxoviruses, novel coronaviruses. So there's a potential that the next pandemic might just come from one of these virus families or one of these viruses that happens to jump into a human. So there's always that possibility remaining. But it is good to understand what exactly is out there in animals and what exactly can be pandemic potential. But how there are millions of these viruses and there are millions of things that are getting discovered every day. How do we know which virus amongst all of these are actually pandemic potential? So here there's a evolutionary mathematical modeling methods that have come in. These basically scientists and mathematicians are predicting changes in sequence of the virus which could be potentially dangerous. For instance, you could look at the receptor binding site of spike, and you can see, so you can right away tell when you look at the receptor binding site of spike protein of coronaviruses, that area is important. So any changes in that receptor binding area, it could be a potentially dangerous virus. Similarly, it's like you could go into a bit more complex details about the mathematical modeling, but all that they're trying to do is to identify what kind of sequences are happening, sequence changes are happening the most, and what these mutations could lead to, whether it will be something benign or whether it will be something that can have increased pathogenicity or increased spread of the virus. And I'd like to show one of the success stories of this kind of studies is that a new strain of flu was found in China very recently, actually last year, which has the potential to become a pandemic. So instantly, CDC and other agencies have taken an action to screen all sorts of animals, all sorts of populations, looking for these kind of viruses with pandemic potential. So scientists have already figured out what the virus could be that has a pandemic potential. And so if we know that, we can already start developing therapeutics and vaccines against these viruses. So our advantages over the virus is that we have highly effective vaccines. They produce good antibodies. We have long-term protection through memory cells. And if some highly resistant variants arises, which these vaccines don't work against, we can make a booster vaccine shot pretty easily. But the virus does have advantages over us, which mostly deal with our carelessness and taking the virus lightly, problems in vaccine distribution and administration, and vaccine skepticism and fake social media news, so which we'd always be careful about. These factors will basically decide how well and how quickly we can beat COVID-19. So with that, I would like to end, and I would like to specially thank Dr. Gautam Kundu, principal for Vidyashara College, Dr. Shubhadeep Ganguly, for all contacting me and for the invite. It has been a pleasure to present to physiology department and to the physiology community. And I would like to really thank you, the entire audience, for your attention.
Thank you, Dr. Bose, for your informative and elaborate speech. Now the session is open for discussion. If anybody wants to ask anything, then you can please unmute yourself and please proceed.
Dr. Bose, I am Alokh Chaudhary. I would like to ask you a question. In one of your slides, you have shown that the tissue titer, that the titer of ACE2, angiotensin converting enzyme 2 receptors, titer is next to that of lung tissues. Okay.
Mm-hmm.
So prostate gland comes next to lung tissue as far as the ACE2 concentration is concerned. But amazingly, prostate fluid secretion has a acidic pH. Now the question is, coronavirus can infect prostate glandular tissues, but how the coronavirus is how do the coronaviruses get rid of the acidic environment so as to infect the prostate glandular tissues? What is your opinion?
So one of the things that it requires also is the cleavage. So when the spike protein refolds, it is dependent on the pH, not just for its refolding. It is dependent on the pH because it needs the cathepsin enzyme. So it is actually so unlike flu. So flu is different because you can lower the pH and you can have the
It shares sialic acid as a receptor protein.
And you can and the protein also refolds in presence of pH, just in presence of pH. It doesn't require any other, just the cleavage and then pH. But here the SARS spike protein is slightly different because even if you lower the pH but you don't cleave the protein, it will not refold. So the virus remains safe in this acidic environment.
Okay.
It is only active when that cathepsin enzyme is there, and that is usually not present outside the cell in tissue fluid of prostate. So that protects it.
Thank you. And my last question is, in most of the papers, we have found that the antibody titer following corona infection is very short-lived. Not titer, lifespan of antibodies is short-lived. But how the lifespan of the antibody can be enhanced through vaccination? It is not so much pronounced as we can expect. The lifespan of antibodies cannot be increased. To that extent, it can provide protection at least for the coming 3 or 4 years. So there's a paradox in the problem. How could we solve it? But how could we address the problem?
So when the antibody titers are measured and the B cell amounts are measured, usually because the memory B cells, they remain inside the marrow. So they are, it's very hard to detect those. So there are ways to detect it, but it's usually later on, a long-term infection or later, in many years later, it will be the memory B cells that will expand out and propagate. But the problem is, of course, if there's less number of circulating antibodies, you can get infected. But after that, the memory B cells start making more of these, and then the infection is taken over. So you will probably be safe, like you will not suffer, you have severe disease, but you can still get infected. That's the main problem. So probably what will happen is that there would be like a booster shot every few years, like we have for tetanus every 10 years. And then measles usually has very long-lasting immunity. So certain viruses have longer and certain viruses have lesser. So this seems like one that has lesser immunity in circulating antibodies.
I dare to ask you the very last question. Do you believe that the vaccines which are found in markets right now all over the world are underprepared? Do you believe in this dogma? They are underprepared.
No. So actually, I don't think so, because the vaccines, they are built on very simple principles, all of them. And they are, for instance, the mRNA vaccines, the vaccines, they are encoded by LNPs or the LNP particle. And the LNPs have been studied for about 30 years. So we know individually that the LNPs are safe. We know that mRNA treatments are safe. So it's just a matter that they combine the two. So a lot of these things the platforms existed before for some other diseases, treatment of other diseases, but they've just introduced the spike protein or the spike protein RNA or something in that and combined the two approaches. So yeah, of course, the vaccines are novel. They are new. They haven't been made like this before, but the original approaches were there.
But we don't have any highly efficient HIV vaccine. Although the HIV is RNA virus, we don't have still a highly efficient HIV vaccine.
Yeah, HIV actually has, because of HIV has other issues as well, because HIV, once it infects a person, it goes into these niches in the body. So it's very hard to eliminate the virus because it becomes dormant. And it also incorporates it's RNA virus, but it incorporates it makes DNA. It's a retrovirus, so it makes a DNA and then incorporates into the human genome. itself. So this the coronavirus doesn't do, thankfully. So because of that, then the cells themselves start producing HIV proteins.
Its incubation period is very high, 8 to 9 years.
Mm-hmm.
Okay, thank you.
Yeah.
Thank you, Dr. Bose. Thank you.
Sure.
Any other questions, Mona Lisa?
Yeah, I have another question. Can I ask a question?
Yes. Sure.
Dr. Bose, I'm Chandana Sharkar from the same department with the Sharkar College. Thank you for a very lucid and informative lecture. We are really enriched for it. And I want to ask a simple question. Might be I missed that the virus is being taken up by naive B cells. That is what you said might be. And is it T helper cell independent for making humoral antibodies?
No, actually, the T helper, I think it's the CD8 T cells that are important.
And if it is CD8, then that means what I want to say that since the virus stays in the circulation for such a short period, it internalizes and infects new cell. Will the cell-mediated immunity be more responsive or more important for protection against this virus? Means the humoral immunity, the vaccines part we normally always talk about the humoral immunity. That means we say the antibodies are normally taking part of the getting rid of this virus. But will the cell-mediated immunity be better off? Means the vaccine, whether the vaccine is taking care of the cell-mediated immunity part also?
That's a very interesting question, because the T cell, it is true, what you said is true, that it will probably be more important. But on the other hand, the major, like the inflammation part and the cytokine release part is what is causing the damage in these tissues. So these cells are also involved in causing that damage. So when we are designing therapeutics, it's like we would like to enhance T cell immunity or maybe T cell activation. But on the other hand, this might also affect how much damage the tissues are getting. So
that the inflammatory response is increased so much that shows that the cell-mediated immunity is taking a huge role. That is why all the cells that are associated with this, they are rushing there and they're causing all the cytokine release, and so that's getting a problem. Another thing is the bat part that is normally that my question is there for a long time, since they are harder reservoirs for this virus. And since when the reservoirs, they're using the cellular machinery for its multiplication, but at the same time, it is not pathogenic for the bats. Means don't they have any way of infecting the bats? Means do they escape the infectivity somehow? Has any work been done by that?
It's kind of similar to what our body reacts, and in like whenever we get an infection, the fever happens, like increasing of body temperature to reduce the. So bats constantly have a body temperature which is at the level of fever for us. So for mammalian viruses which replicate at 37 degrees, when it goes into bats, it's kind of like at 39 degrees always at fever level. So that's why they replicate at that level.
Means what I want to say, the virus somehow it's a reservoir. It means it's multiplying within the bat, but still the fever is taking part of the which part? Means it's not infecting it. Fever, that means the high temperature is killing the optimal load of the virus, decreasing the load of the virus.
Keeping it in check. Yeah, it is keeping it in check. Like that is exactly what our body's fever tries to do.
Yes, that's true.
Constant fever level does that for bats.
Okay, thank you so much. Thank you so much for your answers.
Actually, there are a few questions in the comment section. So if you allow me, then I can read one or two to you.
Sure.
Okay. There is a question from Dr. Barnali Ray Basu. What would be the possibility of severity of neurodegenerative diseases as a post-COVID threat?
That I don't know much about that actually. So I haven't. My part is mostly like a basic biological part. So I haven't read more into the clinical part for neurodegenerative diseases. So unfortunately, I won't be able to answer that.
Okay. There is another one from Orno Banerjee. He is asking, recently some immunobiologist flagged some risk linked with plasma therapy to combat with COVID-19. So if plasma therapy fails for some patients, is there any chance to trigger the infection by antibody-dependent enhancement?
It is, I briefly talked about antibody-dependent enhancement there. So after viral infection, if the dose is sublethal, or not sublethal, it's suboptimal to generate the level of antibodies and the level of T-cell response, then certain, like the virus itself, can be taken up using the antibodies that are generated. So. There is definitely a possibility of AD happening if the person gets infected for the second time. But with viruses, and it can also be a problem if a vaccine dose is suboptimal. So that's the reason why it has to be regulated, so that the vaccine dose is very high, or high enough to generate the amount of antigens that can trigger the response.
Okay. Thank you, Dr. Bose. I would now like to request our respected HOD, Dr. Shubhadeep Ganguly, to formally convey our gratitude and thanks to Dr. Bose.
Practically nothing to say. Outstanding presentation with very lucid diagrammatic analysis, so that all of our students can understand your today's presentation. Thanks a lot, Dr. Bose.
Thank you very much. Thank you again.
On behalf of this department, I'd like to convey my sincere gratitude. And now I request Dr. Chandana Banerjee Sarkar to inaugurate our souvenir for this webinar. Mona Lisa, I'd like to request you to please share our souvenir.
Yes.
The screen.
This is our souvenir. This is the top sheet.
Okay, so these are the abstracts from the scholars who are involved with modern research, modern biological research. So I think that the abstracts will enrich the others also. Okay. So it will be a pleasure for me to go through the abstract sometime.
Yes, there are quite a few. And these are for the oral presentations. And now these are for the poster presentations.
Mona Lisa, how many posters are there?
I think there are. Sir, let me check once.
I think 14
14.
posters are there.
14. We
14.
must congratulate all the participants on behalf of the college, because they are newer enough, and that's why they must deserve congratulations. Okay, so start the program immediately.
Now, finally, on behalf of the department, I would like to share the certificate of appreciation to Dr. Sayantan Bose. Mona Lisa, kindly share the certificate.
Okay, just a moment. Sharing.
Thank you, Dr. Bose.
Thank
We
you.
all hope you will come again sooner or later as you find the time. Okay. Thank you. Thank
Yes.
you.
I'm actually coming to Kolkata next week.
And that I have heard from Subudip. Well, come to our college. No problem. Subudip will accompany you. Although it is the time of COVID,
Yeah.
so no problem. We will be there. Okay.
Thank you.
Sorry for the delay. I'm just trying.
No problem. No problem, Mona Lisa.
So, this is the certificate.
Thank you very much.
The pleasure is ours.
You are most welcome.
Yeah, this has been a very, really, I mean, it's kind of like homecoming, as I said, that presenting for physiology community.
Thank you.
Thank you. And now we will proceed towards the poster presentation session of this webinar. We have received many posters, which we will be sharing with you all now. I'm sharing my screen, and one by one I will scroll down across the posters. So this is our first poster titled Effect of LED Lights in the Nocturnal Insects in Urban Setup, a Preliminary Study by Payal Ghosh, Shoikot De, Orijit De, Mondakranta Dashgupta, and Ankur Bhawal from Department of Zoology, Vidyasagar College. The next poster is ant assemblage in a managed urban garden, a preliminary study by Orko Roy, Mandakranta Dasgupta, Arijit De, and Ankur Bhawal from Department of Zoology, Vidyasagar College. Well, due to time constraint, we are just scrolling through. You can go through this in our souvenir. The next one is entitled Pathophysiology of Congestive Heart Failure by Sami Akhtar, MSc, third semester, Department of Physiology, University of Calcutta. Moving on to the next one, Bioremediation and Aid for a Clean Nature by Debanjan Bhattacharya, MSc, third semester, Department of Physiology, University of Calcutta. The next one is people with Parkinson's disease experiencing hallucination by Ankush Kumar Das, Department of Physiology, Vidyasagar College. The next one, Raise Awareness About Alzheimer's Disease by Ashikul Haq. He is from Physiology Department, Vidyasagar College. This is an interesting one. Tyrannosaurus rex, one of the fiercest predators of all time, by Shohum Chattopadhyay, BSc 5th semester, Department of Physiology, Vidyasagar College. Moving on to the next one. It is Thalassemia, it is serious, by Monoshija Chatterjee, BSc 5th semester, Department of Physiology, Vidyasagar College. The next one is stress, by Melissa Poddar, BSc 5th semester, Department of Physiology, Vidyasagar College. The next one is bug of malaria, by Srijita Bhattacharya, BSc 5th semester, Department of Physiology, Vidyasagar College. The next one is The Gut Brain, by Harsh Agarwal, BSc 5th semester, Department of Physiology, Vidyasagar College. Moving on to the next one. Hemophilia, by Roshmita Shaha, BSc 5th semester, Department of Physiology, Vidyasagar College. Next one. Auditory ill effects from overusage of earphones in this digital era, by Shumon Parui, BSc 5th semester, Department of Physiology, Vidyasagar College. There is another one. I will just share it with you. This is the one, the crosstalk between stress and cancer by Kanchan Mukherjee from 6th semester, Surendranath College, Department of Physiology. So with this, we came to an end of the poster presentation session. Now to begin with our second technical session, it is our great opportunity to have with us former Professor Amar Kumar Chandra and Dr. Sparanika Patnayak. Professor Amar Kumar Chandra, Emeritus Professor, Department of Physiology, University of Calcutta. He is actively engaged in teaching and research in physiology, especially in the postgraduate level for more than 4 decades. He is the president for the oldest association of physiology in India, the PSI. He has served as a general secretary for South Asian Association of Physiologists and many other associations related to physiology and allied sciences. He is an outstanding scientist related to iodine, goitrogen, and other thyroid-related compounds, which is already evident through his over 95 publications with exceptionally good impact and lectures delivered in different national and international conferences. We are honored, sir, to have you here today. We also have with us Dr. Sparanika Patnayak. Dr. Patnayak, MSc, PhD, DSc, is currently serving as an assistant professor in the Department of Biotechnology and Bioinformatics, Sambalpur University. Dr. Patnayak has a research interest in the field of medical microbiology, microbial genomics, and biotechnology with more than 65 publications in renowned national and international journals. And she has also delivered invited lectures in different countries. It is our honor to have you here, ma'am. I would now like to request Professor Amar Kumar Chandra and Dr. Sparanika Patnayak to chair this session.
Thank you, Mona Lisa. Hello, I am audible.
Yes, sir. Yes, sir. You are audible.
Thank you, Mona Lisa, for your nice introduction, especially in this webinar on contemporary innovation and challenges of modern biology. I am too much thankful to Dr. Subhodip Ganguly, Departmental Head, Vidyasagar University, for inviting me in this very nice session. He just casually told me that, sir, I am too fond that I am requesting you please participate. I have not thought at that time that this webinar is so much serious, because just now I have gone through the lecture of Dr. Sayantam Bose. It is very nice, very excellent, very lucid, and very rich. I hope those who have listened to his lecture, they have enjoyed. I am also thankful to Professor Gautam Kundu, the principal of the Vidyasagar College. I have nostalgia about Vidyasagar College because I have started my career in Vidyasagar College as a part-time or guest teacher or like that. During that time in Vidyasagar College, the physiology in the past classes used to taught, but at that time, the honors has been introduced. And it is a great occasion on this webinar, wherein I am seeing lots of known people. For example, I am seeing, after a long time, Dr. Alok Ghosh-Choudhury. I am seeing Mousumi Dey. He was one of my students. Dr. Chandana Sarkar, one of the important faculty of the department. Dr. Devashish Bandyopadhyay, once he was my colleague in the University of Calcutta. And Dr. Rohindra Chakrabarty, and so on. So I am not, I do not want to pass much more time, but the topic is very, very interesting. It is contemporary innovation. It is a very important term, and the challenges of modern biology, biology research. It is also very important term, and the two terms are very correlated. Contemporary innovation, always, every day science is changing. To keep one balance in his subject, in his field, in a contemporary innovation is very important. Contemporary, in addition to his classical knowledge. On the other hand, challenges of modern biology research, it is also important. Because 1 year or 2 years back, we have no idea about the COVID-19. But now, lots of things has happened. It has taken the life of more than 3 million. Most of the population, common people has no idea. Even the scientist has no idea that such a thunder comes. And after that, lots of research is just going on. How it is occurring, how it will be prevented. These are very, very interesting. This is the modern science, modern biology, modern medical biology. However, the modern medical biology stands on classical science, classical biology. So the two terms, contemporary innovation and challenges of modern biology research, is very interesting, and I hope we will listen this time. With me, the Madam Dr. Sparanika Patnayak is here, and how far I have listened, that she is also a very good scientist. His face, her face, her appearance shows that he is a very good scientist. She is with me, and now I am giving Dr. Sparanika Patnayak to say something. Thank you, Mona Lisa, for your nice introduction. I am seeing you after a long time, and for this I am enjoying a lot to see my old fellows, old students, and so on. Thank you.
Thank you, sir.
Thank you. Thank you, Amar sir. And this is, I am privileged to be here, and because Subhodip Ganguly he's there, and he's a nice chap, and I think with this scientific congregation and from a beautiful, means it is so scientific, that is from Dr. Bose, that is from USA. He has delivered a talk, and we are enriched with that one because of this COVID-19 pandemic and all. So it is nice to be here. As the time is up, and now, sir, we can start the session, and the students and the research scholar who are on the queue, so they can start their topic. So, Mona Lisa, thank you, and you can take the ring and all. So, I am here, and Amar sir also there.
Thank you, ma'am. Okay. Now, with the kind permission from our chairpersons, we will begin our oral presentations from the participants from different parts of our country. I would now request our participants to start their oral presentations. Our first participant is Chandrasekhar Tripathi from ICMR Regional Research Center, Bhubaneswar, Orissa. The topic is in silico investigation of phytochemicals from Sesbania grandiflora against HER2 for oral cancer therapeutics.
Good morning to all the exchange person present in the webinar and the research scholars present here. And myself, Chandrasekhar Tripathi. I am from RMSC Bhubaneswar. Today, I am going to present my webinar topic. Yes. My today's topic is in silico investigation of phytochemicals from Sesbania grandiflora against the HER2 protein of oral cancer therapeutics. My corresponding author is Dr. Santosh Kumar Behera. He's from Nagpur, Ahmedabad, Gujarat. Okay. Coming to introduction part, the current study is based on in silico approach to find out the natural compound from Sesbania grandiflora against the oral carcinoma. In the investigation, the protein, namely HER2, human epidermal growth factor 2, has been targeted to study the oral cancer. Here, the structure of the HER2 protein has been downloaded from PDB database with the protein ID 3PP0. Here, the chain A of the protein has been selected for the study. Here we have a targeted from the base pair like 706 to 993 base pair. Total is 286 base pair we are going to study. Here the computational drug designing process has been used to screen out the best phytochemicals from Sesbania grandiflora to create the oral carcinoma. Coming to oral carcinoma, this is the sixth most commonly occurring cancer in the world. It's oral carcinoma. Oral cancers include the cancer of the lips, tongue, cheeks, floor of the mouth, heart and subpalates, sinus and pharynx of the throat. Excess consumption of the tobacco and alcohol are recognized as the foremost risk factor for this oral carcinoma. It is a very crucial time to develop a drug from natural apart from the synthetic drug. HER2 protein it plays an important role in occurring of the cancer in human body. HER2 is a member of epidermal growth factor receptor family, having the tyrosine kinase activated. Demonization of the receptors results in the autophosphorylation of the tyrosine residues with the cytoplasmic domain of the receptors and initiates a variety of signaling pathway leading to the cell proliferation and tumorigenesis, which leads to the cancer. The overexpression expression of HER2 protein causes the oral cancer. There are some of the pictures that of the oral cancer affect all the mouth parts like tongues and teeth and the upper part of the mouth and all the parts of the mouth affected in this diseases. And coming to Sesbania grandiflora, which is my target plant, it's common as a vegetable human grade West Indian pea, or in Indian local names Jayanti, Agasti or Agati or Kachurai in the Telugu. Genus is Sesbania, family is Legumin. With medicinal properties is anti-cancerous, treating the diarrhea, dysentery, microbial infection and inflammatory disease. It is available throughout all of the India. In the current in silico investigation, in the current state, we have selected 18 phytochemicals from Sesbania grandiflora for our in silico investigation. Reported in various research papers. The details of the phytochemical have been downloaded from the PubChem databases. This is the list of the 18 phytochemicals that I have selected for the study. Then the phytochemical are screened out various online tools for checking their pharmacological properties. In the TargetNet server and Potox server I have used for my study to check the aromatic properties of the ligands. The results are given below. In the results, we have targeted 18 phytochemicals from the Sparanika grandiflora, and which compounds which pass through the to both the server, like TargetNet server and Potox server. I have skinned out the skin out the phytochemicals. And TargetNet server, we have targeted the Lipinski rule of 5, which has been very efficiently studied throughout the world for the pharmacological properties, for checking the adsorption, distribution, metabolism, etc., and toxicity. And in the Potox server, we have screened out the 6 phytochemicals found to be non-toxic in nature. Comparing both the result from the TargetNet server and Potox 2 server, we have selected these 6 phytochemicals for our study. Then, out of the 80 phytochemicals that I have selected, with 6 phytochemicals we have selected for our study, which pass to both the server and follow all the rules of admin properties. So we're going for molecular docking study with the HER2 protein with the oral calcium, along with a reported drug, namely fluorouracil, which has been reported drug in the various papers found that this compound has been studied effectively. But in the study, we have targeted the plant, Sparanika grandiflora in the Augusti plant. Then we have docked this HER2 protein with the orthodox 4.2 tool. I have predicted the binding sites of this of the protein using the CAST-P web server. These are the I have mentioned. These are the binding sites of the protein and the git coordinates that I have mentioned: X78, Y86, and Z76. Okay, I have done the docking process. That is, I have docked the molecular docking process I have been used, and I found that 1-propanol 2-methyl with the higher binding affinity, that is minus 5.5 kilocal per mole, with among the 6 phytochemicals, and with the 3xn 2,1 and 3,4 dimethyl is found to be second highest. When comparing, as we have made a comparison study with fluorouracil, when the fluorouracil targeted to HER2 protein, it is found that binding affinity is less than the 1-propanol 2-methyl. This has been applied. It is a phytochemical that is found from the Sparanika grandiflora. It is about minus 3.68 only that has been binding energy that I have got. Binding energy effectively studied in the molecular docking, how the ligand is how closely attached to the protein, which will neutralize the activity of the or stop the overtake expression from the of the protein. With 1-propanol 2-methyl is found to be hydrogen bonding capacity with AsP863 residues of the HER2 protein, and with absolute bond distance of 1.80182 angstrom. These are the 2D and 3D interaction of the 1-propanol 2-methyl with the HER2 protein of the oral cancer that I am studying. This is the 2D and 3D interaction of the fluorouracil with the HER2 protein of the oral cancer that have been selected for synthetic drug, which commonly been used. Okay, coming to this DFT calculation. DFT calculation I have performed using the ORCA program, which is a quantum mathematical calculation. I have perform this work with my ORCA program. Here, the 1-propanol 2-methyl is found to be found with more electronic energy than fluorouracil, which with a HOMO-LUMO gap is higher also. HOMO means Human-LUMO gap energy. LUMO stands for lower unoccupied molecular orbital, and HOMO stands for highest occupied molecular orbital level. Here in the picture that I have shown, the red color is in the ligands that I mentioned for positive electron density, and the blue color is showing to the negative electron densities of the ligands. That when plus to HOMO and LUMO, they are transferred to their energy. I have calculated for both, and for 1-propanol 2-methyl and propanol and for the LUMO of the fluorouracil that we have been synthetic drug. Both the cases I have studied that. Finally, coming to my findings that I have got in observation, that from the in silico investigation approach, it has found that the photo compound, namely 1-propanol 2-methyl from Sparanika grandiflora, can be effective drug candidate against the oral cancer. Hence, this in silico inhibition could be preliminary evidence for the in vivo and in vitro studies, which need to be carried out to confirm the efficacy and study the anti-cancer potency of the phytochemical from Sparanika grandiflora, which is a common medicinal plant found in all over the India, so that the cancer people can be able to cure from this disease. This is all about my topic. And especially thank to the Vidyasagar College Principal Sir Dr. Gautam Kundu Sir and all the respective members of the and other research persons for giving me a chance to present in the webinar. Thank you.
Thank you, Chandrasekhar Tripathi. Now for the question answer session. If anyone wants to ask anything, I can proceed.
Yes.
So, any questions from the student side? Students? Okay.
Thank you.
Right. I would like to ask one question.
Yes, ma'am.
This is what you did, Lipinski's rule.
Yes,
so,
ma'am.
So can you define what is Lipinski's rule?
I'm going to my slide, please. Okay, Lipinski rule of height mostly depends upon how the molecular reactivity, how it pass to the overall body system or to the sinus, and mostly probably the log peak value that I'm showing in my skin. It should be the less than 5 potentiality of the ligand, and whether we are to check in the admit properties like absorption, distribution, metabolism, and excision process of the human body when the drug target will be passed through the entire body system, how they will, if it is found to be toxic, then we can proceed further for that in this in silico inhibition that it will get a result. This is the role of height depends upon how the intricately the compound would move inside our body, and the target will be specific. The targeted protein should be specific or disease should be specific. Then it will be targeted specifically, and we will get a better result from it.
Right. But Lipinski's rules, there are 5 rules are there.
Yes,
so what are these rules? This ADMT is not coming under the Lipinski. This is these are pharmacological principles. What I know.
Yes, ma'am.
Can you say?
Created that will be AMR, the molecular reactivity, study molecular weight and hydrogen bond donor and hydrogen bond acceptor and the log value. This is the basic. This is basic concept that we taken in our in the Lipinski rule of height study.
How many violations you can skip? Suppose 1 violation or 2 violations. How many violations you can skip up?
Yes,
means there may be the 1 or 2 violations will be there. You cannot. That means it can be also mutated or something.
Yes,
how many violations?
When in the server, I have only taken the which patch which 100% integrity that I have the server with with 100% indeed I have taken only that, but I have not taken the less than 100%. I think 2 to 3 lakh or they did not follow the rules that I have not taken into consider my account. To that so can go for the low taken cannot risk, so that it will affect the process of the insulin, and that will be harmful to us.
Right. Can it be integrated with the in vitro study? Because these are only predictions. So
yes,
in vitro studies are recommended.
Yes, ma'am. Yes, ma'am. In vitro, coming to my this has been that I have much known that I'm presenting it in vitro studies needed to be carried out study in elaborately. So for it needs to be described and studied more effectively through in silico before going to the in vitro studies or in vivo also.
Right. My part is over. Now I can pass on to Sir. Sir. Amar sir.
Sir, I think you are muted.
Hello.
So, shall we move on to our next participant? Or okay, Thank you, Chandrasekhar.
Thank you. Welcome, Mr. Should I stop sharing my slide?
Yes.
Thank you.
So our next participant is Adrita Banerjee from Oxidative Stress and Free Radical Biology Laboratory, Department of Physiology, University of Calcutta. The topic is emergence of oleic acid, a monounsaturated fatty acid as an antioxidant, a brief insight.
Presentation is visible?
No, Adrita.
Is it now?
Yes, it's coming, I think. The slides are not visible till now.
Is my screen is visible?
Yes, your screen is visible.
Okay, I'm trying. Is it visible?
No, I can't see anything. Ma'am, are you saying something?
Adrita, Adrita is presenting. One window you can pin in. Window from your computer. Wherever your PPT is there.
My screen is there.
Yes, there. Again, you can means again you go back. Then you open. First you enter inside. You just present yourself. Then you open. your PPT. So that will come. Opening should be post entering.
I have opened. Is it visible?
No, I'm not saying anything.
Mona Lisa, if there is any problem, give the chance to next one, and in the meantime, Adrita will be ready to herself.
Okay, sir. Okay, sir.
It will be better, I hope so.
Yes, sir. Yes, sir. Okay. Okay, Adrita, you better try again later on. So in the meantime, I am calling our next participant. Our next participant is Shanchari Chakraborty, Junior Research Fellow, Department of Physiology, Surendranath College. The topic is Revisiting Polycystic Ovary Syndrome.
Hello.
Good evening, respected sir and madams. Am I audible?
Yeah.
Yes, you are audible.
Okay, thank you, ma'am. Now I am sharing my video. Is the slides are full screen, ma'am?
Yeah.
Okay, ma'am. Thank you. My topic is revisiting polycystic ovary. Myself, Shanchari Chakraborty, Junior Research Fellow, under the guidance of Dr. Bornali Rai Basu, Department of Physiology, Surendranath College, Kolkata. Polycystic ovarian syndrome is a metabolic and endocrinal and reproductive disorder. It's a cumulative expression of multifaceted and complexity, and they are mainly in women within their reproductive age. Infertility is expressed through the syndrome. There are some pictures which we have collected during our survey work, like acanthosis nigricans, dark patches of skin, alopecia, thinning of hair, hirsutism, excessive hair growth, and acne like pimples. These are the phenotypic manifestation, and this indicates the hyperandrogenic, we can say the excess androgen level in female relative to the normal individual. That indicates the first or primary indication of PCOS. And there is another very complex as well as. Hello, ma'am. Excuse me. Just a second, ma'am. I will recheck. Just a second. Ma'am, the slide is sharing. Excuse me, ma'am.
Sanchari, go on.
Slide is sharing, sir.
Yes. Slide is quite visible.
Visible and moving. If the slide is moving also.
Yeah. The slide is visible, quite visible.
Visible and moving, sir. Slideshow. Like.
Slideshow, it is. Please go ahead.
Polycystic ovary.
Okay.
Insulin resistant. Okay. Thank you. The policy. There are the four indication of polycystic ovary that are the phenotypic manifestations. Hyperandrogenism, like acanthosis nigricans, that is the velvety dark patches, alopecia, hair loss or hair growth reduction, hirsutism, excessive hair growth, and acne, like pimples development. And the behind of this manifestation, there may be excessive androgen surge that trigger the this manifestation, and these are the highly correlated with PCOS. The next slide is insulin resistance. Is it visible?
Yes.
Okay, thank you. Insulin resistance is another common but not universal. It's a very common condition nowadays, and various studies have shown that there is a very cooperative and intricate interaction with PCOS, because insulin resistance is the can be said the cell's responsiveness towards the insulin is reduced. That's why it triggers hyperinsulinemia, and the excessive insulin in circulation has the effect, or we can say has the receptor in brains and as well as in our reproductive structure, female reproductive structure, that stimulates the LH hormone, luteinizing hormone, and ovarian steroid hormone, specifically or specially the androgen one, so that the estrogen level and progesterone level gradually decreases, which are the major female hormones, and male pattern hormone increases that reduces the ovarian follicular growths that turns into cyst development. Next, there is a crosstalk crosstalk, or we can say. May I continue?
আচ্ছা শোনো আমি একটা মিটিং এ আছি। আচ্ছা তুই হ্যাঁ আচ্ছা ঠিক আছে।
Sanchari, you continue.
Okay, sir. This is a loop, or we can say there is a cycle that try to. There is a we can say hypothesis that to try to explain the mechanism, biochemical as well as molecular mechanism, how PCOS develop and progresses. When the hyperandrogenism, when the hyperinsulinemia reaches, there is a interaction with hyperandrogenism, and this hyperandrogenism and hyperinsulinemia both stimulates GnRH hormone level as well as LH level surge that inhibit the progesterone secretion that goes to ovarian cyst development, and that's why there are the multiple manifestation like oligomenorrhea and amenorrhea, and sometimes infertility can be associated. In the emerging studies, there is another molecule and very interesting one, that is adiponectin. Adiponectin is a adipokines, which is majorly released from adipocyte tissues, that helps to maintain insulin level in a indirect way via various protein adiponectin protein interaction, so that to maintain the balance of our feeding and our bodily structures maintenance. But when the level adiponectin is a very versatile as well as very the isoforms are present, when usually high molecular weight adiponectin level decreases, that indicates there is a triggering of insulin resistance as well as inflammation that somehow interacts with PCOS, because there is a crosstalk. But till date there is multiple confusion as well as unknown area where how these molecules, and there are multiple veiled molecules are present, they crosstalk to express and penetrate the PCOS expressivity. So there is another aspect, that is the genetic one, because the human organism is an open system and there is a continuous constantly interaction, nature versus nature, or we can say endogenous and exogenous environment continuously interacting. So there are multiple factors in our environment as well as our lifestyle that can affect our genetic structure, and that can be propagated throughout our generation from parental to their offspring. And that is the various studies shows that PCOS has an autosomal as well as sex-linked characteristics, so that PCOS expressivity can be sex determining, that's why. So here we are focusing now to study SNP, single nucleotide polymorphism, where we we are trying to locate a specific nucleotide can alter the, as we know, the central dogma pathway, where the single nucleotide alternation may change if that is not the silent, that mismatch mutation or nonsense mutation, that can change the protein expressivity as well as functionality, so that we can, there may be multiple harassment. PCOS is one of them. There are some studies that shows the PCOS interaction with PCOS with insulin resistance. Here the majorly insulin receptor gene and the other genes or intron regions or localized region where SNP RS gene are studied in various continents as well as various countries. So, and there are some studies shows that there is a significant correlation, but some studies there is no significant correlation in point of view or SNP or genetic basis. But that doesn't that may not mean that insulin and PCOS is not interconnected, that there may be after genetic level, in the biochemical level there may be interaction. There is another very crucial and interesting and important area where in West Bengal there are very the SNP study is very low and very low reported. So there are requirement of more studies in ethnicity dependent manner, so that these unknown factors can be revealed. This table is another interaction of PCOS and adiponectin, where various area, or we can say various countries shows the interaction, but still there must be study with various gene as well as the pathways, molecular pathways, so we can interpret the exact pinpoint mechanism. There is my understanding about the entire interaction among PCOS, insulin resistance and adiponectin. Here there is an indication of PKB mutation, protein kinase B, of the insulin receptor pathway, where the mutation, if there is a mutation occur here, as well as there is a IRS, insulin receptor responsive substrate, where if there is a phosphorylation in serine instead of tyrosine, that may deregulate, or we can say deregulate the entire insulin signaling pathway, so that insulin resistance generate, and there is the insulin resistance that triggered the hyperinsulinemia that stimulates LH secretion as well as PCOS. And adiponectin has an interaction indirectly with the insulin receptor via the adiponectin binding protein. There is a crosstalk. Okay. And the same time, there is a as the insulin responsiveness is deteriorated, the GLUT4 is not working properly, so that the glucose level in blood increases, and there is a biomolecular metabolism also affected. Biomolecular in the sense lipid level, carbohydrate level, and as well as protein level. There is a catabolic anabolic imbalance that show the oxidative stress as well as thermodynamics imbalance. So in conclusion part, the PCOS is a very old, recognized around 1721 in Italy, recognized, but the recognition is not fully completed in terms of the reason why this disease is generated. How can we recover from the diseases? Which factors are responsible for the PCOS condition, and which genetic factor, and as well as gene environment interaction. So there is requirement of lots of study based on molecular as well as genetic, as well with the background of our anthropometric and biochemical basis, so we can compact our study in a manner. I would like to thank Department of Physiology, Vidyasagar College, and our funding agency, Department of Science and Technology, Biotechnology, Government of West Bengal, for their financial support, and Surendranath College, where I am pursuing my research work under guidance of Dr. Barnali Rai Basu. This is the paper which was published in October session 2020, based on stress, anthropometric, and PCOS interaction in International Journal of Scientific Research. So thank you.
Now the session is open for discussion. If you have any question for Sanchari. Any question?
Hello. Hello.
Yes.
Yes.
Sanchari,
Yes,
thank you very much. Yes, can you hear me?
Yes, sir. I'm hearing.
Yes, yes. Thank you very much for your very nice presentation. Okay, congratulations.
Thank you, sir.
And I have only one suggestion for you to consider in your future studies, if possible, that how you find a correlation between adiponectin metformin interaction, because many of the PCOS patients consume metformin for a long period of time. Okay,
Yes,
so whether there is a crisscross relation between adiponectin and metformin will enrich your PhD thesis work, I think, in future.
absolutely, sir. I will definitely try. Sir.
Yes,
But
yes. And thank you, thank you very much for your very smart presentation.
Okay, sir. Thank you so much.
I love to hear you in the seminars. Thank you very much.
Thank you, sir.
Any other question? Sanchari,
Yes, sir.
I have some suggestion.
Yes, sir.
First of all,
Yes, sir.
control your movement, posture during delivery of lecture, because you are moving continuously. You are comfortable, but the audience feel something else. So it will be easier. But what is your work? You have just a glimpse about the literary. The things that you have presented all are in the available literature.
Yes.
So what is your actual work? Whether your work is survey based or whether your work is laboratory based, what is your study? What is your aims and objectives?
In our study, sir, the aim is to study the three aspects of PCOS. That is, first one is anthropometric, and second one is biochemical, and third one is genetic basis, and the correlation among these three components. And our study sample is human organism, and we are collecting the samples from PCOS patient who are recognized as PCOS patient by Rotterdam criteria from SSKM hospital, and the study is going on. Hopefully we can conclude a specific pinpoint discussion or conclusion, so that in our area, especially in West Bengal, we can say there is a data can be found that can be referred by us, that would be used by further research work. So
No, it is nice. But if you share some of your result, it would have been better for us.
okay, sir.
Because there are lots of people, there are lots of scientists, they would give suggestions to you, you will interact with them with your results. So in future presentation, you will take attention in this regard. Thank you, Sanchari.
Okay, sir.
Madam, you have anything to say?
Sir, thank you, sir. I have one question, but others are there. Now time is up. Anyway, one question is there. Suppose you will be carrying out the SNP study. So how you are going to do SNP?
First of all, we collect the saliva sample from the patients as well as control individuals. From the saliva sample, we target to isolate the DNA molecules in a procedure. Then the isolated DNA would be further studied PCR RFLP. Then after that, we will then perform the gel electrophoresis and then genome-wide association study, so that we can say if there is any changes in nucleotide or not.
But because we are now traveling with NGS, next gen sequencing, so you can take that opportunity, and that is why you can go for the either the draft genome sequencing or the whole genome sequencing. Then that also that is I think it will be easier for you to get the SNPs using
Yes,
the
ma'am.
omics technology also.
Yes, ma'am.
Right.
Yes, ma'am. We also thought about the next generation sequencing. So, can the genomoid sequencing can I think target the widely SNPs more, around 10%, more than 10%. But in next genome sequencing, we can reach the less than 10%, but greater than 1%, so that there can be recognized and further studied. Thank you, ma'am, for your suggestion.
Thank you, sir.
Thank you, thank you. Thank you,
Thank you, sir. Thank you, sir.
Okay, so moving on to our next participant. Our next participant is Kaustubh Bhattacharya, Department of Physiology, Vidyasagar College. His topic is isolation, characterization, and development of cadmium resistant strain, a possible eco-friendly method of wastewater treatment.
Are the slides being seen on the screen, sir?
Not visible.
Visible. May I start, sir?
Please make it full screen. Not visible till. Stop. It is yeah. Please make it large. Okay.
Good afternoon to all the respected sirs and ma'ams. I am Adrita Banerjee, MSc in Microbiology. I am a CSIR fellow. Here is my letter number. I am working under the supervision of Dr. Shubhadeep Ganguly, Assistant Professor and Head, Department of Physiology, Vidyasagar College, 39 Shankar Ghosh Lane, Kolkata 706, West Bengal, India. Today, I am here to present my research work named Isolation, Characterization, and Development of Cadmium-Resistant Strain, a possible eco-friendly method of wastewater treatment. Now, Industrial Revolution is considered as a hallmark of civilization that gives rise to the omnipresence of heavy metals in our environment, and which are potentially dreadful elements. And cadmium is such a heavy metal having no physiological role as by some metals that plays roles as trace elements in cellular metabolism and is potentially toxic even in trace amounts. And after entering the human body, it accumulates lifelong and poses various health hazards named nephrotoxicity, hepatotoxicity, pneumotoxicity when gets inhaled, teratogenicity, and above all, it has tremendous carcinogenic properties. It is the 67th most abundant element in the earth crust, and its concentration is 0.2 milligram per kg. The references are given. Usually, unpolluted natural water bodies bear cadmium concentration lower than 1 parts per billion, according to Fiber Gretel 1986. There are various natural and anthropogenic sources of cadmium release. The worth mentioning natural sources are weathering of rocks, volcanoes, wind-blown dust, forest fires, etc. Anthropogenic sources, zinc minerals generally receive isomorphous replacement from cadmium, and as a result, and sphalerite, Zerdenis is the principal industrial source of cadmium. Others are manufacturing phosphate fertilizers, metallurgical processing industries, incineration of cadmium-laden sewage sludge, dumped nickel cadmium batteries in the landfills, poses soil cadmium pollution. Cadmium is principally released into wastewater from fertilizers and local air pollution. Drinking water may also become affected by the toxicant from the impurities of zinc, galvanized pipes, solders, and metal fittings. And Pollution Control Board, the north and south the groundwater in north and water bodies in north and south 24 Parganas bear cadmium concentration higher than the prescribed permissible limit by EU and BIS. According to Sharma and Datta, they have assessed some groundwater samples in Punjab. They found considerable cadmium laden groundwater samples, and in Firozpur it is considerably higher, 2.9 parts per million. And in water bodies in Uttar Pradesh, Gujarat, and Chennai also bears the same scenario. As a result of this drastic condition, various remediation technologies have evolved to combat with the toxicant related pollution. These are referred to as physicochemical remediation technologies, but in today's scenario, these techniques have already become outdated and suffers from significant drawbacks that have been mentioned, detailed in the slide. And it can be summarized that the principal drawbacks are high operational cost, generation of toxic sludge, that requires additional removal cost and inefficiency in the removal of metals from dilute solutions. And as a result, a novel technology needed to be sought out that will be efficient, cheap, and eco-friendly, which gave rise to the emergence of biological remediation technology or bioremediation. And biosorption is defined as the adsorptive capabilities of materials of biological origin, such as microorganisms, whether it is viable or not, and the principal mechanisms involved are ion exchange, covalent and non-covalent interaction, complexation, principally chemisorption and physical adsorption, and surface precipitation. It has been diagrammatically represented. Advantages of biosorption here is it is a cheap process. The biosorbents possess sufficient tenacity to the toxicant, selectivity to the toxicant because of their complex surface structure, and after adsorbed with the toxicant, they can be dissolved and be used in the toxicant removal process again and again. Now, the aim of our present study is to isolate, characterize a cadmium-resistant strain from highly polluted wastewater. After that, development of the resistant strain by gradually mediating higher cadmium dosage, tolerating the strain to higher cadmium dosage to get until the minimal inhibitory concentration is reached, beyond which no growth will be observed. As a result, the strain that will be emerged will possess considerable cadmium tolerance and removal capacity. Let's see in the results and discussion section. Here are the routine microbiological techniques we have followed. We have collected samples from wastewater samples have been collected from Tollygunge Canal because it is known to be sufficiently contaminated with various liquid and solid wastes and transported in the laboratory in a cold line, aseptically collected in sterilized bottles. After that, routine microbiological technique by agar-plate serial dilution technique by using yeast extract peptone dextrose agar have been used by employing chloramphenicol and fluconazole to avoid possible contaminations. And eventually, in our study, we have isolated 35 bacterial, 9 yeast, and 5 filamentous fungal strains. And each of the strain was assessed for their cadmium biosorption capacity by employing 10 parts per million in the sample. And eventually, each strain, initially named as GIC, evolved to possess maximum biosorption capacity, named 43.60. This is the composition of the medium. After that, the strain was subjected for phylogenetic analysis, and after that, the strain was subjected to gradual adaptation process with using gradual higher dosages of cadmium until the minimal inhibitory concentration was reached, and the strain that emerged showed considerable cadmium resistance and biosorption potency. And here, instrumental detection and analysis was performed to observe any kind of change in surface morphology in the resistant and untreated cadmium treated and untreated strain by FEACM and SIM analysis. And EDX energy dispersive X-ray analysis has been performed for the treated and resistant strain to observe any kind of morphological changes on the, have been performed to analyze the elemental composition of the cell surface. And FTIR analysis was also observed to analyze any interaction of surface exposed functional groups with the toxicant. Equilibrium biosorption studies have been performed by using Langmuir and Frohnlich adsorption 2-parameter isotherm models. Here is the Langmuir model, and it is the linearized version. And from where we have calculated the RL value, the Qmax and KL value have been calculated. Qmax defines maximum adsorption capacity, and KL is the Langmuir constant. And we have also calculated the Frohnlich isotherm that describes heterogeneous adsorption, and it is the linearized version. From where we have calculated K and N, it is a heterogeneity factor that determines the nature of the adsorption process. We have also undergone the adsorption study by applying kinetics of the pseudo first-order kinetics posed by Lagergren and second order by McKay and Ho model. And after that, optimization of all the physical parameters have been done by considering one factor at a time, considering the parameters pH, food temperature, age of inoculum, volume of medium, volume of inoculum, containing 82 to the power 6 cells per ml. And after that, considering all influential effects of all the factors have been considered by applying response surface methodology, and the exponential data was analyzed by using this polynomial equation, and it was central composite design was applied for the study. And these are the instrument software generated ranges, and we used the design expert 0.7.1 software for calculation analysis. In the results and discussion section, I have already said that we have, after exposing the strains with 10 ppm of cadmium, this strain named as GIC showed maximal bioabsorption capacity. This is the formula. We have calculated that, and after that, morphological and biochemical analysis of the isolates have been determined. And after that, molecular phylogenetic analysis have been determined by genomic DNA isolation, amplification of the ITS region, and the sequence that had been obtained. It has been analyzed by pairwise sequence alignment by taking 100 BLAST, and after that, maximum sequence identity score 10 sequences were selected, and multiple sequence alignment was carried out by cluster W, and distance matrix and phylogenetic tree have been generated. And it has been shown that the strain shows 98.64% approximately sequence similarity to Candida tropicale strain, and from here we can conclude that our strain we have isolated is a Candida tropicale strain. The sequence has been submitted to the NCBI GenBank database, GenBank, and we have obtained this accession number NZ798900. This is the sport plate and streak plate cultures. And in the gradual, in the due course of strain development, in the gradual adaptation process, the maximum tolerance level for the strain was reached at 3,850 ppm, beyond which no growth was observed for the organism could not tolerate it anymore. Cadmium addition, and the strain thus developed showed bioabsorption potency, bioabsorption potency by applying 500 parts per million cadmium. It showed 59.6%, and the strain thus obtained was named Candida tropicalis XTF1874. After that, the strain was subjected for morphology changes in any kind of surface morphology for FESEM and SEM analysis, but both undergone cadmium in treatment. Intention to see whether any kind of change in surface morphology occurring or not in the developed and in the control strain. We see prominent changes in cell surface morphology in the strain that has been developed, that has been adapted to cadmium stress, but the control strain, untreated strain, unadapted strain retains native cell surface morphology, and after that, same analysis have been performed. Scanning electron micrographic analysis, analysis have been performed, and in the micrography we observed prominent after the resistant strain undergone cadmium treatment, but the control strain did not. The prominent changes in cell surface morphology, and here we observed a cluster of cells possessing a cup-like structure and accumulates cadmium microparticulates, but the control cells retains more or less native cell surface morphology, and this proves that surface accumulation is occurring. And after that, elemental composition of the biomass, both the treated and untreated one, have been observed by energy dispersive X-ray analysis. And in the spectra, EDX, we observe presence of cadmium peaks in the Candida tropicale strain, XTM1874, the resistant strain that undergone cadmium treatment, incubated the presence of cadmium, but no presence of cadmium peaks in the untreated one. And this proves that, and this is a very good definitive evidence that surface adsorption is occurring in the presence of cadmium. And these data have been verified further by doing FTIR analysis, and the predominant functional groups participating in metal-ion interaction are hydroxyl, amino, and carbonyl groups. And the kinetic analysis for bioabsorption have been assessed by taking initial cadmium concentration range in the 550 to 500 ppm, and the equilibrium concentration reached. These values are shown, and from where we have calculated the QE values, the amount adsorbed at equilibrium, and equilibrium bioabsorption capacity has been calculated by applying this equation: initial concentration at equilibrium, and after that, adsorption. These data have been analyzed by applying at the 2-parameter Isotherm models of Langmuir and Frohnlich. That Langmuir describes homogeneous surface adsorption, and it were from the linear plot of 1 by QE by 1 by C, we have calculated the Qmax value. That is very much good Qmax value, higher Qmax 109.45, and KL value have been calculated. The RL value has been calculated by applying equation 1 by 1 plus KL into C naught, and we observe that RL value lies between 0 and 1. That defines the preferability of the adsorption isotherm model, Langmuir adsorption isotherm model in defining the adsorption process. R squared value we have obtained 0.972, but in the Frohnlich linear fitting we have obtained from the intercept and slope the values of 1 by n, and Kf values have been obtained. Kf have been obtained from the anti-log of intercept, and 1 by n has been obtained from 1 by slope, from where n has been calculated, and we observe that n is greater than 1. That defines that adsorption is a physical process, and if it was smaller than 1, then it would be a chemical process, according to the postulate. And from where, after assessing the RL values and the correlation coefficient in Frohnlich and Langmuir, we observe that from both sides, Langmuir model is a much preferable model in defining the adsorption process in case of this strain. After that, kinetics of bioabsorption have been assessed by taking 100 ppm of initial cadmium concentration, and bioabsorption have been assessed. The rate of bioabsorption had been assessed with respect to these times, and we observed that after 25 minutes, no further increase in bioabsorption capacity, and from where we have calculated the QT value, and then from where we have calculated the natural logarithm of QE minus QT. And the data have been analyzed by applying pseudo first order kinetics, and the QV value have been that we obtained from the calculation is pretty much lower than that we have obtained from experiment, and the R squared value is we obtained is 0.6957. In the pseudo second, or by applying pseudo second order model in the linear plotting, we have obtained the calculated QE value is very much closer to the QE value we have obtained from experiment by applying 100 ppm of cadmium. And the R square value obtained is 1, is a perfect correlation, and which describes that pseudo second order model best fits in defining the kinetics of the cadmium bioabsorption by the strain. After that, optimization of all the physicochemical factors have been done, and by taking sample size n equals to 6, and at the conclusion we have reached that maximum bioabsorption by taking 500 ppm of initial cadmium concentration. The optimum bioabsorption at approximately 78.6 was observed at pH 6.5, temperature 27 degrees centigrade, age of inoculum 48 hours, volume of medium 25 ml, volume of inoculum 4%. After that, all the data have been, the bioabsorption process have been assessed in the presence of all the effects of all the factors have been assessed by considering the cumulative effect by response surface methodology. We obtained 79 runs from the software, For all the possible combinations, bioabsorption efficiency have been calculated, and after linear plot, we opt only the significant, considering the significant factors that have been obtained. We obtained a lack of fit non-significant. We considered the effects of pH and dry cell weight on the bioabsorption process, and from the 3D and contour plot, it is observed that with increasing pH and dry cell weight, bioabsorption increases. And from the effect of pH and inoculum, age of inoculum, we observed that with the age of inoculum, and with increasing pH, bioabsorption increases. Small but significant increase in cadmium bioabsorption have been observed when the effects of dry cell weight and age of inoculum had been considered. And so, although not significant, the effect of temperature and pH was also assessed. And we see that with increasing pH and temperature, bioabsorption increases from the 3D and contour plots. And we obtained and from the software we obtained the optimized value: pH 6, temperature 27 degrees centigrade, age of inoculum 48 hours, volume of medium 20 ml, volume of inoculum 4%, dry cell weight 2.1, and as the bioabsorption predicted by the software, it was observed that 78.6562, and experimental analysis of the bioabsorption percentage has been performed in the lab by maintaining all the optimum conditions, and we have obtained this value, 78.6223% bioabsorption. And lastly, in the conclusion section, we can conclude that we have isolated a cadmium-resistant yeast strain named JIC that showed maximal bioabsorption capacity among all the isolates, after that it has been identified by phylogenetic analysis. Later, the cadmium-resistant strain was developed by a gradual adaptation process with higher dosages of cadmium. And after that, and the bioabsorption efficacy have been calculated by applying equilibrium adsorption isotherm models and by kinetic modeling. After that, we have optimized all the physical factors that have been for their effects on bioabsorption process, and using the initial concentration 500 ppm, and we observed 78.623% of bioabsorption. And other factors such as effects of complex nutrients, vitamins, amino acids, and trace elements are under study. And it is to be hoped that after completion of the study of all the effects of all the factors, it can be hoped that bioabsorption capacity will be aggravated further. Our future perspectives is thus the strain that has been emerged will be applied successfully in cadmium bioremediation capacity, and the instrumental analysis that has already been done, it provides definitive proof that surface adsorption is taking place. Additional evidences will become from bed analysis of surface area, pore size, pore volume, to observe whether any kind of intraparticle diffusion is occurring or not, or the surface chemistry analysis can also be performed by XPS. One of them will be performed based on in this COVID situation, based on our facility. The basic and for the zeta potential measurement, that also provides a very good evidence of bioabsorption. Cadmium treated and untreated cells have been cultivated in our lab. It has already been sent to the Indian Association for the Cultivation of Science for assessment of the zeta potential. The experiment is underway, and it is also thought, and it is also hoped that it will provide a definitive proof of bioabsorption. I am thankful to the Council of Scientific and Industrial Research because the entire work was founded by them. CS Council of Scientific and Industrial Research, CSIR, Human Resource Group, Extramural Research Division, Government of India, New Delhi, 110012. I would like to express my sincere gratitude to my supervisor, Dr. Shubhadeep Ganguly, Assistant Professor and Head, Department of Physiology, Vidyasagar College. The concept was scientifically postulated and executed through his sincere guidance. I am honored to acknowledge Dr. Gautam Kundu, Honorable Principal of Vidyasagar College, for providing me the necessary facility, laboratory facilities, without which I couldn't be able to explore the new era lying at the interface between biology and applied chemistry for the benefits of human civilization. A sincere thanks to CRNN, University of Calcutta, Bose Institute, Indian Association for the Cultivation of Science, Jadavpur, Kolkata, SN Bose, National Center for National Center for Basic Sciences, and CSIR, Indian Institute of Chemical Biology, for providing me the necessary technical supports for analysis of the samples. Finally, it is my pleasure and honor to acknowledge Department of Microbiology, University of Calcutta, for allowing me to conduct this work, for allowing me to give this, for giving me this golden opportunity. And these are the references I have consulted in the due course of the study. And lastly, thank you all for hearing me for a long time. Thank you again and again.
Thank you, Costa, for your nice presentation. And now I request the audience to ask question if it is in their mind. Please.
Costa, I have a question.
Yes,
can you hear me, Costa?
Yes, ma'am.
Yes, you have a very good data regarding the uptake of cadmium, and I think this strain has been developed as a treatment for the sewage treatment. This is a very good organism that you are developing, and there are quite a good data regarding the cadmium uptake that is happening with this microorganism. But what I wanted to ask you, which I don't have knowledge, whether the strain that you have identified, that is the Candida tropicalis, if you increase the concentration of this organism in the water, that means the water that needs to be treated, how adverse effect regarding the pathogenicity of this organism can take place? Means what is the ultimate usefulness of this whole study that you are undertaking? The cadmium uptake part and the development of the strain is okay, but when it comes to the applied part, do you have any literature regarding this or any study regarding this, how pathogenic the microorganism is?
Yes, the pathogenic potential will be assessed in the due course, and I am already integrated in assessing the bioabsorption potency. The pathogenic potential, it is a very good, because Candida is a pathogenic strain, and it will be analyzed later in the animal model.
Because this particular strain, I think, is already known to be quite, it has a potential of causing a few infections. Means it is already established that it causes a few infections, especially in immunocompromised patients, because fungal infections are normally more prominent in immunocompromised patients. And if this in rivers and canals where this water, sometimes canal water is not used for drinking purposes, but at large, if you think about rivers which are prominent, and if that this organism, means if you increase the concentration of this organism, whether it would adversely affect the biodiversity that is present in the river. So the study is still awaiting, I think.
Yes, it may, because it may affect. Candida is a pathogenic. Candida has sufficient pathogenic.
Madam, can you hear me?
Yes, of course.
Madam, can I answer this?
Yes.
Actually, we have already studied the virulency of this strain in mice model in Department of Chemical Engineering, Biochemical Subdivision, Biotechnology Laboratory, University of Calcutta. It is a common protocol that...
Can I interrupt you? Virulency of the organism means the one which is taking care of the cadmium in large proportion, that strain that you have developed?
That strain we have developed.
That one, virulency of that organism that we have already checked.
Particularly the organism which was identified as the highest potency for cadmium bioabsorption, will go for particularly, it is a common practice in any absorption technique, that we have to go for virulency test in mice model. And this strain has already shown that it is a non-virulent strain in mice model, and it is a certified strain.
Okay. So this
otherwise pathogen toxins will be released from these microorganisms that will again contaminate the water, and that will create a further problem. So our purpose will not serve.
Okay, the more study is required to find out the pathogenicity of this cadmium, this high cadmium.
Actually, virulency test is a compulsory part in absorption in animal model.
Okay, thank you.
Welcome.
Any other question? Any other question? Cadmium has different form. Whether it will, I am inviting the attention of Pustav. Pustav, please. Your strain will act on only cadmium 2. Cadmium has other form. Whether your strain will only act on cadmium 2.
Because cadmium in water generally persists in the +2 form, as I have mentioned. It is a common toxicant in wastewater, and it is our motto to remediate wastewater, and we have only focused wastewater systems in focusing cadmium bioabsorption in the ionic form, because it persists generally in the...
I do not want the answer, but so elaborately. I am asking you that whether it is only against cadmium 2 or any other form of cadmium.
No, it has not been assessed. It has not been assessed.
Number one, whether you have isolated the strain.
Yes, the strain has been isolated. It has been isolated, and identification has been done by phylogenetic analysis. The strain has been sent to, and phylogenetic analysis has been done by the help of Eurofines, and from where we have it is the certified strain, and the phylogenetic analysis has been done. We have obtained the phylogenetic tree, and after that, the sequence that had been obtained had been sent to NCBI GenBank, and we obtained the accession number.
So whether you have selected wastewater, not other water, why not drinking water? Because wastewater is already waste. But why you have selected that? Because drinking water is entering our body, causing many harmful effects. Wastewater has no use at all. So why you have selected?
Wastewater, because
definitely you have a reason for selection of wastewater. Definitely you have reason, but my question is that in lieu of drinking water that is entering in the body regularly, that can cause harmful effect, in spite of that you are using wastewater. What is the benefit for that? I am asking only that part.
Because in general, water bodies, as I have mentioned, the cadmium concentration is very generally lower, but in the wastewater, from where we can obtain the strains that have already been tolerant to the stress, and it is to be assumed that after tolerating those strains at the higher dosages, the strain that will be evolved will show considerable potency in removal of cadmium from various water samples.
Okay. You are giving elaborating answer. I want the answer should be brief, because we are not expert in your line. You have lots of knowledge, but we have not that. And lastly, I have gone through your work that you have done extensive work. In the meantime, you have done extensive. Whether you have any publications?
Still not now. One paper has already been conducted, review paper, and other one is on the way. But we are also working. But publications, I have still not now any publications.
So what is the full form of JIC? JIC. Your strain.
I have only named the strains for the sake of identification. It is a trivial name, but
why? Why it is JIC? You mentioned it. That it is JIC. Okay.
I have
Thank you for your nice presentation. But one suggestion is that when you present any paper, you should not go through the literature through your eyes. You will not see it because you are a very good student. You have done extensive work. But my suggestion is that you will try to explain your work by yourself. You're just not going through the slides because it may be easier for you, but it is difficult for the audience. Thank you. Thank you for your good work. I wish you all the best in future. Try to do publication at your earliest. Thank you.
Thank you, sir. Thank you, sir. May I stop presenting?
Okay, Mona Lisa, if any questions, anyone can have any question. Otherwise, Mona Lisa, go to next.
Okay,
if anybody has any question, ask to postdoc. You will be happy.
If anybody want to ask anything, you can please proceed.
Please proceed to next. Achha, how many lectures, how many presentations are there?
And sir, there is total left presentation. Still, we have 3 or 4 presentations left in this session. 4 presentations.
Okay, very good.
Okay. Okay. So,
Sir,
yeah. Yes, please.
Sir, may I add one comment on Gustav's work?
Of course. You are, because for the researcher, comments is very important. Those people who are giving comments, they are his friends. They are his research colleagues. There is research. But definitely, if you have any comments, definitely. In public seminar, you must express it.
Actually, sir, Tollygunge Canal or any sort of wastewater, we are choosing because so far as heavy metal toxicants or something like that, whenever we are going for any sort of resistant strain development, we are trying to increase the level of tolerance of these microorganisms. So it is our preference that select such microorganisms which has already been tolerated in any sort of heavy metals. That is why it is a routine protocol. So far as heavy metal tolerance is concerned, we are choosing wastewater instead of normal water or groundwater or drinking water. So we can apply for purification of these any water body, applying this method, bioremediation process. Secondly, so far as Gustav is presenting, his research work can be divided into three sections. One is surface chemistry, applied chemistry. Second one is molecular biology. Third one is industrial microbiology. So far as his work has been done, it is related to surface chemistry. It is almost near the verge of completion because 2 mechanisms will be studied further, and and completion of all these things will publish this paper as per your suggestion. Thanks a lot, sir, for your comment.
No, not for my suggestion, because the part of the work that he has already been conducted should be published immediately. It is my suggestion. It is not only suggestion to postdoc, but to all, that what you have done, try to publish it. Get some comments, it will be beneficial, because we are almost a layman. Myself is a layman, or most of the audience are not idea. So if you communicate the paper, you will get comments. These comments are very much important for the researcher. It is my suggestion.
Sir, hopefully in the next week, we'll communicate his first paper so far as experimental part is concerned.
I wish the best luck of
Thank you, sir.
Supervisor and the student.
Thank you very much, sir.
Maybe.
Research criticism is always good. Always good.
Yes, Kaustav, I think you can stop presenting. So, moving on to our next participant. Our next participant is Payal Loshkar, Department of Physiology, Vidyasagar College. The topic is work-related musculoskeletal disorders among fireworks workers in West Bengal.
Mona Lisa, hopefully Payal is not from Vidyasagar College. Carefully read it.
Actually, it is written there, so I have actually... I have just copied
Biological
it.
Sciences, Alia University. She is from Biological Sciences, Department of Biological Sciences, Alia University.
Okay. Okay.
It was
thank you for the rectification. Actually, it was written there, so I just copied it from there. Anyways, I would now like to request Payal to start her presentation.
Yes, ma'am. Good evening. Myself, Payal Loshkar, MSc Zoology, PhD scholar of Alia University. I am working under the supervision of Dr. M.D. Zakir Hossain, Assistant Professor, Department of Biological Sciences, Alia University, and co-supervision of Dr. Shubhadeep Ganguly, Assistant Professor and Head, Department of Physiology, Vidyasagar College. Today, my topic is work-related musculoskeletal disorders among fireworks workers in West Bengal, India. Musculoskeletal disorders are disorders of muscles, nerves, tendons, joints, cartilage, and spinal discs. Work-related musculoskeletal disorders significantly connect with the work environment and performance of workers in the field of their profession. Now the question is why we have selected fireworks industry and fireworks workers in West Bengal as our research work. We have chosen fireworks industry and fireworks workers in West Bengal as our research topic because there were no research work has been done on the fireworks workers in West Bengal till now. Though some research work has been done in Sivagashi, Tamil Nadu, but it was in the field of economic, not in the field of occupational health or any other musculoskeletal disorder related to fireworks workers. Beside the other industry in West Bengal, fireworks is also a popular industry in West Bengal. It is very old and cottage industry. In West Bengal, Howrah, Lilua, Bojboj, South 24 Parganas are famous for fireworks. Our study is in the area of South 24 Parganas, Champati. The largest fireworks hub of West Bengal is situated in Champati, South 24 Parganas, which includes around 19 villages across 4 Gram Panchayat. Approximately 500 workers are involved in this profession, which involves 800 trades directly with an estimated revenue collection of around 25 crore per annum. Here are some pictures of fireworks industry in Champati. The objective of the study is to assess the work-related musculoskeletal disorders among male fireworks workers in West Bengal, India. Here we exclude female workers from the study because maximum female workers in this study work in the packaging department, not affiliated with the manufacturing department. We selected 152 male fireworks workers who are physically and mentally healthy as our experimental group. And we also select 100 workers as control group who don't attach with the field of fireworks and don't involve in the awkward posture like firework workers. We divided we divided both groups into four subgroups according to their age. Group A 20 to 29 years old, Group B 30 to 39 years, C 40 to 49 years, and D is above 50 years old. The workers of control group work in the field of mainly police, shopkeeper, teacher, different office, etc. We used a modified Nordic questionnaire for this study. The questionnaire was represented in the form of multiple choice. The subjects were informed about the objective of the study and all had their consent. And the questions are divided into two groups on the basis of their general information of workers and pain of discomfort body part. For analysis of working posture, we use Reba and Rula method. After identifying by Reba and Rula method, here was shown the risk level of posture by a 10-point scale, where 1 represent no risk and 10 represent high risk. Here, the process of producing fireworks. First, they mixing the raw and dust materials of fireworks, and then cutting the paper and metal parts of fireworks. After that, they filling the mixing dust into the cutting paper of fireworks, and then shaping and rolling the papers filled with dust. Now, this is ready for drying into the sunlight, and after all, at last, it's ready for packaging for sale. Demographic data of workers is mandatory for the study of occupational health. It also connected with the musculoskeletal disorder. That's why we collected the demographic data of fireworks workers and control group. The data of age is not significant. Others are significant. And workers work 8 hours per hour, and working day per week is 6. This is the table of comparison of discomfort in different body parts between fireworks workers and control group. Here, the subgroup 20 to 29 age, the maximum workers suffer from neck pain, and according to 30 to 39 years subgroup, the lower back portion affected maximum among the fireworks workers. In the subgroup 40 to 49 age group, the fireworks, the prevalence of lower back pain is showing here high, and 50 and above group subgroup, the lower back and knee portion, lower back and knee pain showing high. Here we also see the types of injury between fireworks workers and control group. This is the table analysis of working posture of fireworks workers by Reba and Rula. On the basis of these pictures, we draw the ball and stick models, and then we calculate Reba and Rula score. And we have also mentioned here 10-point scale score. Among all of these, awkward posture, fast posture, mixing the dust of fireworks occurs in high risk, and action will must be taken necessary soon. Other posture risk level are medium, and Reba and Rula score is quite low. For further study, we test some radiological tests, and here are some radiological pictures of body parts showing musculoskeletal discomfort among fireworks workers. And now conclusion is work-related musculoskeletal disorders are rapidly increasing among workers working in both large-scale industry as well as in small-scale cottage industry in India. In our present study, maximum work-related musculoskeletal disorders were found out to 71.73 fireworks workers, predominantly involving the lower back. However, pain and stiffness were also reported in upper back, neck, wrist, elbow, knee, and ankle. In this present investigation, increased prevalence of work-related musculoskeletal disorder were also reported with advancement of age. Recommendation: provision of backrest with support is strongly recommended. Ergonomic stools which height will be 2 cm lower than elbow length of workers who mixing the chemicals, and 2 cm higher than elbow length of workers who involved in packaging of fireworks are recommended to reduce the musculoskeletal discomfortness of shoulder, neck, and hand. Here are some pictures of mine during study. The funding, the entire work funded by University Research Fund allocated to Alia University and Swami Vivekananda Merit Means Scholarship for PhD, Government of West Bengal. I would like to express my special thanks of gratitude to my respected guide, Dr. M.D. Zakir Hossain, and as well as my co-guide, Dr. Shubhadeep Ganguly, who gave me the golden opportunity to prepare this presentation on this topic, which helped me in doing a lot of research, and I came to know about so many new things. I am really thankful to them. I also would like to acknowledge and thank the fireworks workers of Champati who kindly participated in this research along with all others who provided their help in any form. Secondly, I would like to thank my parents and friends for helping me a lot in finalizing this within limited time frame. Finally, it is my pleasure and honor to acknowledge Alia University for allowing me this to contact this work. This is some reference. Thank you.
Thank you, Payal, for your nice presentation. But I like to see you. I am not seeing you. You are not visible.
Hi. Yes, sir. Actually, I am presenting from phone, and that's why my video is off.
Okay, it would have been better if it would have been better if we would see you in the one hand and other hand listen your lecture.
Yes, sir. Okay. Yes, sir. Yes.
Okay. Now the session is open. Please ask the. Please, if you have any question, ask the Payal. Thank you, Payal. Thank you, Payal.
Can you hear me? Hello.
Dr. Bandwad, Professor Bandwad, please.
Yes, yes. Payal, very nicely presented, you know.
Thank you, sir.
And musculoskeletal disorder in such type of industries, you know, are a very common problem today. That is why, you know, Government of India through Department of Health has, Professor Chandra knows also, that they have prepared a musculoskeletal disorder section, okay? And because, you know, many diseases finally arise from the musculoskeletal disorientation problem, okay? And to that end, your study, whatever data, it will take time to develop the data, develop the project and have the publications and other things. But, you know, is very appreciable, very much appreciable, because in India as a whole, we have very minute amount of data. Very few scientists in India, I can count in my hands, who work on musculoskeletal disorders. And because people need solution to their musculoskeletal problems. So to that end, work hard, work nicely, and I hope some good data will come out. Thank you very much.
Thank you, sir.
Any questions?
হ্যালো স্যার, আমি সন্দীপ বলছি স্যার।
Please.
Thank you, Payal. It's a very good presentations. Just I have some suggestions for you from your radiological study you present here. I think when you present the radiological studies, you must be mention what type of changes in your pictures. From your pictures, I can't understand what type of changes are actually going on through your radiological study, but you can't mention or you cannot recognize through this particular picture. So in future, if you present that type of radiological image, then I think you should be point out that this is the control pictures and this is the affected pictures, affected persons who are basically going some musculoskeletal disorders or musculoskeletal changes are actually going on. So that is my suggestions for you for your better presentations. Thank you.
Yes, sir. Yes, sir. Thank you for your suggestion.
Any questions? Payal, the suggestion that has been made by Professor Debashish Bandyopadhyay is very, very important, because the data that you have in your hand should be communicated to the particular authority or their information and taking necessary action. Secondly, Dr. Sandeep Sinha has also given good suggestion. When you are comparing the two data on radiological basis, then you should clarify, because you are well aware about the changes, but we, the common audience, are not capable of observing this. And now you will go through it, because both the suggestions are very, very important. My question is that those people who are working in fire fields, their posture definitely affected. In any occupation, postures will be affected, but they are more exposed to different types of chemicals.
Yes, sir.
There are lots of chemicals they are exposed during that period, and these chemicals may have many much more effect. So what are these? You are doing these studies is very good, but simultaneously you will try to do. You must have that idea that the chemicals they are exposed through inhalation, through eyes, through nose, through mouth, through hands, they are inhaling. So this may cause very adverse effect, and this may have carcinogenic effect. So my suggestion with Professor Debashish Bandyopadhyay and with Dr. Sandeep Sinha, I have the suggestion that in future studies on your subjects, please go through this, that whether they are affecting, besides musculoskeletal disorder, whether there is any abnormality, your attention will be also in that direction. It is my suggestion. So if there is no question,
thank you.
Because yes.
Sir, may I say something?
Yes, please.
Actually, so far as Payal's work is concerned, she has already done few hematological data, hematological parameters.
But she has not presented yet.
No, first of all. Second thing, liver deterioration is there. Okay. We have identified two biochemical parameters. Then certain toxicological effects like xenotoxic effects. It has been found along with some immunological changes, along with lung function and chest X-rays, because there are TB patches are there along with some sort of immunological changes that we have already done. We are now communicating because this paper has already deposited in biological property. So that is why we are presenting over here. And in the due course, within 2 or 3 months, hopefully we can able to communicate these areas also. And moreover, psychological parameters, few psychological parameters, especially during the course of COVID pandemic, it will severely affect along with some sort of endocrine factors. So that has also already been considered, and we have collected blood and doing the assay. It is going on.
Very good.
Thank
Very
you.
good. Very good. Very good. Thank you, Payal, for your presentation. Please go ahead with the suggestions. And as Dr. Ganguly has suggested, you are doing this work, but you have not presented yet. So my suggestion is that this part is also very, very important, like the musculoskeletal disorder. Okay, thank you.
Thank you, sir.
Sir, can you move to our next?
Yes, of course.
Okay, thank you, sir. So our next participant is Krishna Kumar Das of Medical Microbiology, Department of Biotechnology and Bioinformatics, Sambalpur University, Orissa. The topic is bioefficacy of actinobacterial metabolites, a review.
Hello, ma'am. Am I audible?
Yes, you are audible.
And visible also?
Yes.
Okay, ma'am. Thank you. Am I PPT is visible, ma'am?
It's coming. Yes, I think. Yes, it's now there.
Okay, ma'am. Thank you. Good evening, all. Good evening, respected sir, ma'am, and the participants, and also the audience who are listening to me. Today, my topic is bioefficacy of actinobacterial metabolites, a review. This is the topic which I have just started in my PhD work for few days before under Sambalpur University. So this is just an introduction and just a few information about actinomycetes. Now, an introduction to actinomycetes. What is actinomycetes? Actinomycetes belongs to the kingdom bacteria, and that's also in class, also in order Actinomycetes, and in family Actinomycetes. This is a prokaryotic bacteria, and also gram positive and gram positive in nature. Actinomycetes contain filaments and filamental structure that should be in a network manner. For this, its appearance is like its appearance is like a rod shape, or in filamentous, or in networking structure. If it is rod shaped, then if no filament is present, then it will be characterized as a bacteria section. Because if Actinobacteria is having filament, then it shows fungal mycelia. So it saw some fungal characteristics. Morphologically, Actinobacteria is fungal in character, but in cell and cell organization and intercellular structure, it is purely a bacteria. Now, Actinobacteria is highly G plus C, means guanine plus cytosine percentage highly content. That should be approximately 60 to 80%. In microscopic, in optical microscopic or microscopic observation, Actinomycetes is approximately 1 to 2 micrometer in diameter. or its filament is approximately 5 to 10 micrometer. That should be between these. Yeah. Now the cultural characteristics. As my preliminary work means this is at this stage I will do my PhD work. Here the left, the culture of the actinobacteria. This is my culture, and this is the optical microscope visualization of actinobacteria. Now this is the description of what I said before. This is a description of actinobacteria. Now its habitat. Habitat means it is present in mostly in soil and approximately small amount in marine. So its habitat is both terrestrial and marine. Now actinobacteria are the reservoir of antibiotics. The antibiotics produced by actinomycetes include all important drug classes used in clinical today, such as beta-lactams, tetracyclines, macrolides, aminoglycosides, glycopeptides. Virtually 80% of world's antibiotics are known to come back from actinomycetes. Thus, today, almost 80 years after Salman Waxman introduced the genus Streptomyces and then the first antibiotic produced from actinomycetes, that is actinomycin, that was isolated from actinomycetes. These bacteria still represent a threat just of the identification of new antibiotics. Now, beta-lactam. Beta-lactam is the most prominent class in beta-lactam is penicillin, where among the first medication to be effective against many bacterial infections, casually in Staphylococcus and Streptococcus. Antibiotics in penicillin class are one of the most commonly prescribed antibiotics and include many individual medications, such as these are those underlined part are examples of penicillin antibiotics. This is the structure of penicillin, chemical structure of penicillin, and R is the variable group. Tetracycline antibiotics are well known for their broad spectrum of activity, spanning a wide range of gram-positive and negative bacteria, obligate intercellular bacteria, as well protozoan parasites. The first tetracyclines were natural products derived from the fermentation of actinomycetes. Chlorotetracycline, produced by Streptomyces aurofaciens and marked as auromycins, was first reported by Benjamin Duggar in his laboratory in 1948. Tetracycline are broad-spectrum agents exhibiting against a wide range of gram-positive and gram-negative bacteria. The underlined parts are the examples of tetracyclines, and the picture is the chemical structure of tetracyclines. Macrolides are very active against Staphylococcus, Streptococcus, and Diplococcus, gram-positive bacteria, and among gram-negative cocci also. Macrolides antibiotic class methimycin produced by Streptomyces species. In the main representative of 12-membered macrolides with only a few other compounds in this class. Erythromycin is the best known member in 14-membered group and was isolated from the Streptomyces erythrosis and or Erythrobacter species. Oleandomycin and lancomycin and picromycin are also important member of this group. The last group comprises the 16-membered macrolides, with the most important members being tylosin, carbamycin, and nidamycin. These underlined parts are the examples of macrolides. This is the structure of macrolides. This is the chemical structure of macrolides. Now, aminoglycosides. Aminoglycosides that are derived from bacteria Streptomyces genus. Examples of aminoglycosides. These are the classes of antibiotics that belongs to the aminoglycoside group. Aminoglycosides displays bacterial concentration dependent killing action and are active against the wide range of aerobic gram-negative bacillus. They are also active against the Staphylococcus and certain mycobacteria. This is the chemical structure of aminoglycoside. This is derived. The structure of the antibiotics are derived from the Google image. Glycopeptides. Antibiotics have been a key weapon in the fight against the bacterial infection for over half a century, with the progenitor, the vancomycin, teisoplanin. This and the others are the classes of antibiotics. These classes of antibiotics are used in the treatment of infection caused by gram-positive pathogens, especially methicillin resistant Staphylococcus aureus, MRSA, and the gut anaerobes, Clostridium difficile. This is the chemical structure of glycopeptide. Now, these are the reference where I get the information from the literature survey. Thank you. Thank you all of you for listening to me, and a special thank to Dr. Subodhi Ganguly sir for inspiring me to share my topic in the webinar.
Thank you, Krishna, for your nice presentation. If the audience has any question, please ask. Any questions? Whether any plan for publication of this review work?
It is just a start of my PhD work. This is just a startup. I have just isolate the bacteria from the habit. I don't. It will proceed. It is my ongoing work. As it is my ongoing work, after my completion of my work, then I will proceed for the publication.
Okay. Okay. Any questions? Very good. Nice presentation.
Thank you, sir. Thank you. Am I quite, ma'am? Or
Madam,
am I get offline from video?
Yes.
Hello, ma'am. I'm not listening. Ma'am, your sound is not clear, ma'am.
Right. Connection will be interrupted. Okay.
Okay. Hello.
Hello.
Yes, sir. Ma'am, am I quoted from the video, ma'am? Yeah, I will be.
Yes, Krishna, you can.
Okay, sir. Thank you, sir. Thank you, all of you, sir. Thank you, Ganguly, sir, for inspiring me.
Thank you.
Sir, now I am calling the next participant.
Yes, of course.
Adrita Banerjee is pending. Mona Lisa, Adrita Banerjee is pending.
Okay, so we will now call Adrita Banerjee, our participant. She is a junior research fellow from Vidyasagar College, Department of Physiology, working as a DST project fellow. And her topic is emergence of folic acid, a monosaturated fatty acid as an antioxidant. A brief insight. Adrita, are you comfortable in sharing your screen, or should I share the screen? Do you want to try once? You are muted, Adrita.
Shall I try once?
Yes, of course you can try.
Okay. Is it visible?
Yes, it is visible now.
Okay. A very good evening to all present over here. So today my topic of presentation is emergence of oleic acid, a monounsaturated fatty acid as an antioxidant, a brief insight. I am working under the supervision of Dr. Adrita Banerjee and Professor Debashish Bandopadhyay from Vidyasagar College and University College of Science, Technology and Agriculture. So I will talk a little about oleic acid, the properties of oleic acid. So you all know that the oleic acid is a monounsaturated fatty acid with 18 carbon in its chain, and it has a single double bond at its 9th carbon position. So the presence of this single bond has made the molecule some exception, gives the molecule some exception by making it less prone to oxidative attack. So olive oil is the main dietary source with almost 70 to 75% of oleic acid content. So we all are well aware about the cardioprotective actions of olive oil. But what is the major contributory molecule behind the cardioprotective action of olive oil? Why it is being taken from long? Why it is being recommended from long as a remedy for coronary heart disease? So researchers has proceeded involving this question, and researchers have found that oleic acid content, high oleic acid content of olive oil is the main cause behind such cardioprotective action, such blood pressure lowering action of olive oil. So not only olive oil, many other sources are there like canola oil, peanut oil, corn oil, avocado, etc. And now researchers have found that different parts of several plants, different parts means stem, bark or leaves of various plants have been identified as a rich source of oleic acid. And here Terminalia arjuna is worth to mention. Aqueous bark extract of Terminalia arjuna has been identified as a rich source of oleic acid from our laboratory. And many researches have been carried out on the protective role of oleic acid, Terminalia arjuna and olive oil, but the underlying mechanism behind its cardioprotective or tissue protective action remained masked for a long time. But now with the proceeding of research, it has been found that antioxidant, anti-inflammatory as well as anti-apoptotic properties of oleic acid is the principal factor behind such protective action of oleic acid towards several tissue system and cellular system as well. So now I would like to share some research articles that have been helped to establish oleic acid as an antioxidant. Here you can see the first one. Here Sola et al. Have showed that oleic acid prevents the formation of T-bars, that is a biomarker of oxidative stress, the increase of T-bars in oxidative modification of HDL, that is high density lipoprotein, and it further prevents the formation of atherosclerosis. So it is clear that when oleic acid is in high content in the high density lipoprotein, then the chance of occurrence of atherosclerosis is low. And another one in a similar way, Perona et al. In 2005 has shown that upon 3-week supplementation with high oleic acid oils, lipid peroxidation of rat hepatic microsome has been prevented. So lipid peroxidation, that is the main the contributory factor behind generation of oxidative stress, it has been prevented by oleic acid. Here in this paper, the stress has been induced by the combination of ascorbate iron and NADPH. Coming to the next, here it is, you can see a very important one, that in rat, diabetes and both diabetes and hyperlipidemia has been made in the rat model. Streptozotocin has been used for reduction of diabetes, and cholesterol has been fed to those rats to create the model of hyperlipidemia also. And definitely as an obvious outcome, there was disturbances in total lipid content, lipid profile, and as well in antioxidant enzyme levels also. You can see here the GSH level has been disturbance in GSH level, and which cumulatively leads to cardiovascular complications. But whey consumption for 3 months has been successfully ameliorated those situations and keep the cardiovascular in proper health, cardiovascular tissue system in proper health. Now it's a case control study. You can see here Alcoberry et al. Has found an interesting thing that significant lower intake of MUFA and specifically oleic acid is there in diabetic retinopathy patients. That is, there is an inverse relationship they have found in diabetic retinopathy when in patients there was lower intake of monounsaturated fatty acid and specifically oleic acid. Now come to this article that has been published in 2019 in Life Sciences. What Mishra et al. Have shown, they have used cardiac mitochondria as their working medium. And we know all that mitochondria is a major seat of Ross generation. Mitochondria in-house all metabolic enzymes, all enzymes involved in oxidative phosphorylation that all leads to energy generation. And so stress generation is inevitable in mitochondria. And they have chosen cardiac mitochondria as their model. And definitely adrenaline bitartrate produce enormous stress in the cardiac mitochondria. That has been assayed by several stress biomarkers and assay of antioxidant enzymes as well, which all leads to alterations in the mitochondrial morphology and intactness. And when intactness is changing, we can say here that there will be leakage of several ionic materials from mitochondria into the cytosol, which ultimately lead to alteration in the mitochondrial membrane potential. But what oleic acid did, it at the first hand prevents the occurrence of oxidative stress, as they have assayed from biomarkers and antioxidant enzyme assays, which leads to giving protection, preservance of mitochondrial morphology as well as intactness. And a very important thing to mention here in this article, authors did an isothermal titration calorimetric study where they have shown that oleic acid bind with high efficacy with adrenaline bitartrate. So from this, we can say that oleic acid not only bring back the normal situation of cardiac mitochondria that have already been worsened by adrenaline bitartrate, but also it prevents the worsening situation to create by adrenaline bitartrate. It prevents the worsening function of adrenaline bitartrate. It gives protection to cardiac mitochondria for its preservance. So now let us focus a little on its lipoprotective action. We are talking about cardiac protection. Now Jianrang Guo et al. Have shown some data about lipoprotective action. In the in vivo part of the study, ischemic reperfusion injury in mice have been induced, where oleic acid when oleic acid was given intragastric way at the particular dose of 250 milligram per kg body weight for 2 weeks, liver injury was completely ameliorated and consequently there was complete preservance of the hepatic enzymes. And as the in vitro part of the study, HepG2 cells were exposed to hydrogen peroxide and there was increase in apoptotic cell number, as you can see here, and decrease in cleft caspase 3. That means the apoptosis was there. Apoptotic cell number, they were measured through measuring the annexin 5 positive cells. And cleft caspase 3 was measured by doing Western blot. And oleic acid pretreatment of HepG2 cells completely prevented the occurrence of the apoptosis. So oleic acid not only acting as an antioxidant, it is also acting as an anti-apoptotic agent also. In a similar way, another work is presented here. When HepG2 cells was exposed to palmitic acid, there was increase in caspase 1, NLRP3, IL-1 beta TNF alpha, as well as increase in CHOP BIP was observed. So, in one hand, there was apoptosis induction, there was inflammation, as well as there was ear stress also. And weight treatment for 24 hours had ameliorated all those situations. So it brings back the situation in normal in the HepG2 cells. And as the in vivo part of the study, when Sprague-Dawley rat were fed with high-fat diet for 16 weeks, liver steatosis was there and inflammation was there. Inflammation was measured by pro-inflammatory markers. And when olive oil was replaced, when the high-fat diet fed to those rats were half replaced with olive oil, there was no liver steatosis and inflammation has been observed in those rats. That is, lipotoxicity has been ameliorated by oleic acid. Now come to the next one. Bhattacharjee et al. In the year of 2019 presented a work in published a work in Life Sciences where 10 mg per kg body weight when given to the rat orally, it prevents the generation of ROS and subsequent mitochondrial morphology and intactness has been preserved. So here the stress inducer was used cadmium. We all know we are very much with the stress attack of the cadmium, which is abundant in our environment. So it makes deleterious actions on cardiac and hepatic tissue, leads to inflammation, leads to ROS generation. But oleic acid, with its efficacy, prevent that ROS generation and subsequently protect and preserve mitochondrial intactness and mitochondrial membrane potential alteration, as we have seen earlier. But the new thing is, it has presented that it prevents cytochrome C release. Oleic acid prevents the release of cytochrome C from mitochondria. Cytochrome C release is a very primary and marked marker of the progress of cells towards apoptosis. So apoptosis is being ameliorated by OA and definitely through antioxidative mechanism. As it is in first hand, it is preventing the ROS generation. So in the similar way, similar dose of oleic acid has been used for in this study where gastric tissue of Wister rat was used by Mishra et al. And it has been published very recently in Helion in this year. Adrenaline bitartrate is used as a stress inducer, and 10 mg per kg body weight when given to the rats orally, it prevents the generation of oxidative stress. It conserves the mitochondrial surface topology, but not only the structure, it preserves the metabolic enzymes as well. Mitochondrial metabolic enzymes, as I have told earlier, that it is very important, and non-competitive inhibition of metabolic enzyme was there when adrenaline bitartrate, when gastric tissue was exposed to adrenaline bitartrate, but oleic acid successfully ameliorated those situations and bring back the activities of those enzymes in normal condition. Now, I would like to share a brief about this article that has been published in toxicology reports from our laboratory in 2020. Here we are talking about cardioprotective and lipoprotective actions of oleic acid, but here the hematoprotective action is also evidenced in our study, where red blood cell was exposed to phenylhydrazine. We all know phenylhydrazine is highly hematotoxic. It generates enormous ROS. It completely malfunctioned RBC, destruct its complete structure, its biconcave shape. And when oleic acid was given at the specific concentration 0.06 micromolar per ml that we have standardized in our laboratory, it prevents the generation of oxidative stress. And along with prevention of stress generation, it preserves the metabolic alteration that has been happened when phenylhydrazine was intoxicated into RBC. As you can see here, the stress generation has been completely ameliorated. You can see the DCFDA. This is the ROS generation measurement of total ROS. This one is measurement of superoxide anion radical, and this one at your right side, it is the hydroxyl radical content measurement. So in all three parameters, there was a significant increase in the phenylhydrazine group, phenylhydrazine treated red blood cell group. We are used both red blood cell in this study. But oleic acid treatment at the fourth column of every figure, oleic acid co-treatment of red blood cell and oleic acid co-treatment brings back the situation sometimes nearly to the level of the control, and sometimes gives almost 50% of protection. ROS level, superoxide anion radical and hydroxyl radical has been decreased in a significant way from the phenylhydrazine group when it was co-treated. So oxidative stress amelioration of oleic acid property is there also in red blood cell. Now, metabolism of red blood cell is a very important thing since RBC depends on the energy generation. It has to traverse through narrow capillaries. It has to carry oxygen in every parts and corners of our body. And for its flexibility, it needs high amount of energy. And so it depends on glycolysis and other metabolic pathways. So here you can see the hexokinase, PFK, aldolase, all enzymes. In all enzymes, the activities has been drastically declined in phenylhydrazine treated group, which has been which has been ameliorated in the phenylhydrazine and oleic acid co-treated group. And in the lactate dehydrogenase group, phenylhydrazine treated RBC showed a significant rise in their activity, and it brings back to normal in the oleic acid co-treated group. And G6-PDH activity, you know, G6-PDH, the important enzyme of our pentose phosphate pathway, it has also the same thing is reflects in the G6-PDH activity also. These all go for the functional side of the red blood cell. But if we focus, when we have focused on the morphological side of the red blood cell, there was also change. There were also changes in RBC diameter and in RBC roughness, and these data were obtained through atomic force microscopic study of red blood cell. So a slight change, since the structure and function of RBC is highly correlated, a slight change in structure leads to complete malfunctional RBC. So RBC diameter has been decreased, and RBC roughness, which is very important, that has also been decreased in phenylhydrazine treated group, but significant alteration like similarly to the control group has been observed in oleic acid co-treated group at the concentration of 0.06 micromole per ml. And to substantiate this study, we did a facts study where you can see here in the control group and in the treatment group, by actually biconcave shape used to give two population, used to show two population in fact study. So here control group, oleic acid group, that is our negative control group, as well as our co-treated group, shows completely defined two populations are there. But you can see in the treatment group, in the second one, phenylhydrazine treated group, one population is there. So it means that there was complete destruction of the biconcave shape of the RBC, which is highly needed for a red blood cell to reach to different parts of the body, to traverse through the narrow capillaries of our vessels. So here I would like to conclude. So I would like to express my heartfelt gratitude to West Bengal DST for giving financial assistance for proceeding the project work, and my sincere gratitude to my supervisors, Dr. Andrita Chattopadhyay and Professor Debashish Bandyopadhyay, for their immense support for their encouragement at every steps of my work. And obviously, I'm grateful to my family members and my fellow mates for supporting me till my journey. And I am very much thankful to all members of this webinar for allowing me for presenting my work. And thank you all.
Thank you, Indrila, for your nice presentation.
Sir Adrita.
Okay. Thank you, Indrila, for your nice presentation. Now I request the audience to put up their questions. Please.
Very smart and confident presentation.
Yeah.
Thank you.
So can I ask you a question, sir? If your condition go on with the
If anyone have no comments, then I can comment on Adrita. The presentation of Adrita is not only confident, but his approach is also very good. Through paper, through published articles, he raised his problem. And he showed that how oleic acid is important. He is not only confident, his approach is very nice. And I have no question, but I just like to ask one question. That from your laboratory, melatonin has been established as a potent antioxidant. This time I am listening from you, Adrita, that oleic acid is also an antioxidant. In rank, what is most potent? What is most potent one? If you compare between the two, because that work is also from your laboratory, and this work is also from the same laboratory of Professor Debashish Bandyopadhyay.
Yes, sir.
If you are asked to answer that which one is more potent in what respect, if you give us some knowledge, that will be beneficial for our social life.
Sir, comparative study of melatonin and oleic acid is very much welcome. I think no such study have been done yet, but melatonin has already been established as a potent antioxidant, and we are trying to establish oleic acid. Trying to establish, and many, sir, you have seen that many of the papers many of the papers are from our lab, and we are trying to establish it, to see.
No, we have already, it will be established because the paper that you have published is a good reputed journal. But you are avoiding my question, or you are going away from my question. I am asking that if you are asked in the present situation that which one is more stronger, then what you will say. I want to listen from you in that direction.
No, sir, no comparative study is there, so I can't without studying this. Sir, if anyone then, or if we are, then it will be worthy.
Okay, thank you very much for your excellent presentation, excellent publication, confident presentation, everything. You are quite matured now. Okay, thank you. Wish you all success.
Thank you, sir. Thank you so much.
How many presentations are left, Mona Lisa?
Sir, in this session, only one presentation is left.
Okay.
Should we move on?
The number of participants has been decreased from 80 to only 31.
Sir, can we move on to our next participant? Or?
Yes, of course.
Okay, sir. Thank you, sir. So, our next participant is Romit Mojumdar from Department of Physiology, Vidyasagar College. And his topic is protective effects of melatonin against ketorolac-induced gastric ulcers.
Am I audible to everyone?
Yes, you are audible.
Okay. Are the slides visible?
Yes.
Okay. Good evening, respected teachers and participants of today's meet. I am Romit Mojumdar, currently working under the supervision of Dr. Venvila Chattopadhyay and Professor Debashish Bandyopadhyay. Today, I would like to talk about the protective effects of melatonin against ketorolac-induced gastric ulcers. So let us start without any further ado. So in the 21st century, in this rat race of life with improper diets and enhanced dependency on fast food, gastroduodenal anomalies have become a massive concern for physicians and researchers across the world. Gastric ulcers today make up a major bulk of all gut-related disorders. According to a study, approximately 4.6 million people are affected by gastric ulcers in the US annually. Apart from improper lifestyle and unbalanced dietary habits, there are two major causative factors that are responsible for the onset of these ulcers. One is an uncontrolled H. pylori infection, and secondly, overuse of NSAIDs. Although there are numerous fine-targeted antibiotics to control the H. pylori infections, we lack proper interventions to control NSAID-mediated ulcers. Currently, antacids and proton pump inhibitors are commonly used to deal with such conditions. However, these drugs are not sufficient to combat with NSAID-mediated ulcers, and this is mainly because these are either not effective enough or pose some serious health issues upon long-term use. So, to look for an ideal remedy, we must first understand what NSAIDs are and how they function. Well, NSAIDs, or non-steroidal anti-inflammatory drugs, are the most commonly consumed pain medications available over the counter, both with and without prescriptions. Apart from reducing pain, they are also known to control inflammation and reduce fever. Functionally, NSAIDs are classified based on their selectivity for inhibiting the different isoforms of the enzyme COX or cyclooxygenase. These are non-selective COX inhibitors, preferential COX-2 inhibitors, COX-2 inhibitors, analgesics, antipyretics with poor anti-inflammatory actions. Now, how NSAIDs work? NSAIDs mostly block the catalytic conversion of arachidonic acid to prostaglandins and decrease the presence of PGE2, which is implicated by aggravating ulcerative wound in the gastric epithelial lining. As far as existing literature is concerned, it is evident that COX-1 inhibitors impart better pain-relieving action than COX-2 inhibitors. Seemingly, selective COX-1 inhibitors are also greatly associated with an increased risk of forming gastric ulcers. For our study, we have chosen ketorolac promethamine, which is a traditional NSAID that is tagged as one of the best analgesics and is currently the most effective, the best COX-1 inhibitor available in the market. This is a comparative representation of COX selectivity for different NSAIDs. According to our study, it was even stated that ketorolac at certain doses outruns morphine and many other opioids in combating pain. Yet, there have been instances where ketorolac, despite its high efficiency and efficacy, is either avoided or discontinued after 5 days of use because of its severe ulcerogenic properties. Now, to look for a remedy for this, we must first understand how ketorolac induces these ulcers in the GI tract. Currently, there are 2 plausible pathways that are either by Ross generation, which directly tampers with the epithelial membrane and results in the formation of ulcers, or secondly, by selective COX-1 inhibitors, they downregulate the synthesis of PGE2, which is directly related to the synthesis of mucus responsible for protecting the epithelial lining of the stomach from corrosive action of HCl. While we were extensively looking for a cure, we came across melatonin, an indole amine of pineal origin having potent gastroprotective effects with greatly pertains to its high antioxidant and free radical scavenging activity. Apart from pineal glands, significant high levels of melatonin were found in the gastrointestinal tract, which also is associated with an elevated expression of MT1 and MT2 receptors in the gastroduodenal tissue in an ulcerative event. Hence, melatonin can be prescribed as an alternative or along with the conventional triple therapy regime, thus substantially reducing the risk of gastric ulcers, thus enabling physicians to use NSAIDs like ketorolac more conveniently. Now, melatonin supposedly preserves tissue prostaglandin levels to reduce acid secretion, elevate mucus and bicarbonate secretion, enhance blood flow, thus offering gastroprotection. As I have worked with male Easter rats, I have collected approval from the IEC to carry out my experiments. This is my ethical clearance certificate. Now, coming to the experimental design, all experiments were performed on rats, and the rats were kept in the University of Calcutta Animal House. The first experiment was aimed to determine the best effective dose of ketorolac to induce ulcers. And to do so, we divided the animals into 4 respective groups. The first group received water and was tagged as control. The remaining were fed with different doses of ketorolac. The doses were 12.5, 25, and 50 mg per kg body weight. After treating the animals, they were observed closely for 4 hours and then sacrificed and checked for ulcers, and few basic parameters were also tested to determine the best effective dose of ketorolac. After determining the best effective dose of ketorolac, we proceeded with the second part of our experiment, where we again distributed the animals into 4 different groups, namely control, ketorolac, melatonin, and protection. The ketorolac group received the best effective dose of ketorolac that we determined from our last experiment, and the melatonin group received 60 mg per kg body weight, which was earlier determined from a previous study from Debashish Bandyopadhyay. Melatonin was fed 30 minutes prior to the treatment with ketorolac. Post-sacrifice, the parameters were tested, and the parameters tested were mean ulcer index, oxidative stress biomarkers, and antioxidant enzymes activity. Now, coming to the result part. Now, here we can see that mean ulcer index, the number of ulcers have increases linearly and significantly with the increasing doses of ketorolac. And when we have tested for the oxidative stress biomarkers in the stomach, we have seen that a linear and significant change in GSH and LPO was also observed. And coming to the antioxidant enzyme activities, there are similar results were also found where CUZ and SOD and gastric peroxidase yielded similar results. Now, coming to the second part of our experiment, in mean ulcerative index, here we can see that in the stomach, the number of ulcers have decreased significantly when compared to the ketorolac group, and in the melatonin and ketorolac group, the number of ulcers are very less. And we can say that protection is being established. And when we are testing for oxidative stress biomarkers, the enzymes like the reduced glutathione levels and LPO, we have seen that there was a distinct protection was observed in the melatonin and ketorolac group in both GSH and LPO. And in case of CUZ and enzymes like CUZ and ZNSOD and GPO, protection was also observed there. So in conclusion, we can say that from our findings, so far, ketorolac has severe ulcerogenic effects in the stomach post 4-hour treatment, which further is reversed with melatonin pre-treatment. However, this requires further experiments to validate our findings. Accordingly, we are planning to carry out more extensive experiments to determine the different roles by which melatonin and ketorolac can be used together, and they can be used more different NSAIDs like ketorolac can be used more easily, conveniently, and we hope that this will bestow us with a better understanding of the development of gastric ulcers, as well as enable us to use potent NSAIDs like ketorolac more conveniently. These are my references, and I would like to show my heartiest gratitude to Dr. Venvila Chattopadhyay and Professor Debashish Bandopadhyay for their valuable guidance. I would also like to thank the chairperson, the convener, and all the respective members of the organizing team for this international webinar. And last but not the least, I would like to thank my friends, my family, and my lab mates for helping me immensely for this work. Thank you.
Thank you, Rohit, for your excellent slides and nice presentation. The slide that you provided or that you shown is very good. Now I request the audience to put questions to Rohit.
Sir, can I ask a question with your permission, Bornali here?
Yes.
Romit, thank you. The presentation is really very crisp and lucid. And I have just not a question. I want advice from you as expert of this field. You are expert of this field. If a person have previous experience of gastric ulcer and prone to gastric ulcer, what will be your advice through a natural way of taking melatonin in everyday life?
Well, as our initial experiments, we have tested that melatonin is capable of protecting gastric ulcers. But as you are suggesting that if someone is already suffering from gastric ulcers, in that case, they can take melatonin with ease, but that definitely needs clinical trials, further clinical trials, which we have not performed. We have performed as I've said, that all my experiments were done on rat models, and you are talking about human beings, which is a big thing. And before I can suggest any drug, any sort of drug to a person, I must say that there needs rigorous human trials. And
if we want to take through natural sources, so there might be no harm of that everyday routine or life.
Yeah, melatonin itself, I can say it to be nothing can be more natural than the things that are synthesized by our body itself, because melatonin is synthesized in our body only. And I guess you are talking about natural sources, like something from external, right? Like plant extracts or something like that. Yes. If I'm not wrong.
Yes, from the natural sources, natural foods or something.
Yeah, true. I understand. Yes, that dimension can be explored with antioxidant. As I have said that we have already proven that there is a incident where oxidative stress is happening and natural resources can work here. But as I'm working with melatonin, and I'm going to vouch for it only. So in that case, I would say that melatonin is synthesized by our body only, and nothing can be more natural than things that are synthesized in our body. So it's synthesized from our pineal gland, it's synthesized from the gut. Yes, what we can do is, one thing I can point out, that we can increase things in our diets which can promote melatonin synthesis in our gut. Like there are lots of things, protein-rich food, proteins which are rich in tryptophan, we can consume them, and that way we can stimulate the synthesis of melatonin in our body. And in case of natural resources,
what kind of food?
Protein-rich food. I'm going to vouch for protein-rich foods, and there are a lot of even. It's really alarming it's really fun to say that the rice that we consume is very much rich in melatonin. Even the rice that we consume is rich in melatonin. There are a lot of foodstuffs that are rich in melatonin, and the list never ends. But to be very honest, any protein-rich food which has high amount of tryptophan in it is going to be beneficial for our health, and we can assume that it is going to control ulcers.
Okay, thank you for your advice. I'm looking for further resources from you in future.
Thank you, ma'am. Thank you.
Thank you.
Any question? If no question, then I have one question.
Yes, sure, sir.
In the context of Dr. Bornale Raivasu's question,
yes,
he discussed a lot in this aspect. His discussion is very good. My question is that in our body, melatonin is present. It is secreted. And ulcer is also producing our body.
Yes.
In one day, ulcer is not produced. At that time, why melatonin is not capable of protecting that? Melatonin is there, and ulcer is going on.
Yes, yes. I got your question, sir. I understand your question. It's a really interesting question. In one slide, I have said that there has been a study where they are saying that there has been enhanced expression of MT1 and MT2 receptors in the gut, which means that the gut is asking for melatonin to bind to it. If talking in layman's language, okay. So in such situation, we can understand that melatonin is required and it is protecting. But what you are saying is melatonin already is there in our body. It's always there in our body. When we eat food, it is being secreted. At night, from the pineal, it is being secreted. In the gut, it is being secreted. But yet, there are events where ulcerative wounds are happening. So to answer that, I would say only one thing. The melatonin that is secreted in our body is secreted at a basal level. Okay. And ulcer happens mostly in case of external stimuli, like any toxic or any like NSAIDs I'm talking about, or exaggerated H. pylori infection. In such instances, the melatonin level rises. It's true. It tries to combat it. But at some points, it fails because our body also has its limits. It can synthesize melatonin at a limit only. No? We can't push it to beyond that limit. And that's when we are proposing to feed orally exogenous melatonin, which would help the fighters, the melatonin that is fighting already against ulcers in our body, to help them out and cure ulcers more easily. And to answer your question, like in normal cases when ulcer is happening, melatonin is definitely acting and is trying to stop the ulcers. But when the oxidative stress is immense, melatonin is incapable in that case. At that point, the endogenous melatonin is totally incapable of preventing it, and that's when we require an exogenous melatonin. Am I able to answer you?
Thank you. And do you consider melatonin should be considered as a drug now?
I'm bound to consider, sir. I'm bound to consider because in countries like Canada and many other developed countries, they have stopped providing melatonin over the counter. Like you need to provide a prescription to procure melatonin from such countries. So I am bound to consider melatonin as a drug because in the many developed countries there, and when you need a prescription to consume something, it should be considered as a drug.
So what is the method for the measurement of melatonin in our body, in blood?
We don't usually measure melatonin in blood. Usually measure it in serum, in plasma, and we usually measure using kits. I have personally measured melatonin and in rat samples, in rat tissues, gastric tissues, and we have used kits. We have procured kits, and we have used them to estimate the melatonin levels. It's found in some picograms per mg tissue.
Most of the hormones remain present in picogram. Okay, but you are measuring in tissues.
Yes, I'm measuring it in tissues.
Not in blood.
No, not in serum, because there's a reason behind that. When I was working with rats, as I have provided treatment for 4 hours treatment, I have stated that I have used 4-hour treatment, which is a very short treatment, and the ulcers are very prominent in those cases. I have used ketorolac here just as a model to see how ulcers are formed. So in such short time, there are changes in serum parameters. I won't deny that, but it is the levels are very prominent in case of in tissue samples, because and moreover, the second point that I would like to say, that when we are dealing with serum, the melatonin level is also coming from the pineal gland and from other organs. But when I'm targeting gastric tissues, my focus is on the melatonin that is synthesized from the stomach only. So that's why I have measured it in the stomach tissues, gastric tissues.
Thank you. I have lots of questions, but I am not asking you. When I will meet you, then we'll
Sure, sir.
discuss.
Sure, sir. We will have a lot of great discussion.
Okay. Thank you.
Thank you, sir.
Yes, and now to Mona Lisa. Where is Mona Lisa?
Yes, sir. Yes, sir. Yes, with this, we have come to an end of our second technical session. So I now request our honorable chairpersons to summarize and conclude this session with their valuable remarks and advice.
Chairperson means myself.
Yes, sir. Yes, sir. You and ma'am.
Yeah, where is ma'am? This session is entitled Contemporary Innovation and Challenges of Modern Biology Research. In this context, there were a number of young scientists. In total, they were about 7. They are about 7 or 6 like that, because Oindra's name is twice here, and they have nicely presented their lectures. They have presented their works very nicely, in spite of lots of difficulties. And many of the study, they have taken their research project as challenges. They try to solve it through by using many techniques, including the modern biological research. And in spite of the COVID-19, since its initiation in March 2020, during the time they are involved with research, they have presented their presentation very, very nicely. And the initiative that has been taken by one of my favorite students, the present head of the Department of Physiology, Vidyasagar College, Dr. Adrita Banerjee, is very much encouraging. He used to take such type of initiative. He has taken such type of initiative earlier. I wish that he will get more feedback from this conference, and he will arrange this type of webinars more or less regularly. And I thank all the audience for their patience hearing. And I find Mona Lisa has changed herself much. She is now quite smart. She is now quite active. And in one word, the webinar that has been organized under the leadership of Dr. Subhodip Ganguly is very nice, very excellent. I wish it all success. I hope the comments that has been received by the speakers, by the young speakers, young researchers, will keep it in mind and will try to utilize this in their future work. With these few words, I like to conclude this session is successful. Mona Lisa, please.
Thank you, sir. Thank you. We are honored by your presence today, sir. Okay. So now, yes.
Mona Lisa.
Yes, ma'am.
Hello?
Yes,
Am
yes,
I
ma'am.
audible?
Yes, ma'am. You are audible, ma'am.
In fact, I was in the department now 9:23. That is why I came back to my quarter. So the visual it is not possible because of the poor network. But I am happy that this type of scientific congregation it could be because of all these scientific persons are there. Mostly our Amar sir, he was there. Mostly the guide and the pride of our we should say the nation. And so anyway, thank you very much. I should quit now, no next session or whatever it is. So thank you. Okay.
Yes, thank you, ma'am. Thank you, ma'am.
Okay,
Thank you, madam. Thank you, madam. Your cooperation has enriched us.
Thank you, sir. Sir, namaskar, sir.
Okay, so now we will move towards our third technical session. And in this session, we are honored to have with us Professor Debashish Bandyopadhyay, Department of Physiology, University of Calcutta, and Dr. Barnali Roy Basu. Professor Debashish Bandyopadhyay is well known worldwide for his work on melatonin since last few decades. Moreover, he has also made an impact in the field of development of new drugs and antioxidants of synthetic and natural origin. He has national and international collaborations and has published more than 120 research papers till date in national and international peer-reviewed journals. Professor Bandyopadhyay is a regular reviewer of numerous leading journals published by Elsevier and other internationally renowned publishers. He is one of the editors of the journals Cardiovascular Regenerative Medicine and Melatonin Research published from USA. It is our honor to have you today, sir.
Yes.
And Dr. Barnali Ray Basu is currently an assistant professor in the Department of Physiology, Surendranath College. She is actively engaged in teaching for the last 17 years, with 14 years of postdoctoral research experience and several national and international publications. Dr. Basu is now involved in research in the field of metabolic, endocrine, and reproductive physiology of women, neurodegenerative diseases, nutritional biochemistry, anthropometric survey, stress physiology, ethnicity-based polymorphism study of human disease. So I would now like to request Professor Debashish Bandyopadhyay and Dr. Barnali Ray Basu to kindly chair this session.
Yes, thank you, Monalisha. Can you hear me?
Yes, sir. Yes, sir.
Okay.
You are audible.
So first of all, I like to thank Dr. Shubhodip Ganguly, head of the Department of Physiology, who has invited me to chair the session, and also like to thank Principal Sir Dr. Kundu for allowing Shubhodip to organize such a wonderful webinar. And really, I was from the beginning attending the webinar and enjoying every moment of it with young researchers presenting their research findings. I also am happy to have my co-chair, Dr. Barnali Roy Basu, whom I consider one of the finest young generation researcher I have seen, with so much energy and so much intellectually handling projects and the research students. In fact, I am very happy to see some of our students working in our laboratory. So with this few words and thanking all the participants, I like to begin the session because it is almost 9:30. So I would like to request presenters to keep a watch before them and try to finish your presentation within 8 minutes time so that we can have 2 minutes for discussion or questions. And because, so much so that we can finish the program by 10:30. So Monalisha, I would like to request you to, call the first presenter of this session.
So I like to convey my thanks to the organizer for inviting me in this. It's a great honor to me, and also obviously to the principal sir and Shubhadeep. And it's really a privilege for me to share the session chair with you, and also the previous session aims the Professor Amol Chandra with such leading scientists and professors. It's really a lesson for me for ahead. And just I want to say a few words as it's the time is approaching to the dinner. So that the theme of this webinar is really very interesting, and because if we look at the timeline of the history of the research, we can see that the majority of the breakthrough research discovery actually happened during the First and Second World War. Actually, the necessity is the mother of invention. So innovation, the innovation term, and really the innovation ideas is actually turned in reality when it's really requires. And now the time is a war between a virus and the whole human population. It's really a great war, rather bigger than the world war. So definitely the recent time is the time for challenges and also the time for innovative research ideas and reshaping the research. And today's speaker, the eminent speaker, Shayan, is very nicely titled his topic, a 30 kb RNA brought the world to its knees. So we, the human population, have 34 GB of haploid genome. So that is about 34 into 10 to the power 4 kb of genome. So why we could not face the challenge? And we definitely do have to able to do the innovative research, and our real workforce are there, our dear students and scholars who are the powerful missionary of this innovative research and ideas. So definitely the actual success of this webinar will be through the hand of these powerful researchers in the today's webinar. So for this word, I like to say Mona Lisa to start the session, and also as said by Devashish, the presenters should restrict their time within 8 minutes. Now call the first speaker.
Okay, thank you, ma'am. Thank you, sir. So now I would request our participant to start their oral presentation. Our first participant for this session is Truptimoyi Roth from Laboratory of Medical Microbiology, Department of Biotechnology and Bioinformatics, Sambalpur University. The topic is citrate utilizing Nestorenconia ethiopica.
Am I audible, ma'am?
Yes, you are audible.
Whether my slides are seen?
Yes, your slides are visible.
Okay, thank you. Good evening to everyone who are present over here. I am Truptimoyi Roth, a PhD scholar of Sambalpur University from the Department of Biotechnology and Bioinformatics under the supervision of Dr. Sparanika Patnayak. So today my topic is citrate utilizing Nestorenconia ethiopica. Let's come to my introduction part. Actinomycetes, which are the gram-positive bacteria known to produce a wide variety of industrially and medically relevant compounds like antibiotics, chemotherapeutics, fungicides, and herbicides. Actinomycetes shows both the characters of bacteria as well as fungi. So the organisms are able to produce thousands of metabolites due to its typical lifestyle. The organisms require more amount of carbon source for their metabolic activity. So this present communication is defined with the studies on screening for utilization of citrate in the growth medium. As the citrate is the main source of carbon, as it is provide carbon source as it provides energy to the actinomycetes. So here I am using citrate as the main carbon source of energy for the actinomycetes. For this purpose, citrate, which I have used 2.4% in the growth medium, and observed that there was absolute utilization of citrate by the soil, which was isolated the novel strain named as Nesterocomia aethiopica, which is an actinomycetes. In materials method, I have collected the soil sample from the pond in Sambalpur district of Western Odisha. Here I have used a 4-plate dilution method followed by Patnayak 2018 was used for isolation of actinomycetes. Here I have used 1 gram soil sample which was mixed with distilled water, and a series of dilution was made. Then the diluted soil water was inoculated into the 0.2% actinomycetes isolation agar, which was incubated at 30 degrees Celsius for period of 3 days or 72 hours. The appearance of powdery colonies on agar plate were considered as actinomycetal growth. And then the colonies were further subcultured and optical microscopic studies were also made. But still, 16S RNA sequencing was carried out to identify that specific strain, which was after 16S rRNA sequencing, it was identified as Nesterocomia aethiopica. This actinomycetes shows some bioactivity against multi-drug resistant strain of E. coli. Here I have used 0.028% of nutrient agar along with 1 isolated colony of E. coli. And the actinomycetes named as Nesterocomia aethiopica at its optimum growth, which I have incubated for 5 days, was showing some bioactivity against the notorious multi-drug resistant strain of E. coli. Then the actinomycetes species was screened for its bioactivity against multi-drug resistant strain of both gram-positive and negative bacteria. I have used for this gram-positive and negative bacteria like Salmonella, Klebsiella, Staphylococcus. I have used these bacteria for showing its bioactivity against these bacteria. And it was observed that there was remarkable growth inhibiting activity of actinomycetes. Actinomycetes shows zone of inhibition against these bacteria. Hence, the actinomycetes was studied for its growth requirement, which supports the optimum growth and multiplication of the actinomycetes, so that it could be produced more and more antibiotics which have biopotency importance. Therefore, a citrate utilization test was performed to define the citrate utilization and biopotency efficacy of isolated actinobacteria. As actinobacteria require more and more amount of carbon as the source of energy, that's why I have used citrate utilization test. Whether the bacteria utilize that citrate as the sole source of energy or not. As we know, gram-positive bacteria utilize citrate for their proper growth. That's why I have used citrate utilization test to know whether this bacteria utilize citrate or not. If it does not utilize citrate, then one experiment which is called citrate utilization test, then the color changes will occur. For that citrate utilization test, this is the methodology part. Here I have used 2.4% of citrate agar, which was mixed with the boiling agar, which was mixed to boiling until the agar was dissolved. Then it was poured into 5 ml test tubes. Then the test tubes were autoclaved for 15 minutes. And then the test tubes were cooled in slanting position. After that, the color of the medium was deep forest green due to the pH of the sample and the presence of bromothymol blue, as it is an indicator reagent. Then the tubes were incubated at 37 degrees Celsius for 5 days. After 5 days, I have seen a color change from green to blue color, which indicates the positive reaction of citrate utilization by the bacteria. These are some of the results and discussion part. This is the actinomycetes strain, which I have isolated. This is the image of optical microscopic image. Then in this part, this green color shows the negative growth of the bacteria, and this blue color which shows the positive growth of the bacteria, where citrate is used in the agar media. This blue color shows that the citrate was properly utilized by the bacteria. That's why this blue color is shown. After 5 days of incubation, it was observed that there was a color change from green to blue color, which indicates the pH changes and citrate utilization activity of the actinomycetal strain named as Neisseria ethiopica. This further also added the citrate agar is used to test an organism's ability to utilize citrate, whether as a source of energy or not. The medium contains citrate as the sole carbon source bacteria, and that can grow on this medium to produce an enzyme named as citrate permease. And this citrate permease enzyme is capable of converting the citrate into pyruvate. And when this pyruvate enters into the organism's metabolic cycle for the production of energy, then the growth occurs in the bacteria, which is the indication of the blue color. And this citrate utilization mainly occurs at the intermediate metabolite in the Krebs cycle. When the bacteria metabolize citrate, then the ammonium salts are broken down into ammonia, and that ammonia mainly increases the alkalinity. So these changes in pH turns the blue indicator from green color, that is mainly due to the changes in pH above 7.6. That's why the green color changes into blue color, due to this increasing in alkalinity of the medium. And come to my conclusion part. In this study, reveals that citrate utilization activity and bacterial growth inhibitory activity revealed by the test actinomycetal strain. The citrate utilization by the test actinomycetes may be considered as an important parameter for the growth of the biopotent actinomycetes, as the citrate is utilized by the actinomycetes so that it can produce more and more antibiotics and secondary metabolites, because actinomycetes are the main producer of many and many antibiotics. Thank you. This is my conclusion part. Thank you.
Yes, thank you for your very nice presentation. And session is now open. If someone wishes to ask you some questions.
Okay.
Bornali, if you want to ask something.
Okay.
No, actually, if the time
Yes,
is
ma'am.
so time constraint,
Yeah.
I want to know some specific thing, but I think I could contact through Shubhadeep in future now because I want to know some detailing about it. So this is not the time for that. Not so much time we have. So if nobody has any question, so we can proceed to the next speaker.
Yes,
Bonali,
Thank you.
I have one specific
Okay,
question.
Shubhadeep have a question.
Why should we go for citrate? Is it thermodynamically favorable?
Yes, sir. It is thermodynamically favorable because actinomycetes can grow in hot conditions and it can survive about 4 to 6 months in hot
No.
conditions.
I am working with actinomycetes for last 14 years.
Okay.
I am specifically asking you that is it thermodynamically favorable so far as metabolism is concerned?
I cannot hear you properly. Sir, can you ask again?
Is it thermodynamically favorable so far as the metabolic process is concerned?
No, sir, I cannot give that answer. I have just started my work on actinomycetes and this is my initial work. So I can give you that answer if I further work on actinomycetes. I have no so much idea about actinomycetes. These are my initial work. About 2 to 3 months I have worked on actinomycetes. That's why I cannot.
Okay, thank you.
Okay.
Thank you, Truptimoi. Mona Lisa, could you please proceed to the
Yes,
next speaker?
Yes, ma'am. So our next participant is Debroop Shen from Vidyasagar College. His topic is a study on strain development and bioabsorption of microorganism to alleviate environmental lead toxicity.
So can I quit, ma'am?
Yes, you can.
Sure.
Okay, am I audible?
Yes, you are audible.
Okay, is the slides are
Yes.
available to you? I don't know.
Yes, the slides are visible.
Okay, good evening, everyone. Without delaying any much time, I am Debroop Shen. I am a faculty member of the Department of Zoology, Vidyasagar College. And the topic of my today's presentation is a study on strain development and bioabsorption of microorganisms to alleviate environmental lead toxicity. The work has been supervised by Dr. Shubhadeep Ganguly, Assistant Professor and Head, Department of Physiology, Vidyasagar College. To give a brief introduction to the subject, actually, just we know about lead, we have heard about lead, and lead is a naturally occurring, it's a bluish gray, commonest heavy metals, and it is present in the earth crust at an average concentration of 13 milligram per kg. Though it is in small amount, but it is the commonest one among the heavy metals. And it shows variety of uniqueness, like the low melting point, softness, and this ductility and malleability, and has certain corrosion resistance property also. That's why it has been used from very early period of the human civilization, because it can be molded or melded in certain forms and can be used. So that's why it find uses in the civilizations from the earlier period, but it has no significant biological role in the human system. So whatever we acquire, whatever we get into our system is actually stays within that, and without having any functionality, rather it's hazardous for us. So all the lead occurs naturally in the environment, but rather the industrial revolution has pushed up its concentration on the earth or rather than the environment because of such as fossil fuels combustions, mining and manufacturing industries. This has actually released a lot of lead into the environment. Now, coming to the inorganic lead, it is undoubtedly one of the oldest occupational toxins, and reports or rather certain references are observed even 5,000 years ago about lead poisoning. So it's not new to us that lead causes toxicity. And because of that, because at the earlier phases of civilizations, lead find several uses even in the food, traditional medicines and all. So that's why they use lead in the wine industry to sweeten it up, and that causes lots of poisoning at that time. Now, lead is regarded as a priority pollutant because it is non-biodegradable. So, and the toxicity and the persistence in the environment is always there. So that's why it's a priority pollutant for us. Now, what are the sources of the lead exposures? We come to that. Lead has many different industrial agricultural uses, I said. But nowadays, most of these things are banned in the developed countries. But still, it is currently used in the production of the lead acid batteries, then ammunitions or the warfares, and certain metal products like soldering or solder and lead-based pipes, which are still used in the developing countries at least. And the devices like this, the shield which are used in the X-rays, they also contain lead. And it, I mean, after wearing and tearing of these materials, lead is easily available in the environment, or it comes as a form of dust and other compound. Now, the main sources of lead for the children's environments are diet, like the lead-based paint in the older housing. They leaches out from there and it comes into their food or other dust particles in there, and because of their peculiar habit of putting hands into their mouths, they accumulate a lot of lead in this way. And so lead is soil and dust from contaminated leaded paint and the gasoline, or in the past or present mining and industrial activity also give rise to this kind of toxicity. And the major sources of lead exposure for the adults or individuals is leaded gasoline. We used to mix lead in the gasolines in the form of certain organic lead compound like the tetramethyl or tetraethyl lead. These were used because it provides certain advantage for the fuel to slowly burn. So there is a lesser knocking of the engines, and in order to do so, they mix lead in the gasoline. Nowadays, these are banned in most of the developed countries, but still it persists in the developing countries. And then their source of lead is also from the pipe drinking water, from where it leaches out when the water is soft or a little bit acidic. And then lead is also present in ceramic containers where it has been used as ceramic glazes. So it can leaches from there when hot chemicals, hot materials are poured into it. And soldering from canned foods or the lead-based paint, and certain traditional medicines also give to this kind of exposure to us. So what are the potential for this human exposure and what will be their consequences? Well, lead cannot be degraded, that I have mentioned, so it to generate non-toxic products, so it cannot be degraded to that. And it can accumulate, and it will go for biomagnifications inside our body, and so that is through the food chain itself. And exposure to lead, it occurs mainly by inhalation of the lead-contaminated dust particles as aerosols or ingested, or other ingestion of lead through this contaminated food, water, or paints. And there is absorption rate of lead through the drinking water for the adults are near 35 to 50%, whereas for the children it is a notch higher. It is near about 50% and even more than that. And as per WHO recommended in 2006, the safe permissible limit of lead content in the drinking water stands at 0.01 ppm. But even though there are certain other organizations who have also given such regulations, like the permissible limits for lead, for example, US Environmental Protection Agency has stated as 0.015 ppm, and the BIS or the Bureau of the Indian Standards as similar as the WHO. But still, there are reports that even below that concentration, lead can cause toxicity. So presently, there is no such particular limit for lead that can be designed as safe. So even a scarce amount of lead inside the body will cause toxicity, and that's why lead is very much toxic for us. So lead absorption is influenced by factors, certain factors like the age that I mentioned, that the children are much more prone to lead toxicity, and as well as the physiological status. I mean, a pregnant woman or children, they are much more prone to lead toxicity in compared to an healthy individuals otherwise. So in the human body, where they are mostly accumulated, the greatest percentage of the lead, it is taken into the kidney. That is followed by the liver or the other softer tissues like the heart or the brain. But however, the lead is in the skeleton represents the major body fractions, because lead mimic calcium, and wherever they find a way out in, I mean, they just simply replace calcium storage with this lead, and that's why lots of calcium leaching occurs inside our body, and the storage of calcium is depleted. And long-term exposure of lead, because of this certain kind of mimicking natures and all, it leads to several health issues such as damage to the nervous system, and then it prevents in the hemoglobin synthesis by interfering with certain enzyme activity, And it also causes kidney damage because it generates lots of ROS because of the leaching of calcium, and ultimately giving rise to the reactive oxygen species and cell damage. And then it also affects the reproductive capability of individuals. Now, lead absorption is increased considerably with fasting or in persons whose diet is insufficient with certain essential minerals like the calcium, iron, phosphorus, or zinc. So if someone is anemic or something like that, he or she is much more prone to lead toxicity. To give you an overall view of that, what leads to next and what leads to the ill effects of lead. So the environment and the occupational sources are like the leaded pipes, paints, and soils, and all these things, and the gasoline mixed with lead, and that actually increase the body burden of the lead. Ultimately, it giving rise to ill health effects like the neurological disorders, the IQ loss or mental retardations, oxidative stress, reduced body store of the essential elements, as what I mentioned, and then it ultimately gives risk of the cancer and increased blood pressure, anemia, and other effects. So how can we remove this kind of a toxic metal? Not only lead, even the heavy metals we need to get rid of from our aqueous system so that it does not contaminate. So the traditional methods for heavy metal removal has been like the chemical precipitation, it's ion exchange, or the membrane filtration process, as well as adsorption on alum or kaolinite. But all these methods are not cheap. It is very much cost. It has huge cost because of this chemical we need to use, and it can generate a lots of secondary waste also. And it is not at all effective at low metal concentration. So you can use it whenever the metal concentration is high, but low metal concentration, these are not much useful, and they are not at all suitable for in vivo treatments if you have lead toxicity inside your body. So we need some alternative methods. For treating ourselves when lead toxicity occurs inside the body, it's like certain chelating agents like the EDTA or the DMSA. They can remove lead from the body effectively, but still there are questions regarding their safety and efficacy, and it is only administered when the lead toxicity is at a very higher side. Otherwise, it is not. So we need some eco-friendly, safe, and a novel method that need to be developed for this procedure so that we can eliminate lead toxicity at the source, not even letting it inside your body. In recent years, bioabsorption, it appears to be an emerging and an effective alternative in this concern. Bioabsorption, it's a biological remediation process. It helps in the pollutant removal using biomaterial surfaces. That's why it is cheap, it is efficient, and it is environmental friendly process. And the predominant mechanisms which are involved in bioabsorption is ion exchange, covalent, non-covalent interactions, complexation, and surface precipitation. What are the advantages of this process? Well, use of naturally abundant renewable biomaterials, it can be cheaply procured, ability to treat large volumes of wastewater due to the rapid kinetics of their surface and all, and high selectivity in terms of removal and recovery of the specific heavy metals. So they can handle multiple heavy metals at the same time with the mixed waste also. So high affinity and the reducing residual metals to below 1 parts per billion in many cases. So it's very much effective in certain cases also. It has been reported. And then there is less need for additional expensive reagents, which is mostly in case of the traditional methods, and there is no such and it reduces the problem of disposal and space problems at the same time, and relatively low capital investment, and that gives rise to the low operational cost, and the greatly improved recovery of the bound heavy metals by desorption from this biomass also help to regenerate the biomass again and again to be used as a remediation. And it greatly reduces the volume of the hazardous waste produced ultimately. So what is the aim of my present study? Well, this investigation was aimed to isolate a suitable microorganisms from an aqueous medium contaminated with heavy metals and to develop a lead-resistant strains for effective bioabsorptions and bioaccumulation. The materials
sorry,
and methods that I have
for interruption. Interrupt you already 11 minutes past, so please precisely.
Okay, I'm moving very fast as I can. Okay, the sample has been collected from the Taliganj area, and the isolation process is according to the standard protocols. I'm not going much into it. We have isolated 35 bacterial, 9 yeast, and 5 filamentous fungal strains by using bactericidal agent and fungicidal agent respectively. And then we have these isolated strains. They were transferred to the 100 ml yeast protein dextrose broth, and then it contains 10 ppm of this cadmium, lead, copper, aluminum, arsenic, fluoride, and nickel for our preliminary studies. And we find eventually a strain which is named as JIC, and it was found to be most successful to remove all the selected toxicants from the aqueous medium at a formidable rate. And the percent removal was measured by this formula. And so then we move for the morphological biochemical analysis of the isolate, and that isolate was tested for colony morphology and some biochemical features like the starch hydrolysis, production of ammonia from urea, utilization of citrate, nitrate reductions, or carbohydrate fermentations, according to Benson. And then we move for the molecular characterizations or for creating the phylogenetic analysis of the yeast isolate. So first, the genomic DNA was isolated, followed by the PCR amplifications using the ITS1 and the ITS4 primers. And ultimately, the product of the amplicons was subjected to gene sequencing. And the sequence that we have obtained is it was aligned with the other sequences by using the BLAST. And then it was under the cluster W software, it was aligned primarily, and then phylogenetic analysis were done using the MEGA-10 software. And the phylogenetic tree, it was constructed based on the evolutionary distance calculated from the number of base substitution per site of the isolate by the neighbor joining method using the maximum likelihood composite model. Now, this is the results. What we have found. We have found that the JIC strain can successfully remove certain selected metal and non-metal compounds. As you can see, among them, lead is also being there with a good amount of percentage removal. So I proceed with lead. I'm trying to develop a lead resistant strain with it. And the morphological and the biochemical characterization of this isolate is as follows. And this is the genomic gain isolation data that was subjected to the PCR amplification, which gives a good crisp band without any contaminations. And then we have sequenced this amplicon and with the forward primer of the ITS1 region, which gives us this faster sequence. We have aligned these things and we have done the phylogenetic reconstruction using the MEGA-10. And the accession numbers that we have obtained is MZ7989001. So to come to the discussion part, I've just started my work, to be honest, and that's why I've just isolated a suitable microbial. I have that isolate, and this isolate appeared to be a suitable one for the effective bioabsorption study, where metal and metalloid and non-metallic pollutants were also removed. So it can go, we can further move along with any one of them. And now development of these resistance strains that I have to do and commutate for different pollutants can be recommended. And then on the basis of the preliminary analysis, the isolate JIC looks promising for lead bioremediation along with others. And we have successfully performed the morphological and biochemical along with the molecular characterizations, and we have constructed the phylogenetic tree for the strain. Now we have to do the isolation, characterization, and development of the lead-resistant microorganisms, and to design a suitable method so that I can generate an alternative, cheap, and eco-friendly method for lead bioaccumulations. And the study of the kinetics and other molecular mechanisms underlying this bioremediation process need to be done in the near future. These are the references. I would like to express my gratitude to my supervisor, Dr. Shubhadeep Ganguly, as well as I would like to acknowledge the Dr. Gautam Kundu, principal of Vidyasagar College. And I would also like to acknowledge Dr. Ankur Bhawal. He is the head department of the zoology, along with my colleagues and other participants. And sincere thanks to the institutes who have provided me certain technical supports and with certain chemicals when it was required. And finally, it is my pleasure and honor to acknowledge the Department of Physiology, University of Calcutta, for allowing me to conduct this work. Thank you.
Thank you for your nice presentation. And I like to request audience if they can have some questions, or Dr. Basu, if you have some questions to ask him.
Thank you, Devdu, for your this illuminative presentation. I just want to know a particular point that normally the toxicity, the cadmium, the lead is present as a valency 2 as a toxic, and the valency 4 is I think is a non-toxic one. So is there any changes of valency during this bioabsorption?
Valency 4 is not that much of stable. That what I have read. I mean, mostly lead, what we get in the inorganic form is lead Pb+2. So in certain organic cases,
is it only the absorption, or it is a conversion of toxic to non-toxic form also? That actually I want to know.
Sorry, is it just the form
Is
of
it just absorption by the microorganism, no, or the absorption as well as the conversion of toxic lead to the non-toxic form also?
Well, we have to study for that. I mean, definitely, because we need to first know how lead gets inside these microorganisms. So we have to study the surface chemistry, also the kinetics for them. So unless and until if it's an absorption, just it's on the surface, it's residing, then we cannot say that the conversion of this lead is occurring over there. It's just
Exactly.
a mere of the
That also requires some specific type of enzymes for this valency conversion.
Yes.
So that
definitely
enzymes need to know
where does the lead go. Sorry, please. Yeah.
Thank you.
So first we need to know.
May I say something? Bonalidi?
Yes, Subhadip.
May I say something? Yes. Actually, so far as multivalent species, ionic species are concerned, so far as the bioremediation process is concerned, reductase enzyme plays a central role, especially whenever we'll go for eukaryotic organism, and if there is any sort of multivalent species, reductase enzyme will play a major role. It may convert toxic to non-toxic form and ultimately pumped out from the cell. So it will take the toxic form, then convert non-toxic form, then pump out from the cell.
Yes,
for chromium, it is very prominent. That is why Bonalidi is asking about this. Okay.
Yes. That is a very important issue also.
So far as bioremediation process is concerned, it is very, very essential.
Yes. Okay. Thank you for this actually this kind of work is very essential in the concept of this today's world, because lead is everywhere. So in digital era, lead is a big problem. Everything is a lead cell or like this. Thank you for this.
Thank you.
So I think we can proceed to the next one.
Yes, sure. Monalisha.
Yes, sir. Yes. So our next participant is Sushmita Nayak from Laboratory of Medical Microbiology, School of Life Sciences, Sambalpur University. The topic is resistance to E. coli BME1 towards Ricinus communis leaf extract.
Hello, ma'am. Is it visible?
Yes, you are audible. Nice.
Yes. Try to restrict you within 8 minutes, okay?
Yes, ma'am. Yes. So it is visible or not?
Yes, visible.
So good evening to all. Myself Susmita Naik. I'm working in the bacteria or microbiology under the supervision of Sparanika Patnayak, Dr. Sparanika Patnayak, in Laboratory of Microbiology, and this is Department of Biotechnology and Bioinformatics, Sambalpur University. So my topic is to resistance of E. coli towards Ricinus communis leaf extract. So nowadays the drug resistance is a very global concern. So in that particular part, first one, the drug resistance, that one, the antibiotic resistance is the adaptive change in bacteria that allow them to grow in presence of drug that would normally slow their growth. Multi-drug resistance in bacteria occurs by accumulation on resistance plasmid or transposon of gene, with each coding for resistance to a specific agent, and due to the action of multi-drug efflux pump, each of which can pump out more than one drug type. Then the next one. So here, this one, the Escherichia coli, which is gram-negative facultative anaerobe rod-shaped bacteria, which belongs to the family Enterobacteriaceae, and it can cause gastroenteritis, urinary tract infection, and neonatal meningitis, and so it may be responsible for the hemolytic urine syndrome. So this is the figure of the Ricinus communis. Actually, this plant belongs to the Euphorbiaceae family, and this one is a soft wooden small tree developed throughout the tropics and warm temperature region. So actually this one is commonly called as the castor plant. This plant and the plant parts like root, seeds and leaves have very medicinal value. The therapeutic potential of the castor leaves are less known. The leaf juice is a purgative, lactagose and immunagogues. In order to introduce castor leaf extract for its medicament, the study was undertaken. So here actually I have used the hydrodistillation method from which I am using the cleavanger apparatus by which the aerial parts of the castor plant or the leaf, mainly the leaves were collected during the commencement of flowering stage in morning session from our locality. So because it is available in large amount in our locality, and after that the leaves are, or this is the amount of the leaves are taken with the ethanol. They are loaded into the round bottom flask of the apparatus. This is the figure, and then the mixture is boiled with water using heating mantle at 70 to 80 degrees centigrade for 5 to 6 hours. Then after the 5 to 6 hours, the ethanolic leaf extract was collected from the tube and stored at 4 degrees centigrade for further use. Next one, the methodology. Actually here I have collected the clinical sample of E. coli from the Vimsar Medical Bulla, which is situated in Sambalpur district. And this one, then it is maintained at our laboratory, that is the subculture are maintained. Then I have taken the hydrodistilled leaf extract, which is collected from cleavanger apparatus. Then sensitivity test was done. Sensitivity test was done against the E. coli by using the leaf extract of Ricinus communis, and again I take the ofloxacin, which is a fluorovinyl group of antibiotic that is used as a standard drug. Because in this topic mainly I actually evaluate the efficacy of this leaf extract or with the drug resistance bacteria. So then it is incubated for 14 hours at 37 degrees centigrade. Then the appearance of zone and the no zone, so that was actually made for the either that may be resistance or the sensitive. So in this way, here these are the methods which is taken for the experiment. Next one, here these are the isolated colony of E. coli. This one isolated colony of E. coli, and this here this is the gram staining or the microscopy study of the E. coli. Next one, here I have found the result. In this result, actually this one, the first plate was the control plate, which is your control plate of the E. coli. The control plate, that is the bacterial lawn of E. coli was done. Against this two, one is the leaf extract was actually put on the agar cup method or the hole, and this one here another one is the ofloxacin antibiotic. But we find that all the bacterial plate or all the bacterial lawn plate, they actually give no zone of inhibition. That's why that one is that gives the idea about the resistance towards both the leaf extract and ofloxacin antibiotic. Actually, this antibiotic. So many paper we have already shown that the lots of the natural products are effective against the different type of the drug resistance bacteria, but due to the mutation some of the strain, maybe the other strains of E. coli, it is your other strains of E. coli that is sensitive towards the leaf extract, but in our locality, which is collected from the Vimsar Medical's Bulla, the clinical sample, that is not effective. The leaf extract was not effective against this type of E. coli. So from my study, the conclusion part is there. From this small fragment of research work, it can be concluded that the test bacterial strain BME1 was resistant to Ricinus communis leaf extract as well as the test antibiotic. The possible mechanism of drug resistance conferred by the bacterial strain E. coli from himself clinical sample could be due to universal drug resistance mechanism, and many research work concluded that this leaf extract show inhibitory effect towards the bacteria, but due to different localities, this leaf extract was no effect on the particular strains of antibiotic resistance E. coli. So therefore, additional experimental analysis are suggested for getting better results. These are my references. Thank you all.
So, Sushmita, you have presented nicely, and
Yes, sir.
if audience have even any question, they can ask you. Or Dr. Basu, if you like to ask you some question.
No, it's very good presentation. Just I want to thank Shushmita for finish it before the time. Thank you.
Yes,
Thank you, Monanita, please.
Thank you very much. Thank you very much.
Shall I quote, sir?
Yes. Yes.
Okay.
Mona Lisa, you
Yes,
can next.
Yes, sir. So our next participant is Madhuri Dotto from Department of Physiology, Vidyasagar College. Her topic is cardioprotective effects of melatonin in diclofenac-mediated cardiac injury.
Is my slide visible?
Yes.
Am I audible?
Yes.
Okay. Shall I start now?
Yes, I think so.
Yes, you can.
Okay. I'm Madhuri Dotto. I'm currently working under the supervision of Dr. Indrila Chattopadhyay and Professor Devashish Bandopadhyay, and today I'm going to present my topic, cardioprotective effects of melatonin in diclofenac-mediated cardiac injury. So in our daily lives, stress has become an inevitable part. With that comes several elements of our body that induce oxidative stress in the cells and tissues of our body. So what is oxidative stress? Oxidative stress is the imbalance caused between the generation of reactive oxygen species and the cell's ability to neutralize it by its antioxidant system. Intracellular ROS pool is generated from both mitochondrial and cytosolic sources like xanthine oxidase, monoamine oxidase, NADPH oxidase, etc. And exogenous sources include UV rays, heavy metals, pollutants, ionizing radiations, etc. So, accumulation of excess amount of reactive oxygen species and their consistent imbalance leads to severe oxidative stress in cells and tissues. Under physiological conditions, ROS signaling has a significant role. It has various roles like cardiac development, regulation of vascular tone, maturation of cardiomyocytes, and it has also a role in excitation, contraction, coupling. However, dysregulation in this ROS production has been implicated to have severe effects in several cardiac diseases like heart failure, cardiac ischemia, reperfusion injury, cardiac hypertrophy, etc. So, among various medicines that we use, NSAIDs are some of the most common ones. They are used globally for their anti-inflammatory, antipyretic, and analgesic effects. Millions of people are exposed to its side effects due to prolonged use. In spite of their high efficacy, they have several consequences like gastrointestinal disorders, nephrotoxicity, cardiovascular complications, and these complications loom large, especially over elderly generation. NSAIDs act by inhibiting the COX enzymes, and their administration lowers the levels of prostaglandins, which are important molecules involved in regulating arterial pressure, vasodilation, and inhibits platelet activation that leads to deleterious effects. They also exert their effect on blood pressure by increasing levels of endothelin-1, which is a potent vasoconstrictor and has an important role in mediating acute myocardial ischemia. Besides,
Madhuri, could not hear you. Is there a problem in your sound?
Madhuri Dotto, are you here? Madhuri Dotto, can you hear us? Are you here?
I think something
connection problem.
Some technical problem, technical
Yes.
glitch is there. Madhuri, you can re-enter into the scene. So I think
she got disconnected.
yes. Actually, he is the mid of the presentation, so
Yes.
I think there is some technical issues with Madhuri. In that case, if she is not able to join just right now, you can hear from later.
Yes. Yes. If you permit, then in that case we can call our next participant, and
Yes,
if she
you can call next participant and
yes. You call the next participant.
Okay. Okay. So our next participant is Rondrita Pal, a junior research fellow, Department of Physiology, Surendranath College. The topic is New Paradigms in Research Orchestrated by COVID-19.
Ma'am, am I audible?
Yes, you are audible and visible.
Thank you, ma'am. Ma'am, is it visible, the slide on the screen?
Yes, it is visible.
Thank you. A good late evening to everyone present here, and thank you for bearing up so long. My topic is New Paradigms in Research Orchestrated by COVID-19. The reason we are here today to discuss about COVID-19, the initial talk was regarding that itself. I am Rondrita Pal. I'm currently a PhD student. I'll be continuing, starting the presentation. So the main focus of current times is coronavirus, which most of us might know, most of it or very less of it, however you can term it. So coronavirus is basically a large family of viruses where the novel coronavirus is a new strain which is recently identified in humans, which is creating kind of a havoc till now. It is just a family where many other viruses are also there, where some of are not that lethal, causing some little cough and cold, and some might be lethal like SARS, MERS, COVID-19 currently. So we all know that it was an outbreak in China where WHO identified SARS-CoV-2 as the new type of coronavirus. So in an early Chinese study of 113 COVID-19 cases, it was found there were two strains LNS, and it is very common that virus is supposed to change, or it is very normal for a virus to change or mutate as it infects people. From the virus goes on changing. Accidentally, it is very interesting that 4% of the world's entire research output was devoted to coronavirus in the year 2020. Now, as models of science, we know the basic 6 paradigms as in vivo, in vitro studies, in silico studies, in situ, ex vivo, and ex situ, which most of us already know since the very earlier times. But the point over here is my focus, or rather a new paradigm, can be considered as the in silico study. Why new? Because currently we do not have that time. Because science is proceeding every day and discovering all new stuff. So the faster we predict, the more we can do validation studies, where again in vivo and in vitro studies are inevitable. But for prediction studies, for I would say increasing a little bit of innovation rate, in silico will be a very good parameter also for preventive personalized medicine, where we will find most of us are actually suffering from that. One disease is helping me out, which might not, one drug might be helping me out, but that drug might not be helpful for another person undergoing the same medication, same problem. So there is the importance of in silico studies. Now, this is just a screenshot of PubMed, which we biologists like vouch on for any kind of research. If we simply just go and type in silico and we maintain a time scale, we will see there are around 3,09,323 results. But if we see over here, only in the year of 20 to 21 rather, we will see there are 36,991 cases. So we now we can say that it is gaining a little more importance than before, where transportation or movement was not that difficult. Now, another point, that is COVID. It was much present earlier, as our keynote speaker had already said, but because we are facing such a trauma right now, so has science proceeded, and because of the necessity, here is the results. Within only 2 years, we have 1,76,450 results regarding COVID. So COVID has taken up a lot of research arena. Now, just a basic comparison for in silico COVID studies, we will also get many studies which are coming into account in only these 2 years. Now, pandemic and research, 2020 year of milestones. Over here, there's a list of dates or list of papers and discoveries that I have jotted down. But what I would ask everyone to just focus is on the left, where this 30th April is the date of 2021, and I have just traced the papers backwards. We will find in each of the slides, if we just focus on the left or on the dates of it, we will see each and every day something or the other related to COVID has been discovered and registered. Not only discovered, not only found, something or the other has come into documentation, to be very precise. If we go back, we will see much every day. Every slide will be having just one day, maybe one day, many discoveries have been made. If I trace back, we will be finding in August 2020 or July 2020, India's Bharat Biotech, in collaboration with ICMR, National Institute of Virology, produced and also approved first indigenous COVID-19 vaccine, that is Covaxin. Also, August 22 saw the phase 3 clinical trials of Covishield that began in India. In January 2016, it was the first dose that was administered. So all of this is basically a paradigm, or I would say a result of all the research, of all the hard work of all the biologists working worldwide. Now, one might question, how come where 4 or 5, 6, maybe more than that, it takes years for producing a vaccine, but how come we are making it so fast? Why? There are certain points. Point number 1, it was a worldwide collaboration. We never thought it was my lab, it is my paper, I will not give my idea to anybody else. All the world came together just to find one solution against the pandemic. Second, there was huge funding for COVID-19 vaccine research. Funding is another important point of research. Next was the mRNA technology. I would be quoting Dr. Kenyon, who is the Chief Health Officer at the Project Hope CDC. When challenged with real virus, your immune system attacks the real surface protein of the virus and inactivates it through the immune system capabilities. So that was another technology which was a breakthrough. The next point would be rigorous guidelines for clinical trials. Although it was not possible for a longer time, yet it was always pushed for that. Last but not the least, safety monitoring after vaccination. But even after research, even after COVID-19 being helpful, we do call it as a torrent of COVID-19. Why? Because 4% of world's research was devoted to coronavirus. But we will also see that COVID-19 pandemic has somehow a little bit disrupted science and transformed the research publishing. As per Dimensions database, the data or the number of journals might even have crossed 2 lakhs, whereas basic general based database has scaled to 1 lakh articles regarding the coronavirus only in 2020. Now, beyond that, initial days it was considered, or rather the focus was only on the virus and how it can be sorted out. But after that, the other parameters were taken into account, like public health, mental health, modeling, epidemic, and such attributes. The another very striking point is the China surge. Although it's because it started, or maybe although it started from China, it is very interesting that China has seen a huge peak, huge surge in the publishing of papers. Also, we will see, because the virus has ravaged Italy, much of papers also were swelled from that area. Now, all the concerns regarding research, which is kind of maybe affected by the virus, that is pandemic, might be permanently altering the scientific publishing, which I am not sure about, but there may be a possibility. The second most worrisome part is the COVIDization in distorting research. Rather, I would say it is not only fault of the researchers or the thinkers, or only we are doing it because we want some publications. No, maybe we are trying to see, because there are so many comorbidities, maybe there's some disease might be related. So why not search for it? But some real scientists might even question, maybe the original research is getting distorted because we are just inclined towards COVID. Another very saddening part, being a female, saying this, that many have said that it is costing the career of female researchers in COVID-19. If we focus on the graph given over here, we will see that in case of PhD students, doctors, or professors, paper publishing, at least, or paper submissions, women are little bit lagging behind than men, which is also a point of concern. Some might say it is because they are told, or they are somehow more inclined towards their homes or home care or special aid caring, but that is point of question, which has to be studied. Now, ending my entire thing is the fundamental shifts that we will be seeing, rather we are seeing in case of research, ethics, and also peer review in this pandemic era. First, I'll be starting 12 o'clock, reframing the research design. Second, adapting methodologies to study a specific type. Also, transitioning research mechanics. We are also changing research methodologies, which might affect over here where the in silico point comes into being. Overcoming data collection and standardizing constraints, which is a very big issue because of the pandemic, transport, mobilization has been affected, and so many of us are actually pushing towards how we can just get in general database or anything from the servers, and then we can just bring it down to some kind of comprehensive study. Also, upholding research standard and ethics. So in some cases, we also had to do, like scientists had to somehow cope that negative side. Maintaining informativeness and social value. And last but not the least, provided guarded peer review flexibility. Some sector of scientists also say that much of papers were just released or just in the hope of something new, and true peer review might be affected for that. it's a great pride for me to acknowledge these three institutes because the organizing committee is my alma mater. I started my journey of physiology from Vidyasagar College. So thank you for organizing this. My second hop station was Department of Physiology, University of Calcutta, and where the bug of research came into me, and last but not the least, Department of Physiology, Surendranath College, who's actually helping me out to just increase the bug in size. Hopefully, I'll be something useful for the society through my research. Thank you.
Yes, Adrita, very nice presentation on a
Thank you,
very demanding topic. And I'm really very happy to see what you have said and what I think is important. And I was just telling few, I think 1 hour ago, that I find Dr. Basu as one of the brightest researcher of the young age group. So that is quite reflected what you have said throughout these few minutes. And thank you very much.
Thank you.
And if audience have any questions, they can ask me. If not, then I will request Mona Lisa to proceed for further speakers.
Yes, sir.
Thank you.
I request Mona Lisa to permit Madhuri for the next.
Yes, ma'am. Of course, ma'am.
Madhuri,
Yes.
you please start from
Ma'am,
where you have to.
is my slide visible?
You are just presenting.
Is my screen visible, ma'am?
Yes, it's visible now. You just
Okay.
start.
Okay. So now that we have discussed about oxidative stress, we come to our everyday medicines that we use, and NSAIDs are one of the most common ones among them. They are prescribed globally for their anti-inflammatory, antipyretic, and analgesic effects. Millions of people worldwide are exposed to the various side effects of NSAIDs due to their prolonged use. In spite of their high efficacy, threats of gastrointestinal bleeding, cardiovascular complications, nephrotoxicity looms large, especially over the patients which are from the elderly generation. NSAID administration lowers the levels of prostaglandins by inhibiting the COX enzymes, which are these prostaglandin enzymes are important molecules which are involved in regulating arterial pressure, vasodilation, and inhibition of platelet activation. This leads to severe deleterious effects. They also exert their effect on blood pressure by increasing the levels of endothelin 1, which has an important role in mediating acute myocardial ischemia. Alongside all these effects, NSAIDs also alter the effects of the levels of antioxidant enzymes and increases ROS generation. So we can sum it by saying that NSAIDs increase oxidative stress, induces apoptosis, induces thrombosis, and all these effects leads to various cardiovascular disorders. So one such NSAID is diclofenac. Diclofenac is a traditional NSAID with COX-2 selectivity, which is similar to COX-2 inhibitors. It is prescribed for treating a multitude of conditions like arthritis, osteoarthritis, ankylosing spondylitis, and various other conditions. It is the most commonly prescribed drug in all income group countries and is also easily available over the counter in most countries. Thus, the cardiovascular risks associated with prolonged usage of this drug are of immense importance for public health, and this area needs to be addressed. Studies have also shown that diclofenac possesses the ability to provoke massive oxidative stress. Various other clinical trials have also implicated that patients using diclofenac for prolonged periods of time have increased incidences of myocardial infarction. So in order to find a remedy, we came upon a wonderful molecule that is melatonin. It is a pineal-produced hormone, very well known for its antioxidant, anti-apoptotic, and cardioprotective properties. Melatonin has also several functions in various biological processes like circadian rhythms, neuroendocrine functions, reproduction, etc. At the molecular level, melatonin induces mitochondrial biogenesis, it protects the mitochondrial integrity, and also maintains cellular calcium homeostasis. It also enhances the vasodilator prostanoid pathway that protects against atherothrombosis. In recent times, its protective potential against diseases like atherothrombosis and myocardial ischemia and myocardial infarction have been widely revealed. It has direct action, it also has indirect action, which are both receptor dependent. It scavenges free radicals, it induces the synthesis and activity of antioxidant enzymes. However, we didn't find suitable literature where role of melatonin in diclofenac induced cardiac injury had been studied. So our study was approved by the Institutional Animal Ethical Committee. The animals were kept in ambient conditions in the animal house, and to determine the effective dose of diclofenac sodium that induces cardiac injury, we divided the animals into the following groups. We divided them into control and diclofenac treated groups, where diclofenac was injected in doses 3, 6, and 12 mg per kg body weight for 10 days. After determining the best effective dose of diclofenac, we went to explore the best effective dose of melatonin, for which we divided the animals into, again, 4 groups. Control, diclofenac treated group, where the standardized dose of diclofenac was given. Melatonin treated groups, where melatonin was fed in 10, 20, and 40 mg per kg body weight. And diclofenac plus melatonin treated groups, where at first, the animals were given melatonin in all 3 doses, 10, 20, and 40 mg per kg body weight. And then after 30 minutes, the standardized dose of diclofenac was injected. After treatment, the animals were kept fasted, sacrificed according to the CPCSA rules, and the following parameters were measured: oxidative status and antioxidant enzymes like GSH, GPX, and serum parameters like CKMB, SGOT, and LDH1. So we can see that GSH levels decreased significantly on diclofenac treatment in doses 6 and 12, and after melatonin treatment, GSH levels were significantly restored in doses DM2 and DM3. CKMB levels also increased significantly on diclofenac treatment in all 3 doses, and in doses DM2 and DM3 on melatonin treatment, those CKMB levels were restored back significantly. SGOT levels also increased significantly in all 3 doses on diclofenac treatment. But after melatonin treatment, SGOT levels decreased significantly in all 3 doses, DM1, DM2, and DM3. LDH1 has similar results, where LDH1 levels increased significantly in all 3 doses, 3, 6, and 12 on diclofenac treatment. And after melatonin treatment, in all 3 doses, DM1, DM2, and DM3, these levels were restored significantly. GPX levels also decreased on diclofenac treatment in doses 6 and 12, and they were restored back in doses DM2 and DM3 after melatonin treatment. So we can conclude by saying that significant decline in the levels of GSH and GPX were observed in cardiac tissues. On melatonin treatment, these levels were again significantly restored, and significant rise in serum biomarkers of cardiac damage were also observed in animals treated with higher doses of diclofenac. On treatment with melatonin, these levels decreased significantly in a dose-dependent manner. These are my reference. I would like to thank my supervisors for their endless support. I would like to thank the Department of Physiology, Vidyasagar College, the principal of Vidyasagar College, and the esteemed members of IQAC who have allowed me to present my work. And last but not the least, I would like to thank the audience for listening to me and for allowing me to present twice. Thank you, everyone.
Thank you, Madhuri, for your crisp presentation. And now the paper is open for discussion. If anyone have any question, please put it. If I don't have, actually not a question, but it's a very interesting to me, your topic. As once upon a time, I have to take NSAID like a food, and it's a very positive paper research to me. If the melatonin is a protective against this NSAID and diclofenac, so waiting for your further output and wish a positive come out of your research.
Thank you so much.
Thank you, Madhuri. So, Monal, please proceed.
Okay, ma'am. So our next participant is Abhijit Hajra from Department of Physiology, Vidyasagar College. His topic is isolation and characterization of a novel methionine producer from Mahanadi river site in Sambalpur district of Orissa, India. Novel isolation and characterization of a novel methionine producer from Mahanadi river site in Sambalpur district of Orissa, India, and development of overproducer by induced mutation.
Abhijit, please start your presentation. The screen is visible. Your presentation is visible. Abhijit, we could not hear you.
I think some sort of technical difficulty.
Yes, I think also. So, Mona Lisa, could you please proceed with the next one? And in that time, Abhijit can resolve his problem.
Okay, ma'am. Okay, ma'am.
Abhijit, can you hear me?
I think some sound-related issues with.
I think so.
Hello, sir. Sir,
Yes, we can hear you now. Please start your presentation.
am I able to see my screen?
No, the screen now it is now visible. Start.
Thank you. Good evening, everybody. I am Abhijit Hajra here, MSc in Physiology. I am working under the supervision of Dr. Subhadev Ganguly, Assistant Professor and Head, Department of Physiology, Vidyasagar College, 39 Shankar Ghosh Lane, Kolkata, 700006. Today I am here to present my research work entitled Isolation and Characterization of Novel Methionine Producer from Mahanadi river site in Sambalpur district, India, and development of overproducer by induced mutation. Introductory part. L-methionine has been widely used as a food supplement, and it is an essential amino acid. It is not produced in our body. It is intake in our body through diet. Vegetarians are highly suffer from L-methionine deficiency due to insufficient content of food. It is noted that methionine deficiency development of various disease and complications. It may have led to Parkinson, liver deterioration, hair loss, muscle paralysis, depression.
Slide is not changing. Abhijit, slide is not changing.
No, we just seen the first slide, the title slide only. I think you have to re-enter into the slide. If we are sharing your slide from the laptop, you just close it and re-enter, reopen it.
Am I able to see?
Yes, you are audible, but the screen is still not here.
I think some sort of technical issue. Bonalidi,
Slide is not visible. Yes.
I think technical issue. So, shall we proceed orally?
Yes, he can. If he is comfortable with that, he can.
Okay.
Or if he just share it to Mona Lisa. Mona Lisa can.
It would be better.
Share it. Yes.
It would be better.
Just share it to Mona Lisa, Abhijit.
Abhijit, send it to me immediately.
Yes, sir.
This type of issues are quite natural.
Actually, he is from East Midnapur.
So might be the broadband width or the network width is a problem.
A lot of connectivity problems are there because of the weather conditions also.
Yes.
I think if he is comfortable orally, then shall we proceed, Devashish?
Yes, yes. If he is comfortable,
he can. Yes.
It is possible. You know,
nothing. Time is pressing.
Yes, yes, yes.
Abhijit,
you please summarize your work. We are very eager to listen to you. And the slides may not be that much important. We will be happy to learn the main findings you have with your observations.
Am I audible?
Yes, yes, yes. You can continue.
Working under the supervision of Assistant Professor and Head Radiology, Vidyasagar College, 39 Sankar Lane, Calcutta, 700. Entitled Isolation and Characterization of Novel Methionine Inhibitor from the in some and producer by induced mutation. But it has been widely used as a food supplement and it is an essential amino acid. It is used in our body through diet. Vegetarians suffer from iron deficiency due to insufficient content of food. It is noted that methionine deficiency development of various disease and complications. It may have led to Parkinson, liver deterioration, hair loss, muscle paralysis, depression. These symptoms overcome through adding methionine in the diet. The Complementary Medicine Evolution Committee suggests that methionine is a suitable dietary supplement and for therapeutic use. Nowadays, the global market value for L-methionine is gradually increased from 2015. The major producer of MSA are based in Japan, United States, South Korea, and Europe. Where it is reported that Asian-specific countries like China, India, Vietnam, and Philippines are major consumers, especially for it use dietary supplement. The global market and to reach $7.6 billion by years of 2015.
Excuse me, with your permission, sir and ma'am, I want to, can I share the slide, Abhijit's slide? If it would.
If you are ready, you can share.
Yes, ma'am. Thank you.
Okay, Mona Lisa, you got the slide, I think. Now you can show it.
Yes, sir.
Okay, that will be better for you.
Yes. Are you in the introduction slide, Abhijit?
Yes, ma'am.
Yes. Okay.
Thank you.
Please continue.
Starting, ma'am?
Yes, you can continue.
L-methionine has been widely used as a food supplement, and it is an essential amino acid. It is not produced in our body. It is intake in our body through diet. Vegetarians are highly suffer from L-methionine deficiency due to insufficient content of food. It is noted that methionine deficiency development of various disease and complications. It may have led to Parkinson, liver deterioration, hair loss, muscle paralysis, depression, and these symptoms overcome through adding methionine in the diet. The Complementary Medicine Evolution Committee suggests that methionine is a suitable dietary supplement and for therapeutic use. Please next. Nowadays, the global market value for L-methionine is gradually increased from 2015. The major producer of amino acid are based in Japan, United States, South Korea, and Europe. However, it is reported that Asian Pacific countries like China, India, Vietnam, and Philippines are major consumers, especially for the huge dietary supplement. The global market has been forecast to reach $11.6 billion by year of 2015. Please. Previously, L-methionine produced by chemical synthesis or by enzyme hydrolysis of protein, but these processes are quite expensive and it's not profitable. So scientists have found solution for overcome this problem. After the discovery of L-glutamic acid producing strain, the encourage scientists to shift their experiment toward the development of L-methionine producing microbial strain. Unlike other L-amino acid producing strain, in most cases, L-methionine overproduce were not possible happen due to strong product-mediated feedback inhibition process. Several work has been made in the field, but unfortunately, no safety report is available regarding L-methionine overproducer. Next. Next, ma'am. The aim of our study to develop L-methionine overproducer from a newly isolated strain via induced mutation using N-methyl N-nitrosubeguanine and Ovary, and use L-methionine is a L-methionine analog to overcome feedback inhibition. Next, ma'am. We done the work through several methods, and these are presented at first. Sample collection and isolation of bacterial strain from Sambalpur district near Hirakund Dam, Odisha, analyzes the morphological structure. After that, we study the mutagenic treatment with NTG and UV, then estimate the amino acid production and for confirmatory test for L-methionine, then purification of L-methionine. Due to lack of time, I am skipping methodology portion in details. Please, ma'am. Please, ma'am. Please. Please. Then this side, previous slide, ma'am. This is the result and discussion part. The isolation and bacterial strain for L-methionine production. The colony was tested for production of L-methionine is submerged fermentation using minimum salt medium. We isolate 7-8 strain, and among this strain abbreviation number ATXCT47 appear highest amount of L-methionine producer. In this table 1 shows that 0.6 milligram per ml L-methionine was produced by the wild type strain, which abbreviation number was ATXCT47. Next slide, ma'am. Mutagenic treatment with NTG. NTG used a potent mutagen. It has performed by incubating bacterial culture with different concentration of NTG and temperature. In this study, the central composite design was applied to study the interaction of the various factors, affecting the optimum condition for L-methionine production. Three variables from NTG incubation time and temperature assigned: high level, low level, and medium level. So, table 1. Next slide, ma'am. The central composite design consists of 17 experimental number and shown in table 2. The result shows that ATXCT41 has able to produce L-methionine under all considered conditions. Next slide, ma'am. In this table, the analysis of variance and regression coefficient for the resulting model are presented. This model has a very low p-value, which implied that model fitted the experimental data significantly. The lack of fit to pure error is not significant in this model. Next, ma'am. Here we obtain the optimum value for each factor to get maximum response. This graphical diagram shows that the effect of three variables on the production of L-methionine, the 3D response surface plot, and the 2D control plot that would evaluate the interactive effect on the response, which is shown in the figure 2A, 2B, and 2C. Next slide, ma'am. Ma'am. Strain for L-methionine production. L-methionine is widely used for L-methionine and to develop it resistance strain to overcome product-mediated feedback inhibition, which is shown in table 4. Among 180 resistance strain developed, abbreviation number 86XT3624 produce 25 milligram per ml of L-methionine. Next, ma'am. Next. Determination of specific nutritional previous slide, ma'am. Determination of specific nutritional requirement of oxotrophy for abbreviation number 86XT3624 for L-methionine production. In the figure 4, it has been found that maximum production obtained L-homoserine was 10 milligram per ml. For this study, it was clear that strain of L-homoserine oxotroph. The maximum production obtained with 12 milligram per ml of L-homoserine in this medium, shown in the figure 5. This actually this slide are skip conclusion part, so I can conclude that L-methionine has been widely used as for a food supplement, though its production by fermentation is beneficial, that microbes can synthesize L-methionine is using eco-friendly process. Fermentation of L-methionine has been applied industrial scale due to economically non-viable complex with chemical synthesis. To overcome this problem, alternative method may be two-step process in which L-methionine precursor is produced by fermentation, which then enzymatically converted to L-methionine. A new strain developed by induced mutation. Considering this importance, mutational induction has been adopted as an essential tool to exploit the diversity of microorganisms for industrial production of L-methionine. Next slide, ma'am. Next, ma'am. Here is my reference which I prepared during this study. Acknowledgement for this is keeping in this slide technically fault issue.
Yes, I think the slide, the presentation that I have received here, there is no acknowledgement slide here. Anyway, you can tell it orally from your side.
Yes, I am honored to acknowledge. I am express my sincere gratitude to my supervisor, Dr. Shubhadeep Ganguly, Assistant Professor and Head Department of Physiology, Vidyasagar College, for providing this opportunity to carry out this work. I am heartily acknowledged to Dr. Gautam Kundu, Principal, Vidyasagar College, for allowing me to conduct my PhD work at Vidyasagar College campus. I sincerely acknowledge Department of Biotechnology and Bioinformatics from Sambalpur University, Odisha, and Indian Institution of Chemical Biology, Bosch Institute of Kolkata, and Cultivation of Science for providing necessary instrumental approach. With this, it was very difficult to carry out my PhD work. Thanks to all for listening.
Thank you, Abhijit. Finally, that you could finish your presentation with all good slides and nice presentation. Despite of,
Actually, sir, my network connection is very poor.
Yes, I understand. And weather is also not good at around the coastal part of the West Bengal. So thank you very much. I asked Dr. Roy Basu to, if she has some questions to ask you.
No, actually no question. It's too late, but I said, Abhijit, you need to add a point in your acknowledgement to Mona Lisa Madam. So that's all. So Mona Lisa, please proceed.
Okay, thank you, ma'am. Now our last participant for this session is Ankita Shamadar, Junior Research Fellow from Department of Physiology, Surendranath College, University of Calcutta. Her topic is Scientific Research and Management of COVID-19, Major Advancements and Limitations.
Is it visible, ma'am?
Yes, it is visible.
Okay. Good evening, everyone. Although it is very late, still in respect to the topic of the day, I am going to illustrate my presentation on scientific research and management of COVID-19, major advancements and limitations. I am Ankita Shamadar, Junior Research Fellow of Department of Physiology, Surendranath College, University of Kolkata. SARS-CoV-2 is a novel coronavirus, and its nature, infectivity, and clinical symptoms were somewhat unknown to mankind. So we face several challenges while combating this pandemic. Some of these challenges are successfully resolved, and some challenges are still tormenting us. SARS-CoV-2 was first detected in Wuhan city of Hubei province, central China, when people with unknown pneumonia get admitted into the hospital. SARS-CoV-2 belongs to the beta lineage of subfamily Orthocoronavirinae. Its genome is enveloped, positive sense, single-stranded RNA containing 26 to 32 kilobases. SARS-CoV-2 is greater than equal to 70% similar to the previously epidemic-causing virus, SARS-CoV, but this mild variation from SARS-CoV enables it to cause pandemic by increasing its transmission and fatality rate. Viral genome mutation significantly increases its infectivity, which enables it to cause pandemic waves and hamper human well-being for near about 1.5 years. Now, it is important to understand how and why this virus causes severe pathological and physiological impact. SARS-CoV-2 has 4 different structural proteins: the spike proteins, envelope proteins, nucleocapsid, and membrane protein. Among all these proteins, the spike proteins are the most significant. The spike proteins plays the most significant role in SARS-CoV-2 penetrance into the cell. These spike proteins interacts with one of the most abundant human receptor, angiotensin converting enzyme 2 receptor, or ACE2 receptor, and after binding to this receptor, several serine proteases, most importantly the TMPRSS2, come into play, and the SARS-CoV-2 enter into the cell through endosomes. Presence of SARS-CoV-2 within the cell activates innate and adaptive immune system and cause activation and infiltration of several immune cells such as monocytes, neutrophils, macrophages, natural killer cells, CD4 plus cells, CD8 plus cells, eosinophils, and basophils. Recruitment of this huge number of immune cells causes the release of cytokines mentioned in the slides and chemokines, which is a characteristic feature of COVID-19. This excessive inflammatory innate immune response and dysregulated adaptive immune response is termed as cytokine storm and cause tissue damage both at the site of viral entry and in systemic level. When viral entry occur in type 1 and type 2 alveolar cells, all these mechanisms happens within the lung tissue, which results in acute lung injury and ultimately leads to acute respiratory distress syndrome. But medical sciences as well as the research labs all over the world put the highest effort to combat this pandemic. From detecting the viral particles to treat the disease symptoms, they are defending the pandemic as much as possible. To take immediate action or to manage this pandemic, rapid and accurate viral detection is extremely important. Due to the advancement of modern research approaches, reverse transcripted quantitative PCR is used successfully to detect viral particle and even the viral load. Assistive medicines like dexamethasone, remdesivir are prescribed according to the severity of the disease symptoms. Not only that, supplementary oxygen is also implemented to fight against the acute respiratory distress syndrome. Although there is a lot of effort invested to manage the pandemic, there are several additional threats also that becomes very challenging. This dysregulated immune response causes tissue damage by increasing the production of reactive oxygen species and elevating the overall oxidative stress level among infected individuals. Therefore, some pre-existing diseases like cardiovascular disease, diabetes, obesity, respiratory distress, insulin resistance, which is significantly associated with systemic stress, aggravate profoundly among the infected individual, and these diseases act as comorbidities of COVID-19. Therefore, even if we are successful to combat the infection itself, these comorbidities are increasing the fatality rate to a great extent. Apart from that, penetrance of SARS-CoV-2 into the peripheral and central nervous system via olfactory route causes inflammation in brain tissues, as well it elevates the oxidative stress within the nervous system, which might have affect the blood-brain barrier and might enhances the chance of having cerebral stroke. Even if the infection is over, an individual is still susceptible to several tissue damage. As SARS-CoV-2 can enter into any organ in the human system, as majority of human body cells have the ACE2 receptor on them. After recovery, the elevated inflammatory state in the body exacerbates the lifestyle diseases so much that even if the infection is subsided, the precious lives has been lost, and the overall well-being of the human population hampered substantially. Not only that, inflammation and stress in nervous system can cause rapid progression of several neurodegenerative diseases like Parkinson's disease, Alzheimer's disease, dementia, Huntington's disease, etc. It can also cause the may cause the premature onset of neurological disorder in post-COVID situation. Moreover, other different comorbidities like brain fog or confusing state, joint pain, headache, sleep alterations in menstrual cycle also hampers the well-being of the individual even if they do not have such lifestyle diseases previously. In present scenario, one of the major concern in reproductive health is the polycystic ovarian syndrome. It is a multivariate metabolic and endocrine disorder that not only leads to the female infertility but also correlated with several lifestyle diseases like cardiovascular disease, type 2 diabetes mellitus, insulin resistance, obesity, etc. Oxidative stress play a major role in regulating menstrual cycle and so-called the hormonal homeostasis among individuals. Stress also significantly increase the PCOS severity and the disease outcome. In case of SARS-CoV-2 infection and the subsequent ROS production due to the cytokine storm, the scenario is getting even more worse. Along with this, irregularities in menstrual cycle as a post-COVID complication also overburdens the PCOS disease outcome and infertility. At the end of my presentation, I would like to express my sincere gratitude to the organizers, Department of Physiology, Vidyasagar College, for giving me this opportunity to present my slides and arranging such an informative session. I would like to thank Department of Science and Technology, Biotechnology, West Bengal for my research funding and Department of Physiology, Surendranath College for allowing me to conduct my research there. Thank you.
Thank you, Ankita, for very nice presentation on a very important topic also, because COVID may be in a curative stage within 3 or 4 years, but the post-COVID effects in human society will remain for a decade, I think, a decade or so.
Yes, sir.
So this automatically makes the research in post-COVID management more important.
Yes. Yes.
So that's very nice. And if someone asks some question, or I request Dr. Roy Basu to summarize what has happened this session.
Thank you all the participants for the patience waiting and delivering lecture. It's started from right 5 and it's almost past 11. So very long and wonderful and really enlightening webinar. And really, the group of researchers or the budding physiologists, researchers in physiologists are really, we can see through this that they are really ready to take the challenge as the theme of this webinar for the innovative research. They prove it for this long time webinar, waiting for this webinar. So I like to again thank the organizer, especially Shubhadeep, for organizing and such an important and very interesting webinar, international webinar. And definitely it's an honor for me. And it's, again, I want to say that to share the chair with Devashish is really a privilege to me, and it's a lesson to me also. And thanks, Shubhadeep, for giving this opportunity. And thanks,
You are most welcome, Didi.
Okay. And thanks, Mona Lisa, for this she actually wonderfully conduct the session. I really like it. So also a big thank to her also. Thank you all. A very good night. Have your dinner and a good sleep with an innovative dream also. Thank you. Good night.
Thank you very much. Thank you all for participating and presenting so many different types, so many different kinds, so many different standards of research. Because, you know, India and Indian scientists, particularly juniors, have to be innovative. Particularly research. I hope research will be more and more promoted in the post-COVID management area, because it is now going to have a long-term societal impact. It will not, the impact will not end in 5 or 10 years. It will remain in the society for more than 30 years, I hope. So thank you all. Thank you very much. Thank you, Dr. Ganguly, for inviting us to chair the session.
You are most welcome, Devashish there.
Thank you so much. Thank you so much. And so many of the interesting participants with, co-chairing with Dr. Roy Basu is an excellent experience for me. And thank you, Mona Lisa, for conducting. Most welcome. Most welcome. So I think we should have now time for good night to all.
Good night. Yes.
And end the session. And we hope more such encouraging conferences or webinars at Vidyasagar College Physiology Department by their head of the department, Dr. Shubhadeep. And really, I feel very happy. So many of the...
Really, it's a grand success.
Yes.
Thank you, Dada. Thank you, Bonalidi.
To organize such.
And his approach, his inclusive approach, attracts me very much. It is inclusive.
It proves that the pandemic could not stop the innovative ideas.
The progress of
in
knowledge,
all aspects.
Progress of science, the progress of believing in good quality science cannot be, rather, we have to increase this. And Shubhadeep I find is a right person in this aspect to elevate, the young minds for beautiful research to come in the future days. Thank you all. Thank you very much.
Thank you, Dada.
Thank you. Good night.
Good night.
Thank you, sir. Thank you, ma'am.
Most welcome. Most welcome.
Welcome.
So now I would like to request our Honorable Head of the Department, Dr. Shubhadeep Ganguly, to formally convey his vote of thanks.
Thank you, Mona Lisa. On behalf of the Department of Physiology and our today's organizing committee of this one-day international webinar, I would like to express my sincere gratitude to our today's eminent speaker, Dr. Sayantan Bose, eminent virologist, chairpersons for the scientific sessions, Professor Amol Kumar Chandu, former Emeritus Professor, Department of Physiology, University of Calcutta, Professor Devashish Bandyopadhyay, Senior Most Professor, Department of Physiology, University of Calcutta, Dr. Sparanika Patnayak, Assistant Professor, Sambalpur University, Orissa, and Dr. Bornali Roy Basu, Assistant Professor, Department of Physiology, Surendranath College. Esteemed delegates, participants for oral presentation, my dear students, for their presence and contribution to make this webinar great grand success. I extend my gratitude to my colleague, Dr. Alokos Choudhury, Dr. Chandana Sarkar Banerjee, Dr. Indrila Chattopadhyay, Dr. Sanatan Mishra, Dr. Monalisa De, Ms. Shatabdi Ghosh, for their continuous support, especially some of my student volunteers. I have to mention, like Ankush Kumar Das, Suman Parui, Shubham Chattopadhyay, for their continuous effort, without which it was not possible for me to organize this webinar. Thank you all for your active participation. Thanks a lot.
Thank you, Dr. Ganguly. Thank you all for attending this one-day international webinar. I request you all to please fill up the feedback form through the given link in the comment section for receiving the certificate. See you all in the next webinar, and goodbye and good night for now. Thank you.
Good night, everyone. Good night, Didi.
Good night.
ঐন্দ্রিলা দি এসে গেছে। ঐন্দ্রিলা দি মিউট